with the keynote that is going to look onto the mechanism behind the formation of Lake Victoria Biodiversity Hotspot and on the causes of the troublesome global trend that human poverty is at, and it's worse where nature is the richest. The session will be moderated by Dr. Mary Kishe from Tafiri. For all of us to give her the confidence that we are ready for her, let's welcome her with a round of applause. Tumpige makofi. Karibu.
Thank you, MC. Good morning, everyone. It is my pleasure to welcome you all to this keynote speaker presentation, and particularly I'm honored to introduce the first speaker, Professor Ole Seehausen, who is a highly respected researcher in the field of cyclists, biology, and evolution, but is also a valued member of our institute, a collaborator, and also my mentor on cyclists studies. And Professor Ole Seehausen is a professor of aquatic ecology and evolution, and also is the executive director of the Institute of Ecology and Evolution, University of Bern, Switzerland, but also is a researcher group leader at AQUACULTURE, Switzerland. With more than 35 years of research on the ecology and evolution, as well as conservation of cyclids fish in the African Great Lakes and aquatic biology. Professor Ole has been working on the evolutionary in Lake Victoria and important role of interspecific hybridization in the origin of cichlid species riches and the dramatic effect of Lake Victoria eutrophication. Professor Ole Seehausen is going to give us a talk on mechanism behind the formulation of the Lake Victoria biodiversity hotspots on the causes of the troublesome global trend that human poverty is at its worst, where nature is the richest. Please join me to welcome Professor Ole Seehausen to give us a talk, and Professor Ole, the floor is yours. Please welcome.
Thank you very much. Can you hear me okay? Yes. Mary, can you hear me okay? Yes. Can someone say, can you hear me okay? Is it okay?
Yes, we can.
Okay, thank you very much. Thank you very much, Mary, for the introduction, and thank you everyone for hosting me today. I'm very happy to speak to you. I'm sorry that I cannot be in Dar es Salaam, but it's a pleasure to speak through the internet. So after, like Mary said, after 30 years or 35 years of working on the evolutionary biology and also the conservation and management of the enormous fish biodiversity in Lake Victoria, I've reached a point where I feel that we must turn our science a little bit also into a different direction and try and understand what that biodiversity actually means to people. So the talk today is about this. It's about the origin of incredible biodiversity and how that's possible in Lake Victoria. I have to make that really short. And then the troublesome observation that led us to do some larger quantitative analysis on understanding why people tend to remain poor in exactly those places that have so much biodiversity. Let me see if my slides move. Okay. Now here's a map of freshwater fish, species richness globally. And you can all see the darker red it is, the more species there are. So the richness resides in the tropics. Next to the Amazon and parts of Southeast Africa, it is the Congo and it is East Africa, with especially the Great Lakes region. With lots of species in there, and you all know about that. The cichlid fish in African Great Lakes, it's more than 1,500 species that are currently known. Largest numbers in the 3 big lakes, but there are many other, also very large lakes that also have many species of cichlids in them. And together these cichlids make about 10% of the world's 15,000 freshwater fish species, which is enormous, and 8% of all the fishes of the oceans and freshwaters combined. Most of these fish in these lakes indeed are cichlid fish. Most of them, almost all, live nowhere else than in the one lake in which you find them. More than 500 are endemic to Lake Victoria alone. And I want to speak about why that is stunningly amazing. Lake Victoria now, as you know it better than I do, many of you know it better than I do, it's an inland ocean. It's of course freshwater, but it is an ocean-like environment, it's large, it's vast. It supports the largest inland capture fisheries, or one of the largest inland capture fisheries in the world, and it's home to one of the highest species richnesses in the world. But we shouldn't take for granted that that's how it has to be. In fact, there are predictions that it will stop being like that in the next 100 years. I think some of you have heard about that. They're credible predictions that with climate change, Lake Victoria might actually dry up completely in the next 100 years. Now, this has happened in the past, and it is not very long ago in geological terms. Until 16,000 years ago, Lake Victoria was dry. It looked like this. It looked like the Serengeti, with probably a few shallow ponds and little streams. And there cannot have been much fish diversity in there. In fact, we have reconstructed from genetics and also from fossils that there was just really one type of cichlid living in these environments. And that's a fish that you can still find in those environments. For the insiders, it's called Astatotilapia nupaina. And this fish must have diversified over the last 16,000 years into the amazing community that we know now in Lake Victoria with 500 endemic species of cichlids. And it's like an evolution engine. And I've put it very small there, you see, this is the engine. There's hundreds of species that are being produced at record rate and adapt to their environments in record rates. And that is a little engine that actually fuels a more than $500 million fishery. Because Nile perch is heavily relying on supply of these cichlids, and the entire ecosystem is heavily depending on the continued existence of cichlids in Lake Victoria. So my research group wants to understand how these species exist, what they are, how they came about, how we can define them. They want to understand also what the ecological implications are of such incredibly rapid evolution and such incredibly large biodiversity. But they also want to understand how people interact and how people benefit from this diversity and what is needed to safeguard the continuation of such systems also for future generations. So I'll talk briefly about the cichlids and then about the people. Now, the cichlids, even though they are very, very young, they are very, very distinct in their traits. So here's just a set of a few photographs of Lake Victoria cichlid fish, each a different species in their breeding dress. So each of them is defined by a unique combination of these male breeding colorations and the stripe patterns, whether they have a midlateral stripe or vertical bars or both or none, and the head morphology. Many of them are food web specialists, so trophic specialists that are highly distinct with their morphologies and adapted to feeding on different types of food. So here are just some CT scans of fish heads that show you variation in the oral jaws. So here's a shrimp eater with his very long jaws. Here's an algae scraper with very short and bended jaws, snail eater, and fish eater. And there are many others. They have very distinct oral dentition. So here, for instance, this one has very coarse and widely spaced teeth. It feeds on eggs and larvae. Whereas this one over here, can you actually see my mouse moving? My pointer? Maybe I need to turn this on here. Maybe that's better. This one here is a fish eater that has this really tall, long, and very sharp teeth, and they are algae scrapers and they're snail eaters with different teeth. They have a second pair of jaws in the back of their head, that's the pharyngeal jaws, which they use for crushing. They're sitting somewhere here, behind the gills, that they use for crushing prey. And depending on what prey they feed on, they can have very, very fine bones with fine teeth, or they can have these massive bones with very big teeth. And indeed, they have evolved not just many species, but they've evolved a food web, where one feeds on the other one. They've evolved essentially the key component of an entire ecosystem. This is how the Lake Victoria fish community looked like prior to the takeover of part of the open water community by Nile perch. The explanation for how this all could have happened, I mean, people have worked on that for many years, including people in my group, to understand how this is possible. And this slide that looks a bit complicated is, I think, a visual summary of what we think is needed. It really needs the coincidence of 4 key factors so that you can get so many different species in so little time. It needs a group of organisms like cichlid fish that are ecologically very versatile. They need to have a mating system by which that can evolve very quickly. Like here, the males have these bright colors, the females don't show them, but the females select their males based on the color of their breeding colors. They need to find ecological opportunity, like a large lake with shallow water, deep water, with rocks and mud and sand, etc., where they can do different things ecologically. But all of that isn't enough. It only really works if they have unusually much genetic variation of old types, so not new mutations, but ancient genetic variation that has arisen through hybridization before the lakes formed. We know that now is true for all the African Great Lakes radiations, not just Lake Victoria. Now, over the recent years, we have done quite a bit of work on sequencing whole genomes of these fish to see what they can tell us about the origin. And we have found, and others have found in other lakes too, that each of these radiations starts with hybridization. So this is the radiation of Lake Victoria, and 100 to 150,000 years ago, cichlids from the Nile met with cichlids from the Congo in this area, which captured headwaters of the Tanzanian Congo, the Malagarasi, and the Nile in Uganda. These cichlids were millions of years divergent before they met, but when they met, they merged into a single hybrid population. From that hybrid population, a large radiation evolved. And each species in this radiation is a unique mosaic of genetic variation from the Congo cichlids and the Nile cichlids. But then the lake dried up, as I said earlier. It dried up around 20,000 years ago, and only filled up again 16,000 years ago. And as I mentioned, there was only this Astatotilapia nobila that survived in these swamps. Turns out that this Astatotilapia nobila existed in many places around the lake, and each of them contained the old genetic variation. The red is the Congo and the blue is the Nile. Each of them contained a unique set of Congo and Nile genes. And they survived in the swamps in the north and the south and in the western areas. And when Lake Victoria formed again, they came through the little rivers that were now captured by the new forming Lake Victoria. They met again in this lake. They merged again into a hybrid population. And this hybrid population radiated out into the 500 species that we now know. We can reconstruct that history from genomic data. We can do that not just for Lake Victoria, but for all the lakes in the region, and we could show that each lake in the region has its own radiation. Even the Kagera lakes in the west of Tanzania have their own little radiation. Like Edward Kivu, Albert, Turkana, even Lake Saka, and the big one here, all the yellow ones, is Lake Victoria. We can also show that within the species, even though they're very young, they're genomically very well defined. So each of these little groups here is a species with multiple individuals from multiple genomes that always cluster together. They're really clearly different species. And it turns out that the hybridization that I mentioned to you, that started with 100,000 years ago with Congo and Nile, but then was repeated when 16,000 years ago, the three different populations of Astrata, tilapia, and nubila from different swamps around the area came together, that was probably very influential for the radiation. So when we look at where the different trophic specialists, like the algae scrapers, snail eater, or these egg eaters and these fish eaters, where they got their key genes from, genes that only occur in these particular specialists, we can see that the algae scraper got a lot of genes from the northern population of nubila, whereas the fish eater got this blue area, got a lot of genes from the western population of nubila. And the snail eater got a number of genes unique to the southern nubila. And we can do that over all the different trophic specializations and paint the regions in the genome that define them with the colors of these three nubila populations. And so we see that the trophic diversity has arisen through combination of old genes that came from these fish. We've also shown that these genes can actually be very old. They can be older than Lake Victoria and the entire Lake Victoria region. So we've shown in a paper a few years ago that fish eaters, even from Lake Muero down here, and Kivu and Victoria, they all share identical gene sequences that define fish eaters, whereas the insect eaters share identical regions in the genome that define them. So if you build phylogenetic trees for this chromosome, then you get all the insect eaters from Victoria, Kivu, and Muero look like very close relatives, and all the fish eaters too. When you build a tree from the rest of the genome, you see that that's wrong. You see that the fish eaters of each lake are most closely related to the insect eater of each lake. So that means that these species have inherited old genes that were distributed by migration between lakes and hybridization, and they reuse them to evolve this trophic diversity. We've very recently, with my postdoc Pooja, been working on trying to understand the entire genetic architecture of the adaptive radiation. For that, we phenotype more than 100 different species, and we genome sequence multiple individuals of each, and we look for statistical relationships between all these traits, the color traits, the feeding traits, the morphology, and variants in the genome. When we do that, we can then map, also here this is a tree, we can map all the traits on the tree. So for instance, here is the classical Pundamilia nyereri that combines the yellow flank with the red on top, and then particular morphologies and vertical bars. On here is a pelagic planktivore, Enterochromis paupeus, that also has the red flank and the red dorsum, the top, and the yellow flank, but now combined with other morphologies and other stripes. And so every species is a unique combination of this set of traits. And statistically, we can find regions in the genome that are highly correlated with each of these traits. So here's the midlateral band, or the red on top, and each of these little gray and white bands is a chromosome. And the dots here show you the regions in the genome and the genes when they are annotated, when we know what these genome sequences, what genes they are. They are those that the higher they are up on this, the more strongly they are, the more tightly they are correlated with this trait. And many of them are known from other studies, from more experimental genetics, that they like this one, that they actually do determine these traits. But we find when we do it across the whole radiation, we find that they are not the only ones, usually those that were known from previous experimental genetics, but there are several others. And that's very important. So we find, in summary, we find that all of these traits, the logical traits and the mating traits, are determined by many genes, multiple genes to many genes. These genes come from different ancestors through hybridization. And we find that this entire radiation works a little bit like Lego game. You know, you can disassemble it and reassemble it and make more. You can start with 2, then disassemble it and build 3 or 4 and so on. And that's really how it worked. So here's a quick summary of how Lake Victoria became so species rich. It is this hub that has been connected in the past to all these lakes. It had been connected to the Congo through the Malagarasi. It's been connected to the Nile until today. It's connected to these western lakes. And so cichlids have dispersed across this network multiple times, have mixed their genes, and have radiated. And so it's an old landscape that where old genetic variation has become accumulated and enriched, that the species are not old. And that's where we need to change our mindset. So we've often thought in the past, these are old lakes, and that's why they have old species. They're not. Species are very, very young, but they're built from old genetic variation. The implications here for conservation, I think, is that we must stop thinking about individual species for conservation, and we must start thinking about protecting the entire dynamic, the system that allows this evolutionary engine to continue running. Now again, I told you earlier that this engine is not just fun to observe and not just important from an evolutionary perspective, it's actually money. It really underlies the success, I believe, in many ways, of the continued production of Nile perch in this lake. And that's not unusual. So that evolutionary biodiversification feeds economies. And this process of rapid evolution, here's another one that many of you in East Africa are familiar with, but maybe not with the evolutionary history, coffee. Coffee has a very similar history. I mean, obviously, you know, coffee is underlying one of the biggest economies globally and has been for a few hundred years. And it originated in the understory of tropical African forests. But their diversification is like the cichlids. So here's a beautiful illustration that shows that. The classical Coffea arabica emerged somewhere around 10 to a few 100,000 years ago from hybridization between 2 older species. And all these places where lines crossed between the branches in the coffee tree is where species hybridized to generate a new type. And this is still not unique to coffee and cichlids. Would be strange, wouldn't it? It's true for cotton, for sugarcane, for corn, for Irish potato, and many, many other important agricultural products that emerged from hybridization between 2 or many species in regions where many species coexist. That means in the tropics. Because usually there are very few species of any one group in northern latitudes. And I believe that's also why agriculture and why most domestication of animals mostly happen in southern climates, in subtropical and tropical regions where multiple closely related lineages and species were available to hybridize. Now, this is the engine that led to this pattern. And we all make believe that this should be good for people, right? So let's see if it is. It is, you would think. This is the global inland fish catch map that you know. And you see how closely it matches the global richness of species in fish. But then this is not linked to local wealth. This is the map of poverty rate. And you would be disappointed, but you know it. You see that where there is most fish and where there is most fishery, there's least economic prosperity. And for Lake Victoria specifically, I've been living with this system for many years, and I've been observing from the late 1980s to now how this system really traded biodiversity for export earnings. Like, this is a nice graph that I really like from a recent paper by your colleague Ahimbiziwe, which is essentially showing how the introduction of Nile perch turned biodiversity, if you like, into export earnings. That could all still be okay and acceptable if that had really led to people in the region that live where the fish live becoming prosperous. But that hasn't happened, at least not in my lifetime, and I don't know how many of you have watched this much longer. people in the lake region stay poor, and it isn't people in the lake region that eat the Nile perch, as you all know, right? So very few people on the shore can actually afford a Nile perch fillet. It's other people that eat that. It's us here in Europe, for instance, where these fillets, or in Asia, where these fillets get shipped to. And so this has made me, over the years, think, think that is there a more general trend here, or is this just an accident of history in Lake Victoria? And it's not. The same happened with coffee, going back to that parallel radiation that is very similar to Lake Victoria cichlids. Coffee, too, served as a major catalyst for European imperialism, and many of you know that. The integration of coffee into colonial economy, starting 400 years ago, reshaped global economies massively. It actually fueled transatlantic slave trade, it fueled land dispossession and coerced labor, it fueled genocide, it fueled this phenomenon of the white highlands, where the regions in Africa and some other parts of the world where coffee grows best are dominated by white settlers. And it led to the green bean trade that I don't need to explain because many of you know it. And it doesn't stop there. We can list many other such examples. Now that seems to sit at odds with the UN biodiversity narrative that tells us that human well-being and planetary health are linked.
Ole, 5 minutes remain. 5 minutes.
Yes, I'm almost there. So if we think, so we started with my postdoc Connor and several other collaborators, we started collecting large global data sets on biodiversity, economy, and history, and try to see what this relationship really is globally. And we see very consistently across very many measures of poverty and biodiversity, we see the same thing. Where there's most biodiversity, whether it's genetic diversity in mammals, whether it's plant plot richness or animal species richness, where there's most biodiversity, there's most poverty. And that has not been like that until very recently. So we also looked into history, and we saw that the richest cultures and the largest cultures resided in regions near the tropics and in the tropics, where there was most biodiversity. Maybe you know that India, China, and Africa had larger global GDP than Europe and much larger than the US until a few hundred years ago. That all changed from around the time that colonialism started. And so we asked ourselves the question, might biodiversity and the production of biodiversity have attracted colonialism? And we built a structural equation model to analyze our large data sets to ask, why is this link there? Why is a negative link between prosperity and biodiversity not a positive one? Is it that biodiversity is bad and causes poverty? Or is it that biodiversity itself is good, but it causes colonization, and through that it has caused poverty? And Connor found that the latter is true. So we found a very strongly supported structural equation model by which species richness affects positively colonization probability. And so richer areas have been colonized more intensively. And that leads to natural resource rent increase, leads to governance effectiveness decrease, leads to national debt and poverty rate going up. So here's my conclusions. I have concluded on how species become diverse quickly. Now I want to just conclude on what it means for us as society. So the dynamic process of this hybridization fueled by diversification has unfortunately also attracted colonial desires, which in turn triggered local poverty. So economic benefits are often based not on single species, but on complexes of species that are needed to generate the traits that we like in coffee, cotton, fish survival. Next time I can tell you about why cichlids are recovering in Lake Victoria. And those cichlids that coexist with Nile perch and feed the Nile perch are not the cichlid species that were there before, but they are newly emerged hybrid lineages. This wouldn't be possible if there were not so many species that could hybridize. And so maybe here's the last slide for summary. Biodiversity is very important for people's well-being, so don't get me wrong, but the socio-ecological coupling is broken at a global scale, whereby colonialism wedged the rift between the riches of biology and the well-being of people. And this rift has not been repaired until today. It's actually continued in the current economic system. And so we must stop uncritically exporting the idea that biodiversity underpins local wealth until we are willing and able to also address the global and local inequalities. Because without doing that, by talking about sustainability and conservation, we actually risk to do the worst thing. We risk to lose people's motivation to support sustainability initiatives and conservation. And we actually are engaged in potentially extremely unsustainable conservation and management. That was my last slide. We published this recently in this paper in Nature and People. People and Nature, so you can take a look at it if you like. And I'd like to thank my research group and especially the 4 co-authors here, Margaret, Van, Connor, and Anna, my wife, who have worked together with me on this relationship between biodiversity and people's well-being. Thank you for listening to my talk. I'm looking forward to it.
Thank you, Ole, for a nice presentation. Colleagues, Professor Ole took us through on evolution, but also he talked about the economic benefit of biodiversity, importance of biodiversity, conservation aspect, but also he talked on indirect links between biodiversity and poverty. So I welcome you all for questions so that we can benefit more from Ole. Questions?
It was a lot, I know.
Hello, Professor Ole, do you hear me?
Yes, I hear you. Hello.
Thank you very much. This is Anthony Tabu, whom you have worked with. And now I have only just 2 simple issues. Number 1, first of all, I want to appreciate you for recognizing the economic benefits initially, although eventually you said led to poverty. But when you were discussing this, you have mentioned that Lake Victoria actually ever dried about 18,000 years ago, and then the fish which is there. So during your time when you are studying this, did you ever get an opportunity of tracing some kind of maybe fossils or some part of Nile perch? Because others have argued that Nile perch initially existed in Lake Victoria before it dried up, and therefore this bringing back of Nile perch was not introduction but was actually restocking of a lake which has refilled. Then secondly, on the issue of, now we see that many of the, Nile perch came from Lake Albert and Lake Turkana. And where it came, it has continuously coexisted with the cichlids populations which were there at that time. Now, here where it was introduced, and a good number of cichlids have also evolved when it is now there, are we likely to see it now coexisting with the new radiants which have evolved this time when it is existing there, so that they can now probably coexist and sustain the fishery? I thank you.
Thank you, Tabu. These are great questions. So first, the fossil question. So, yeah, we have looked, never found anything of Nile perch, but our work only extends to the latest desiccation, 18,000 years ago. So the fossil evidence for Nile perch in the system is a lot older. I believe it's Miocene, so we're talking of a few million years ago, when Nile perch was indeed in that system. Probably it went extinct long before the time that I've been talking about. So we have no evidence for Nile perch having been in the system after the emergence of cichlids 100,000 years ago. Second question, yes, in Lake Albert and Lake Turkana they coexist. Now that's true, but having said that, Lake Turkana and Lake Albert have of course much smaller cichlid radiations. and trophically much less diverse. That's important than Lake Victoria. So Lake Turkana has no fish-eating cichlids. It has only a few insect and mollusk-eating cichlids. Lake Albert has one fish-eating insect that lives inshore. It has no offshore fish eaters. So I think, yes, cichlids do coexist with Nile perch. Also in Lake Tanganyika, of course, there's a whole radiation of Nile perch, which in another project of Katie Wagner that I'm involved with, we've recently shown that that radiation is not old. It arrived in Lake Tanganyika after the cichlids, probably. And the cichlids are still there, but we think it restructured the cichlid community. There are very few offshore fish eaters in Lake Tanganyika, and I think that's so Nile perch will allow the persistence in Lake Victoria of small detritivores and zooplantivorous cichlids, and they will coexist with Nile perch. I have no doubt about that. But the large predators of cichlids will probably not be able to coexist with Nile perch in the open water. They will, some maybe survive on the rocky shores and so, but I don't think in the open water. Does that answer the question? And you are right, the cichlids that coexist, that are coming back and actually coexisting with Nile perch, they are not a random subset. They're not the same, they're not exactly the same species that were there before. They have some of those that have really become abundant now, are of hybrid origin. We're working on showing that for one particular case of a species that is lake-wide distributed and very abundant now, and is clearly of very recent origin by hybridization between several species that are thought to be extinct. Thank you.
We are behind time, so I'm going to allow 2 more questions from you. 2 questions. Any other question? There is one.
Good morning.
Good morning.
Thank you so much, Ole, for a great presentation. My name is Yuda Leonard, a science intern from TNC. My interest is to understand, you have spoken well about the relationship between people's well-being and biodiversity. So I wanted to understand, like, what really the colonization changed and in terms of biodiversity and poverty, and what can we do now to turn things around to direct relationship between biodiversity and people's well-being. Thank you.
Yeah. The first question is the easy one. The second one is the very difficult one. So colonization led to coerced labor and forced labor and extraction of human working power and extraction of people through enslavement and forced labor from exactly those places on Earth where biodiversity is at its peak. In the Congo, in West Africa, in the Amazon, in East Africa, in parts of Southeast Asia. And it of course, it has caused a massive, like if you think of the history of Africa, the enslavement of people and the forced labor for very little pay has impoverished economies massively. It's been a major drain on those societies. And in other places, it has caused genocide, like in South America, as you probably hopefully know. Indigenous peoples in South America were initially enslaved and were then essentially eradicated by a combination of factors in exactly those areas where sugarcane and later coffee have been produced. And people from Africa were brought over as enslaved laborers to continue doing the production of those tropical products. Now, how do we, and of course this has, and the economies, the post-colonial economies are essentially, they've continued essentially unabated. There's been no serious restitution And the way the economies are currently run, who dictates the prices for coffee? It's not the coffee growers. Who dictates the prices for Nile perch fillets? It's not the fishermen on Lake Victoria. So it's this decoupling between where the prices are made and who decides on the rules of trade, and where the products are being produced and who puts the labor into producing them, fishing the Nile perch or maintaining the lake or growing the coffee. And I think it's only when that coupling is restored, when the people that actually live in the biodiverse regions and produce the product can also influence the trade terms. It's only then that we can hopefully start developing truly sustainable economies. Does that answer your question, at least partially?
Anyone with concern? If no one, so thank you, Ole, for your presentation. We appreciate your contribution on cichlids in our Great Lakes in Africa and beyond. And goodbye, and we will communicate. Thank you.
Thank you, Mary. Thanks for having me.
A round of applause.
I'll stay on to listen to the next two keynote speakers.
Okay, thank you, Ale. Thank you, colleagues, for allowing me to be chairing this session, for moderating, and back to you, MC.
Thank you so much, Dr. Mary. Thank you so much, Ole, and please stay back. Interesting presentation is on the line. We are now going to look on to the presentation by Dr. Michael McGlew on high-resolution geochemical and microfossils insight on hydroclimate from Central Lake Tanganyika. But to introduce and welcome the keynote speaker is Dr. Tumaini Kamulali. Let's give him a round of applause. Karibu, Dr. Tumaini.
Thank you, MC. Good morning, everyone. It's my great pleasure to introduce our keynote speaker, Dr. Michael McGlew, aka Mike. He's an endowed professor at the University of Kentucky. If you've ever had KFC chicken, that's where it originated. He also serves as the director of the Kentucky Geological Survey. Also, he's the chair of the Geological Society of America's Continental Scientific Drilling Division. I could say a lot about Mike, but I'll keep it brief in the interest of time. One personal connection I have with Mike is that we had the same PhD academic advisor, Professor Andrew Cohen, who unfortunately passed away last year. May his soul rest in peace. In many ways, I think of Mike as carrying forward Andy Cohen's legacy, especially in terms of American collaboration in the study of the East African Great Lakes. Mike has published extensively on paleolimnology, especially of Lake Tanganyika. In fact, after this conference, he will be going to Lake Tanganyika for field work. Mike is familiar to this platform. He has presented before. He served as a keynote speaker at the 3rd FAR4ViBE Conference. That was 2 years ago. So we are very pleased and honored to have him again. And today, Mike will be speaking on high-resolution geochemical and microfossil insights on hydroclimate from Central Lake Tanganyika. Please join me in warmly welcoming Dr. Michael McGlew.
Good morning, everyone. In the interest of time, I'll keep my welcoming remarks short, just to thank everyone for being here and thank you once again for the invitation. Thank you. Okay. Yes, so I'll be speaking today about Lake Tanganyika and in particular the impact that climate change has on this lake. And I'll talk a bit about how we know this. Just acknowledging my colleagues here, many people have plugged into research using geological records from the lake, and I'd like to appreciate them here for their contributions. Okay, so an outline for this talk, just to be brief. I'll speak a bit first about the nature of Lake Tanganyika and what we know about it from a multitude of perspectives. In line with the conference's objectives, we understand well that there are a number of threats to the fish of Lake Tanganyika. They're listed here: destructive fishing practices, sediment pollution, or more broadly, human interventions and degradation of the environment, which I spoke a bit about yesterday. But most of this talk today will focus on climate change and how we use sediments from Lake Tanganyika itself to understand how this ecosystem is responding to climate. Okay, so that will be the focus for today, and in the end, we'll summarize and conclude. Okay, I think most people here in the audience will know a fair bit about Lake Tanganyika, so I won't dwell too much on it. You can see the lake over on the right-hand side. It's quite a remarkable lake from a global perspective. Of course, it's very large. Its watershed is very large. It's one of the deepest lakes in the world, reaching nearly 1,500 meters deep. It's exoreic, meaning that it flows out into the Congo Basin through the outlet about here. And the lake is permanently stratified, and these features are really important for understanding how sediments accumulate in the basin. As the previous keynote highlighted, the lake is quite renowned for its biodiversity, not only for its cichlid fish, but it actually has a number of different species flocks, gastropods, ostracods, and more. So it's really a remarkable system from the perspective of its diversity and its endemism. Some of this is tied to the great antiquity of the lake. The lake is, to the best of our knowledge, at least Miocene in age, meaning somewhere between 9 and 3 million years ago, this basin evolved. And the physical complexity of the basin actually likely has relevance to the patterns of evolution here. Lake level changes coupled with the complexity of the lake's bathymetry very much likely influenced the species development here. With respect to the modern climate system, I gather most folks in the room here understand well that there's a strong seasonality here in the lake. What you're seeing on the right-hand side is simply a representation of the seasonal contrast in vegetation greenness. And this, I think, nicely illustrates the pattern of seasonal change that we have here owing to the migration of the tropical rain belt. Okay, so we have a clear pattern of wetness in the austral summer, the wet season, and a contrast in a dry season in the austral winter, very much driven by the Southeast Indian Ocean monsoon system, which of course fundamentally changes the patterns of wind and rainfall in the basin. And this is a really important control on the nature of upwelling in the basin. Okay, and from the perspective of nutrient distribution, the upwelling system is quite important. When we have that seasonal shift in wind patterns, the lake is almost perfectly aligned with the winds, the prevailing winds, and this allows the development of changes to mixing and driving upwelling, particularly in the southern basin, and even at the change of seasons, we have a bit of complicated secondary upwelling that can affect the northern basin. This pattern of upwelling is really important for delivering nutrients to the photic zone and helping the algae to bloom and develop. Okay, so that very much influences diatom blooms, cyanobacteria blooms, and so forth, which obviously are very important to the food web of the lake. Okay, so this is a very simplified schematic of the pelagic food web that exists in Lake Tanganyika. There are 4 species of endemic lattes at the top of the food web. These are the predators. We have 2 species of the endemic clupeids. Copepods and shrimp dominate within the zooplankton. And of course, we have our phytoplankton down at the base, represented here in part by diatoms. And so this is what's thought of as a bottom-up type of system, wherein higher primary productivity at the base of the food web likely drives higher production in fish. And of course, the fish are a very critical source of both protein and income to the many people of the 4 nations living around the lake. And so trying to understand the base of the food web has been very important from a geological perspective because it's one of the measures that we can actually infer from the lake sediments. And so as I mentioned, and as folks here will know, there are complicated threats to the fish populations in Lake Tanganyika involved in human activities and climate. A number of threats exist: destructive fishing practices, which we've heard a number of talks on yesterday, sediment pollution and land use change, particularly influenced by deforestation, has a really important and profound effect on benthic habitats that potentially very much influence breeding grounds for pelagic fish. And climate, which is what I'll focus on for the rest of the talk, and how we infer climate is using sedimentary records. Sure, no problem. Is that better? Okay. So let's talk about how we infer climate change from the lake sediments itself. So this is what the bottom sediments of Lake Tanganyika look like. And for those who have not seen a sediment core before, this is actually quite a beautiful representation of Lake Tanganyika sediments. Lake Tanganyika sediments are renowned globally for their beauty. And you can see hopefully the many layers. Okay, and I'd like to describe this. What a paleolimnologist does is reads the layers of sediment in the same way that a historian reads the pages of a book. Okay, so each layer can tell us something about the ecosystem. And we tend to use a couple of different techniques in my group. Geochemistry, which is the data you're seeing here. This is very finely resolved sediment chemistry that's derivable from X-ray fluorescence. And we also look at fossils. And so the fossils you're seeing here are actually the scales of the lattice and the clupeids that exist within the system. So we can actually use the fossils in some ways to even look at predator-prey relationships. And so this is quite a remarkable set of data available if we can read it. And so as Tuma mentioned at the start, there's a long legacy of paleolimnological research at Lake Tanganyika, driven in part by Andy Cohen. This was a data set that he and some colleagues published a few years ago that helped to illustrate the effect of warming on the ecosystem. Okay, so this is a sedimentary reconstruction with time increasing towards the right-hand side on the x-axis. And these are geochemically derived indices of temperature, and hopefully you can see the increase in temperature increases down here towards present day. And that relationship was paired with insights on both algae and fish. Okay, and so this is one of the initial ways that we thought to think about the coupling between climate, particularly warming, and the ecosystem itself. So as the lake was warming, the insight was that there was a decrease in algae and a decrease in our fossil indicators of fish. Okay, a very powerful way to think about the influence of climate on the food web. We subsequently followed that work up with a higher resolution perspective, trying to get at annually resolved insights and influences on the system. And what this information told us was that the winds are very important to the ecosystem of Lake Tanganyika, even at an annual level. The curve that you see down at the bottom is our reconstruction of diatom silica, which is very much influenced by upwelling. And what we learned by looking at these data was that the upwelling system in Tanganyika was enhanced by solar irradiance, okay, in other words, the amount of solar energy reaching the tropics, and by the ENSO system, particularly La Nina. Okay, and so when those global climate features converge, we tend to have stronger winds at Lake Tanganyika, and therefore a stronger upwelling system. And oftentimes the opposite is true as well. And so the warm phase of El Nino, the warm phase of ENSO, tends to falter the upwelling system. So for the most recent research I wanted to share comes from the central part of Lake Tanganyika. The core record that we looked at came from this area of the lake just south of the Mahale Mountains. You can see it over here on the right-hand side. This is the core itself. And one of the really important messages I want to leave with you today is that chronology and understanding the constraints of time in the sedimentary record are very important. This particular record is one of the best dated records from Lake Tanganyika that gives us a perspective of about the past 2,000 years. So at nearly annual resolution, we can actually know something about the past 2,000 years of history of this ecosystem, which is quite fantastic. And so you can see, hopefully here in the core, that there's variability in the material that we find. It tends to be dominated by diatomaceous oozes and detrital sediments coming from the landscape. And here's our age model on the right-hand side, which shows a very consistent pattern of sedimentation over the past 2,000 years. The methods for this particular study focused on sediment chemistry and microfossils. For the sediment chemistry work, we use a technique called scanning X-ray fluorescence, which gives us millimeter scale insights into the chemistry of the sediments. And we tend to focus on a couple of different measures. Silica to titanium gives us a proxy for diatom opal. Okay, so it's a really good geochemical tool for thinking about variability in the amount of diatoms in our sediments. And these elements, titanium, potassium, aluminum, and iron, tend to correlate with or relate to terrigenous detritus, in other words, material coming from the landscape. We also look at fossils. We do that through diatoms, and you're seeing examples of the diatoms here on the right-hand side. That work tends to be done at slightly lower resolution using different aspects of microscopy. So I'd like to walk you through the data here. This is our chemostratigraphy or our chemical stratigraphy, and time in this case is increasing towards the right-hand side. So this was about 2,200 years ago, and here's 1900 AD or CE. Okay, and so the upper curve is our diatom proxy or a diatom indicator, that's silica to titanium, and the bottom curve is those elements that tend to associate with terrigenous detritus. And even if you know nothing about the data and what they necessarily mean, hopefully you can see that there's quite a lot of variability in this curve over time. Okay, that's a very important observation. With respect to the upper curve, our diatom proxy, what we can highlight is 15 peaks in silica to titanium at least, and this gives us a sense of the timeline of high relative diatom production. Hopefully you can see that after about 700 years CE, the number of these peaks increases. Okay, so this is telling us something about high frequency variability in the wind system affecting Lake Tanganyika that's allowing nutrients to enter into the photic zone and allowing those diatoms to bloom. In contrast, there are 18 peaks in titanium, aluminum, potassium, and iron that relate to periods where terrigenous sedimentation dominated. And of course, if we're talking about terrigenous sedimentation, this relates to rainfall falling on the margins of the lake and eroding material and ultimately delivering it to the lake through rivers and deltas. And so we can highlight those instances here, suggesting this greater pattern of delivery associated with rainfall to Lake Tanganyika. We do some statistics on all of these measures, and what we find is that this pattern is really the dominant pattern within the data. This is a multivariate look at the chemistry itself. And what really emerges is that for the first principal component, most of the variance is explained by exactly this contrast, the contrast with elements associated with diatom burial versus terrigenous sediments. Okay, so it's the most fundamental change that's detectable within the sediments. And so we can do a number of different statistical analyses on these data to get a sense of what might be a driver. Okay, and so we do power spectra work, we do wavelet analyses, and that's what you're seeing over here at the top and bottom panel on the right, respectively. And here's the PC1 curve, and these values, as they move up, relate to the terrigenous material, and down they relate to the diatom proxy. And so what these data ultimately tell us is that the system is varying at relatively high frequency, in this case at a beat of about 10 to 16 years, which is a really interesting time time cycle, in part because it likely relates to the coupled ENSO IOD system. Okay, and so what we think these data mean is that the lake is quite responsive to those changes in the global climate patterns, both ENSO and IOD, which tend to interact with one another and sometimes amplify one another. And so cold phases of these particular features tend to enhance the winds. The warm phases tend to bring more rainfall, but may curtail the amount of upwelling. A couple of other important features that we see. This period of time here is the medieval climate anomaly, which is known globally. That happened to be a wet period at Lake Tanganyika. And as we moved into the Little Ice Age, this is another period of global change. It's quite a bit more variable at Lake Tanganyika with a wet early phase here, and then progressively greater variance moving towards the present day. The diatom paleoecology helps to support the story, I think. What you're looking at on the right-hand side is a representation of the diatom stratigraphy with the ages here on the left increasing towards the top. And the different colors that you see here are different diatom species. Using the fossil record, we can actually tell which diatoms are present, and we know something about their ecology, so that helps us to understand ecosystem change. Hopefully what you can see here is that there are intervals on this graph that show a number of different colors, okay? And that particularly tells us that the species composition is more diverse during these time intervals, and particularly in this time period here, between about 150 and 700, this was our period of greatest diatom diversity with the presence of Aulacosira, which is it's a very silica-demanding diatom, so that tells us something about the nature of silica within the water column, potentially connected with periods of strong runoff, delivering that bioavailable silica to the lake. There's a really fundamental change after about 700 years AD or CE, okay? And that's in fact the same time that we see our change in our geochemistry, where we shift into a higher frequency mode of variability. After about 700 CE, the Nitsia genera really takes over. Okay, so there's quite a bit of diversity in Nitsia, which is shown here in the brownish red color. Those are needle-shaped diatoms. They look something like this. And when they accumulate in mass in the sediments, it suggests something about high production in the water column. Because of their form, they tend to dissolve quite readily, and therefore their accumulation in the sediment must necessarily be tied to high productivity in order to get these accumulating within the sediments. And we tend to think of those as a pretty strong indicator of an effective wind system, particularly in the dry season, driving upwelling in the basin. Okay. What's really interesting about those data is we tend not to often see this very high diversity of Nitsia, or at least it hasn't been quantified commonly in Lake Tanganyika sediments, and that may actually be giving us information about the secondary upwelling system in the lake, okay, which really is a function of the change in seasons and internal waves that set up within Lake Tanganyika. And so the presence of this Nitsia diversity moving towards the present day, and you can see the different species listed here, may actually tell us something about that secondary upwelling system, which previously was not accessible to paleolimnological research. So that's quite an interesting observation from this particular record as well. And so just to summarize, what we're doing with Lake Tanganyika sediments is using material contained within the layers to better understand the ecosystem and its sensitivity to hydroclimatic change. And with rigorous chronology, we can actually get to annually resolved insights into proxy records from these sediments are giving us new insight into the past 2 millennium. Chemical records show high frequency variability, and when I say high frequency here, I'm talking about annual to decadal scale change, okay? And that's a really important implication for fisheries management, okay, that the lake is actually fundamentally changing over human timescales is a really important observation, okay, and we know even from the past 2,000 years that the lake can fundamentally shift over a time period of years to decades, okay, and that's quite telling and revealing from the perspective of management of the fishery. We have a number of different chemical measures that tell us about diatoms and their important role at the base of the pelagic food web. We use silica to titanium to infer their productivity, and we correlate that with our diatom microfossils to better understand primary production within the basin. We see some fundamental shifts, particularly around 700 CE, which is about 12 to 1300 years ago or so, that suggest a connection, a more profound connection to the ENSO-IOD system. We also see different periods where runoff has been more aggressive to the basin, likely connected to greater rainfall or longer rainy seasons that give us an insight to intervals of enhanced rainfall. And ongoing research with this work has been presented at the meeting earlier and will be the subject of a workshop later today. Because this core is quite well dated, we've done a number of different analyses with it. There is biomarker work ongoing done by Laura Straub, and ancient DNA work done by Tricia Spanbauer to better understand different aspects of the pelagic food web, not just at the base with the algae and the bacteria, but also with zooplankton and with fish. And so I hope that is within my time, and I'll say thank you and happy to address any questions.
Thank you, Mike, for the wonderful presentation. Just an announcement. Mike will be leading a training session today at 3 p.m. So if you think you must attend, please let me know. And we have some time for a few questions.
Are there any questions, suggestions, comments, opinions, additions? There's a hand at the back.
Thank you, Michael. I'm Christopher Aura from Kemfree, Kenya. Thank you for this very unique presentation. May I know the justification for this? Because diatoms have overlapping ecological preferences, one. Then two, I'm not sure about the water quality of Lake Tanganyika, especially the pH, and it is dissolution ability. How did you handle this so that you may have precision in your results? Thank you.
Thank you. Those following us online, you also have a room to ask questions. Do feel free to send your questions on the chat box. There's a hand at the back. Karibu.
Asante. Good morning, everyone. My name is Kenneth Mbwanji. I'm from Ngaba Biotech. We are one of the sponsors. I don't really have a question. My background is molecular biology. I'm a life scientist. I met Dr. Tumaini in Kigoma in April. And we had dinner. He was explaining to me what he does, and I'm just giving compliment. I've really understood, and I was looking forward to this presentation. It's been very clear, even for us who are life scientists, to understand how segmentation affects, the population and diversity in the lake. Thank you very much.
Asante sana. Fantastic. Final hand, if any. There's a hand this side. No? Okay. One final hand. Karibu.
Okay, thank you very much. And mine is Robert Kayanda from Lake Victoria Fisheries Organization Secretariat, based in Jinja, Uganda. And mine is just a curiosity. Why 1 meter core? And what if in any case that will have gone beyond that? Will the results be different? Thank you.
Thank you for the questions. Hopefully you can hear me using this microphone. I'll address the diatom questions first. You're correct. There are considerable variability in modern diatom ecology. And happily, some of that information is known to us as researchers. There's fairly good information that exists to understand the distribution of diatoms and their ecological requirements, and some of that work has been done even by FAR4ViBE researchers. So there's a nice literature that speaks to diatom ecology and how they function and their requirements. So we have some fairly good baseline information that speaks to nutrient requirements, light requirements, structure within the water column, and so forth. The second question related to diatom dissolution is a good one because, of course, dissolution of those silica frustules could, in fact, bias fossil record. In this case, we can actually track the amount of dissolution microscopically. We do scanning electron microscopy to look for patterns of dissolution within the diatoms themselves. And so it's possible to actually quantify the nature of the dissolution. For the very thin needle-like forms, without heavy abundance within the upper water column, their preservation potential in the sediments is quite limited, particularly in deep water, because that transit, could be 400 meters or more. And so their presence actually tells us quite of productivity and the biomass of those plants in the upper water column. So that's an important thing to constrain when thinking about microfossil insights using diatoms. It's a very good question. And I hope I've answered it adequately. The question related to why looking at cores that are meter long or perhaps that length. This particular effort that we've been engaged in for the past several years with FAR4ViBE has really two objectives. One is to constrain the more recent times. Okay, and so the last 2,000 years, what we think of as the common era, tends to get captured at certain water depths with very, very high fidelity. And so the meter-long cores for that particular objective is really important. The project that we're working on actually looks at different time periods of the past 12,000 years, nominally the times when global climate was warmer, to assess changes in functional diversity and their particular effects on the pelagic food web. And so the last 2,000 years was a major objective within that context. Now, with regards to obtaining longer records, that is certainly possible. Of course, Lake Tanganyika is very old, and it is possible to collect longer cores that give you more time. The challenge there is not that it's not possible, and in fact, it has been done with some fantastic insights coming from those studies. It's that the scale of the observation tends to be measured by millennia. Okay, so imagine we have, for example, a 30 or 40,000 year long record that needs to be collected from certain parts of the lake where sedimentation rates are relatively slow. The amount of time you can resolve with the fossils and the geochemistry becomes longer, or so it's a lower resolution record. So from the perspective of fisheries, if we're trying to understand how the fishery may be altered by changes, we really need those sites that accumulate sediment very, very quickly so we can actually resolve time scales that are meaningful for mitigation and adaptation. I hope that's answered the question.
Thank you, Mike. Any other burning question? We have just like 1 minute. Yes.
Thank you for the presentation. My question is about the fisheries decline that you say it might have started 150 years ago. But as we compare today and past years, it seems like today is too big. The decline is faster than 150 years ago. Now I'm wondering what factors would have caused 150 years ago compared to these days. Thank you.
Yes, thank you for the question. It's a good one. And so I think the change in the fisheries is really attributable to a number of factors. As you move farther back in time, it's likely that some of the environmental factors were more important to the function of the pelagic food web that would connect to the abundance of the fish. So in this case, stronger stratification, curtailing nutrient flux, may have had a fairly significant role at that time relative to some of the other factors. As we move towards present day, I think as many of the talks yesterday highlighted, there's a complex web of factors that influence the decline in the fish. The climatic story is inevitably a part of that, but it's really only one part, right? We have the human factors in terms of destructive fishing practices, illegal catch, and so forth, and the landscape changes, which from the perspective of spawning grounds for the pelagic fish is probably underappreciated in terms of its deleterious effect on the amount of fish available to the ecosystem. So as we move towards present day, and particularly over the last several decades, more fishers, changes to fishing practices, changes in land use, all of these factors converge to, I think, influence the fish catch. Thank you.
All right. Thank you all for being attentive and for the nice questions. Another round of applause for Mike.
And let us acknowledge our moderator by giving him a round of applause as well. Can we do a bit of stretching? Can we all please rise? Can we all please stand up? It's a very simple exercise we are going to do. Can you all have free hands? Okay, don't hold anything in your hands. 2. Let you all leave everything on your chair, including my phone. With your right hand up, those of us who are right-handed, if you're left-handed, we're going to write the word fish on the air, okay? F-I-S-H. I don't know, some are writing in German. I see S for Z. Okay? Was that easy? Good. Now put your hand on your waist. We're going to use your leg now to write fish. F-I. If you're not able to stand like this for at least a minute, then we have to ask, what did you have for last night? S-H. Very good. Was that easy? 2 more. We're going to use our head now, okay? F-I. You want to put a dot? S. Very good. H. Although, once again, I know we are many nationalities here. Some of the alphabets I can't read from your heads. The final one, we're going to use our bellies. Okay? Okay. F. I. With a dot. S. Very good. H. Next time we use capital letters. Let's clap our hands. Please take your seats. Moving on. We have the final keynote for the day. This will be given by Professor Peter Jigstra. And to introduce and speak about Professor Peter is Dr. Priska Mzirai. Let's give a round of applause. Karibu.
Thank you very much, MC. Distinguished guests, researchers, colleagues, good morning. It is my great pleasure to welcome all of you into this keynote session, which is going to take us into a very fascinating area where behavior, ecology, evolution, health, and biodiversity comes together. Our keynote is entitled From Social Stress to Evolutionary Diversification, What African Cichlid Fishes Teach Us About Resilience, Health, and Biodiversity. Now, a question to ask ourselves is that what can a small African cichlid teach us about stress, health, resilience, and the remarkable diversity of life in our African aquatic ecosystems. Now, allow me to welcome and introduce our keynote speaker of this fascinating presentation, who is Dr. Peter Gistra. Dr. Peter Gistra is a distinguished researcher from the biology department at the Central Michigan University in the United States of America, where he leads a pioneering research laboratory dedicated to social behavior, physiology, and evolutionary biology. Dr. Gistra research addresses some of the most fundamental questions in behavioral biology, how the social environment influences physiological function, brain, and evolutionary outcomes. Using African cichlid fishes, particularly the highly social and territorial species called Astatotilapia butoni, his laboratory integrates behavioral experiences with state-of-the-art assays for mitochondrial performance, oxidative stress, gene expression, and endocrine dynamics. His groundbreaking work on social stress has revealed critical links between social hierarchy and brain cellular health, showing how competitive pressure impacts brain oxidative stress. This research holds profound cross-disciplinary significance, helping bridge the gap between ecological resilience in aquatic ecosystems and biomedical understanding of psychosocial stress and health and well-being. Additionally, Dr. Gistra laboratory has explored phenotypic plasticity, sexual selection, and speciation. His studies on color polymorphism and mate competition and has shed light on the mechanism driving the rapid evolutionary diversification on cichlid fishes, contributing significantly to our understanding of aquatic biodiversity. Now let us join hands on welcoming Dr. Gistra.
Good morning, everyone. Can everybody hear me okay? Yeah, welcome to my, thank you for coming to my presentation. And I also wanted to thank the organizers, Dr. Skishi and Kamiri and many others for welcoming me here. Isn't it an amazing meeting? It's great to be here. Okay. So we already heard several talks at this conference, including today, that about the haplochrome cichlid fish or cichlid fish more generally, and how incredibly diverse they are. And so this talk will be about kind of like discussing the importance of cichlid fish, like why should we even care? And also one of the goals is also to really show that the world actually does care about what's here, especially here in East Africa. And so Dr. Seehaus already talked about how these lakes can harbor sometimes hundreds of different species. When lakes fill up, riverine species, such cichlid species, only a few cichlid species arrive in the lake, and then they look at this vast space and so many ecological niches, and then they start to diversify, which is kind of like illustrated here. And these cichlid species flocks, which evolved in many different East African lakes, they have inspired evolutionary biologists for hundreds of years, perhaps. Here's an example of species that evolve the same morphology, feeding strategies. We call this convergent evolution in Lake Tanganyika and Lake Malawi, shown to the right. This is like a thick-lipped species. Incredible, diverse morphologies that allow them to utilize different ecological resources. So in this talk, I will just briefly talk a little bit about why are cichlids so diverse. I will keep this pretty brief because Ole Seehausen already took care of this, of course. Talk a little bit about the history that I've done myself in Lake Victoria. I always have to tell people ask me, were you here before in Tanzania? And the answer is yes, I was here 23 years ago. Can you believe it? And now I'm back. It's a really special feeling. And at the time, I focused on the role of competition in driving speciation, but also in explaining and understanding how so many different species can coexist. So competition for resources can play a role in that. And then the second part is about, why are cichlids also relevant, for example, for people interested in biomedical questions. Okay, so I first wanted to talk a little bit about the fact that cichlid fishes, they have fascinated biologists for a long time. What you see here is an example of a biologist that study animal behavior. So how do animals forage? How do they communicate with one another? How do they fight? How do they court? Right, so that's the study of animal behavior, ethology. And what you see here is a textbook from the 50s of Gerard Barens, Professor Barens from the University of Groningen in the Netherlands, where I did my PhD. He was a student of Nico Tinbergen. And for the biologist here, he's one of the founding fathers of ethology with Konrad Lorenz and Carl von Frisch. So cichlid fish are just a really great model system to understand animal behavior and are really textbook examples of their complex behavior. What you see here at the bottom are some drawings from Barens' work. Some of them was done in the 40s and showing some complex display that these fish show when they are competing for mating or foraging territories. This is a video of Nieri, Punamila Nieri from Lake Victoria. And Dr. Seehausen already talked about the fact that many species are separated based on female mate choice on the male breeding dress. Okay, so they have, there are blue males, red males, and females. They have very specific preferences. And so I don't know if you saw that in the video here to the left. Well, the Punamila Nieri male is still very busy spawning, actually. And when you look very carefully, let me see if I can play the video again. You can see some eggs. The female is going to lay eggs here to the left, and she's going to lay them right now. Yeah, here we go. And then, did you see that? She picked them up with her mouth. And imagine that you are reproducing as a female and you spawn and you pick up the eggs and then you can't eat for 2 or 3 weeks. That's what these fish are doing. Of course, it's really, really fascinating behavior. And biologists have actually studied the neural basis of this type of behavior. So here you see a mouth-brooding female from my own cichlid fish facility in Michigan, in America. And I don't know if you can see the eye here. That is one of the maybe 40 babies that are held within the buccal cavity of these mouth-brooding females. So real quick, a little bit about my work in Lake Victoria. I did this in collaboration with Ole Seehausen. I was here back in 2002 and 2003 doing field work in Mwanza. And actually we're going to go there again after the meeting, see if everything is still the same. So here are some pictures of the work done by Tafiri in collaboration with several universities, including the University of Leiden. And so Dr. Kishi is actually also shown on this picture. And so this system here is of course really well known. Here's Dr. Seehausen in 2003 taking pictures of a cichlid fish, and this is Hilary Morosso, he's now retired, but he used to be based in Mwanza, taking pictures of a cichlid fish. I believe this is at Senga Point, for those of you who are familiar with the ecosystem there. We already heard a lot about this enormous diversity in cichlid fishes and how they decline because of the Nile perch explosion. It has stabilized in the meantime. We've learned a lot about that already. I often tell my students, I often refer to a textbook, sorry, it's actually a novel, a science novel type of book written by Thijs Goldschmidt, who also did work in Lake Victoria describing this incredibly diverse species flock and how the Nile perch decimated it. So a very interesting read. So I'm not sure if Ole talked about this work. He did mention hybridization, and earlier on I talked about the fact that female mate choice for breeding coloration is really important for keeping species apart. Okay, so when there is, like, we already heard a little bit about nutrients like erosion or rainfall, how that can lead to eutrophication, and this leads to turbid waters. And in Seehausen's early work, he showed that this can also lead to species collapse. Okay, so it's not just the Nile perch, but we also have other factors that can be detrimental to diversity. So when the waters are murky, females start making mistakes and start to mix, and that could lead to a loss in diversity. In my own work, I focused on competition. Okay, so you have to imagine that these cichlid fish, you can see some of them in the tank right over there, or at the Tafiri stand outside. These cichlids are highly territorial. I mean, it's something in their system. In fact, they're very difficult to keep within an aquarium. They want to, especially the males, but almost sometimes the females too, and depending on the species, sometimes they kill each other, okay? Why are they so territorial? Well, I already mentioned maybe they are competing for limited resources. It could be algae, it could be like, like food. But the males are mostly territorial because they need a mating territory. And you saw that on the video, the spawning takes place within the mating territory. If you want to reproduce as a male, you'd better get a territory. Okay, so that explains the high level of aggression. And at the time, we focused a little bit on this hypothesis that if we assume that species bias aggression towards their own type, that can facilitate coexistence. So what you see is an example actually done by Koda back in the 90s already, where he showed that territorial males, they tend to tolerate heterospecific neighbors. So if your neighbor belongs to a different species, they're fine with it. And so that mechanism can facilitate coexistence and diversity. And at the time, I also focused on Punimilia, the blue species, and the red species. Fantastic kind of like model system that was actually made available to us by Dr. Seehausen. He described all these different Punimilia species pairs at different locations, and they occur there with varying degrees of gene flow. So for example, in this species pair at Makobi Island, there is no gene flow. So red and blue are reproductively isolated. And when you go to other locations, such as this is at Loanzo Island, there is more gene flow. In fact, red and blue are just panmictic, they're just interbreeding all the time. And then everything in between. So there are also locations where red and blue, they hybridize to some extent, but they can still be identified as distinct species. So we call this like a speciation transect, and a great model system to study, you know, different mechanisms that can promote or actually hinder speciation. This is me in 23 years ago, diving, doing some field work. We also studied another species, Neochromis, really amazing. That's a separate talk. And then at the time, we collected Pudimilia nieri in the water. This is me. This is like a line and hook. We have to collect them at like 10 meters depth or something like that in a plastic bag. And only last year did I publish these field findings. There was a separate data set that I published during my PhD. But then just last year, a couple of years ago, I started thinking about this again. And we published the field data showing that red and blue Pudimilia do not only bias aggression towards their own type using field experiments at Macoby Island in Lake Victoria, but we also show that the degree of, we call this conspecific bias, or the tendency to attack your own species, right? It also depends on the social environment. So you gotta imagine, you know, when you are diving there, you see these territorial males, but they don't live by themselves. They have territorial neighbors. And we found, for example, if all of your neighbors are red, you're gonna shift your aggression preference more towards novel red intruders, males that wanna maybe encroach onto your territory. This is really, I was really excited about this. We got this published. And this is basically a slide showing you that the world cares about the diversity that exists here in East Africa, especially the haplochromin cichlid diversity. We got this really nice grant from the National Science Foundation. It feels pretty recent. It actually started last year already. Dr. Kishi is also a collaborator on the grant. Dr. Seehausen is a collaborator on the grant. Professor Hoffman, he is also in the room here. He is a principal investigator on the grant. And this is actually a continuation of what I did in my PhD. At the time when I was a PhD student, I focused on evolutionary questions. I quantified the behavior. I did some work on hormones and things like that. But what we're going to do now in the next 4 or 5 years, we are going to focus on the brain. We're going to focus on cognition. We're going to ask the question, how do these animals actually think when they are engaged in territorial conflict? And what is going on in their brains? Okay, so this is really a unique opportunity to bring different fields together. And hopefully next time, maybe in 2 years, I can come back and share some interesting findings. How is everyone doing? Still going well? We are sort of like halfway through my presentation. Am I still on time? Right? I know there's going to be a health break, a tea break after my talk. Something to look forward to. But we're going to also talk a little bit about like health, right? Stress resilience, shifting more towards like maybe questions that are relevant to the biomedical field. Okay, so maybe some of you are wondering, like, you were in Lake Victoria over 20 years ago. What did you do in the meantime? Well, in the meantime, I graduated. I met my wife. She's American. I moved to America, and I started working on Astatotolapia bertoni, which is a model species in social neuroscience, people studying the brain and behavior. Fantastic system. They're kept in tanks, like you can see here. You see the same spawning video. You can quantify behavior, you can study the reproductive system, lots of interesting physiological studies. You can also, in the laboratory, alter the degree of competition. I'll show you some example of this in just a moment. And I just wanted to kind of like show you a snapshot of labs that study their species, just to kind of like illustrate that these cichlids are really a very important model system to study behavior. I already mentioned that cichlids played a prominent role when the earliest ethologists became interested in animal behavior. They were looking for a model system that shows complex, intriguing behavior. Well, cichlids are really a great option. So different labs are shown here. We studied the behavior. Some labs are using gene editing approaches, lots of neuroscience approaches. So it's really a nice community, and there are even cichlid science meetings where many of these people present their work. Dr. Hoffman and myself, after this meeting, we're going to go to Mwanza, and then we're going to fly to Belgium, to Amsterdam. We're going to go to a meeting in Belgium, in Antwerp, to attend the cichlid science meeting. And next year we'll have our own North America cichlid science meeting. And hopefully we can organize one also here in Africa in the future. Okay, so why am I studying these cichlids? How, shifting more towards the work that we're currently doing in my lab. I always present most of my work to non-cichlid people, and then I have to kind of like provide a broader context. But I think we all agree that life can be competitive, that we all experience varying degrees of stress. And here you see, for example, some examples of competition, which also exist in our human societies. But we also are group living organisms, and understanding the biological basis of this is getting a lot of attention. And then showing here to the right is an example of territoriality and reproduction. By the way, that's also extremely challenging. So, if you're a parent, then you probably know what I'm talking about. Okay, so yeah, we've done some work on this here, kind of like studying the energetic cost of reproduction. In my lab, I mostly focus on oxidative stress. I'll define that in a moment. I first wanted to show you an example of that. It's not just universities who care. When you publish work on cichlids, it's not uncommon that the media is like, wow, this is really fascinating work. This is based on work that was done by a graduate student in my lab. And we described, this is not a novel finding, by the way, but we described that these mouth-brooding females, they eat some of their babies. So we call this filial cannibalism, and people were like, wow, this is really fascinating. We were interviewed by a big radio station in Canada, and it also was featured in a National Geographic. I'm not bringing this up to necessarily promote our own work, but just to show that the world really cares about these cichlids. They're fascinating. Here's an example of work done by the late Maruska and her students. She passed away, unfortunately, a couple of years ago. And this is actually showing how the brain changes when you are mouth brooding as a female. And some of the peptides that are up or down regulated, they regulate appetite. And so one of the good things that happens when you're mouth brooding, your appetite becomes down regulated. By the way, let me see, this is POMC. And the cool thing is that all of these peptides, they can also be found in you and me. Some of these peptides are actually upregulated right now, increasing appetite. Okay, so really fantastic system. I'm gonna, for the sake of time, I'm just gonna not pay too much attention to the details, but I'm just gonna show you that we can also describe the energetic cost of social competition in these cichlids. So this work was done by one of my PhD students. I'd be more than happy to talk about that after the talk or during coffee or tea break. I just wanted to focus for the final 5 or 10 minutes of my talk, wanted to focus on one of the more recent projects in my lab, focusing on the brain and oxidative stress in the brain and how social stress can play a role. And so I need to kind of give you a bit of background. And so there are many biomedical labs, they use social defeat model systems, especially using mice. And they do these studies where they force animals to undergo social defeat. Have you ever entered a room and you feel small? You feel like I'm at the bottom of the hierarchy, right? You can mimic that situation in the lab by imposing social defeat. So someone basically is dominating you. And what people discovered is that this process of social defeat may have a really negative impact on the brain. And so what you see here is an example of how that can lead to oxidative stress in the brain, which is caused by overproduction of reactive oxygen species, kind of like dysregulation of the antioxidant system, which neutralizes these reactive oxygen species. And a sustained high level of oxidative stress can lead to oxidative damage. Okay, and so this process here has been linked to neurodegenerative disease. It has been linked to mental health problems such as depression and anxiety, and has received a considerable amount of attention. And so we were lucky enough to develop our cichlid species into a model system to study social stress. You've already seen that their behavior is very tractable. They are very territorial. They have very well-defined dominance hierarchies where you have these high-ranking males suppressing the lower-ranked males without territories. Okay, so we got some funding from the National Institute of Health, and we started defining patterns of oxidative stress across different brain divisions, showing that it really depends on where you look at in the brain. The brain is very variable, and we also used some gene expression approaches to understand how redox homeostasis, how that is regulated, for example, by antioxidant systems or DNA repair systems and things like that. And again, here's another example why the world cares about your cichlids. This is a paper that we published about the data that you saw on the previous slide. And then all of a sudden there was this newspaper article in the British tabloids talking about social anxiety. This is something that people can relate to. This work was done in cichlids, by the way, in our lab. I was a little bit disappointed that they showed a goldfish, but it is what it is. And here's just another example, stress off the scale. This is also a great example of how working with journalists can really make your science more fun, okay? And yeah, so that was, that's just another illustration that this work is gaining traction. We can also manipulate the degree of competition. Let me see if I can get the second video to work. So this is a very simple experiment where we house small groups of cichlids in a tank, and they are divided. There is a divider through the middle that's separating different groups. And when you house them like this with a flower pot, one male will occupy the flower pot, and then a smaller male will be forced into the subordinate position. So he will experience social stress, okay, social subordination. They fight across the screen. And what this, we call this the cave distance experiment in my lab. When you place the caves close together, you can see this here. The dominant territorial males, they fight with each other. Did you see that across the screen? As a result, the subordinate males, the non-territorial males, they experience some relief. It's like your bully is distracted. That's a good situation, right? That's what we see here. So more distraction here at the bottom. And this is work done by a PhD student in my lab, Tyler Bayet. So here we see the cave distance experiment. Here's the quantification of aggression by the dominant male, the territorial male, directed to members in their own group versus the
Professor
neighbors.
Pitt, so we only have 9 minutes to conclude.
Okay,
Thank you very much.
9 minutes. That's actually too much time. That's good news. Okay, and then we call this when the caves are close together in the proximal treatment. Look at the green bar here. We see a lot more fighting happening between the neighbors, and as a result, just want you to focus here on cortisol levels, which is a marker of stress. And we actually noticed that in the subordinate males, when the caves are close together, cortisol levels are lower. This is basically a physiological illustration that the lower level of stress also leads to lower levels of stress hormones. And then we found that in one brain region specifically, in the hypothalamus, that DNA damage was 50% higher in males that were in the distal treatment, where they experience a way higher level of social suppression. Okay, so DNA damage was almost 50% higher, which is a pretty dramatic effect. Okay, and so this is still unpublished. We look forward to getting this published. This is just part of a bigger data set where we looked at different markers of oxidative stress, both oxidative damage and antioxidants in different parts of the brain, including the hypothalamus, which plays a really important role in regulating social behavior and also maintaining cellular homeostasis throughout the brain, coordinating that, so just like temperature and things like that, appetite control. Okay? Alrighty. Okay. So wrapping up, I just wanted to go back to the cichlids and explain again that this is a great model system to study social stress or competition, social competition, how that influences the brain. The other thing that's really fascinating about these cichlids is that they can be found in very diverse habitats. Okay? So they're a really great model system also to understand how do organisms adapt to sometimes very extreme environments. We already heard about hypoxia and temperature stress. And this is kind of like bringing it all together. That the world really cares. People interested in biodiversity, they think cichlids are just the most fascinating system in the world. But also the biomedical field recognizes that these cichlids can be used to address questions that are relevant to the biomedical world. In today's talk, I mostly focused on social stress because that's something that I am personally interested in. It kind of like belongs within this framework of competition, right? But I'm not the only lab who is funded by NIH. There are several labs using cichlid fish that are funded by NIH because they think it may provide insights also into human health. So what is the take-home message? I think there are three things that I'd like you to take away from today's presentation. The first one is, of course, that this incredible diversity needs to be protected. We need to cherish it and come up with ways to make sure that these cichlid species flocks remain intact while still being able to utilize the lake for various purposes. I also want you, and I try to do this all the time, to promote, you can be extremely proud of these cichlids, right? They need to be promoted. People need to know about these fish. And I already gave you at least 2 examples, and I have more, by the way, but I just gave you 2 examples of how our published work generated a lot of media attention, suggesting that people are interested in cichlid biology, how they behave or misbehave, things like that. Okay, and then also finally, this is also a really great example of partnerships that can be formed. And I just wanted to also emphasize that this is just like more broadly, right? I think cichlid research brings people together from all over the world. I think our most recent NSF grant is a really nice example. Working with Tafiri, Ole Seehausen is part of it. So bringing people from different places together. And with that, I would like to thank you for listening to my presentation. I will be here for the next number of days. Some of you I will meet in Mwanza even, when we are there on Friday. But yeah, thank you so much for listening to my talk.
Thank you very much, Professor Gistra, for a very interesting presentation on our beautiful cichlid species of our African continent. Now, for the interest of time, I would like to invite 3 questions. If you have a question.
Yeah, thank you very much for the nice presentation. I must declare here I'm stupid when it comes to this kind of topic. I'm just wondering the effect of fishing and the selectivity, maybe by species, even by sex of the fish, and the impact on the, say, on the population, let's say, on one species or an ecosystem. Thank you.
Respond or just take the question?
Thank you for the question. Are you talking about like the female mate choice and how that can I'm not entirely sure if I understand the question. Is it about how that can influence diversity? Yeah, if you can give the microphone back to...
Sorry, because you talked about stress, the social stress
Sure.
on a population. So for example, if a fishing gear is selective, let's say to a certain species, even maybe most likely if at that point of the time, maybe when, let's say one sex is taking more, let's say maybe the females or the males are taking more, and that impact on, let's say, on the productivity or the production of that species. And also vis-a-vis the issue of declining of certain fish species. Let's say in Lake Victoria, for example.
Yeah. That's a good question. So what I wanted to, it's interesting when you can study different markers of stress and see if maybe certain species are under stress. But I think and you can use that also in fact you can, for example, use that to study how that influences the effects of cage farming, for example, on the well-being of native cichlid species. But and that is relevant, of course. But the thing I wanted to emphasize is that these cichlids are actually more interesting, not necessarily to understand like how do they suffer from stress. It's actually they are more interesting also to understand how do you cope with it, right? And especially the biomedical field, but also people interested in sustainability and biodiversity. How do you build resilient organisms? Okay, what are the prerequisites for this? And organisms are surprisingly resilient. Okay, and so that's I think cichlids can really give us many clues into how they do it basically and how they manage with challenging situations. Thank you.
Thank you very much. I'm going to take a second question.
Sorry, I'm Emmanuel Swek, the manager for the Marine Park and Reserve Unit of Tanzania. Thank you.
Thank you, Dr. Sistra. Thank you for a good presentation. My name is Salome from Tafiri. I'm interested to know if there have been any gene extinction due to the lack of these subordinated males not being able to transfer their genes to the next generation due to subordination. Has been there any genetic loss for that? Thank you.
So the question is like if that can maybe affect like population size,
Mm-hmm.
you know, if species are maybe extremely aggressive and force many others into subordination. Mm-hmm. There are two things I can say about this. The first one is you see, and that's why cichlids are also really interesting for that reason. There is a tremendous amount of variability in the degree of territoriality. Okay, so they're also a really great model system to study variation in social defeat consequences across different species. The question was also about like, you know, can it lead to maybe extinction or population declines when you think about territoriality? I think the key thing to understand is that it's a little bit counterintuitive, but like if you have a lot of competitive species, but when there are mechanisms that the competition can help them to coexist and can help them even to divide resources. But at the same time, it is definitely, there are also examples of this, not just in cichlids, especially in birds. Some species can outcompete others. Okay, and so there is this evolutionary force where competition can actually, lead to extinction, but it can also promote diversity, and it's kind of like a push and pull process, and usually one wins over the other. But in my lab, we've mostly focused on the diversity promoting effects of competition. But what you're saying that it may lead to problems like declines in diversity. Or population size is definitely relevant. Thank you for the question.
Thank you very much. The last question. I can see there are many hands. I will only take one. Professor Gistra is still around. We can have time to engage with him later.
Thank you, Professor, for a good presentation. My name is John Mbugani from University of Dodoma. I have just an interesting questions, the curious questions. We have seen that Nari patch has caused a lot of stress to these cichlids. So, in what ways, or how have they managed to undergo resilience and adaptation after introduction of this Nari patch to these waters? Thank you.
Yeah, thank you for the question. So the question is about like how did now perch affect perceived stress levels in these fish. So it's to tell you the truth, I don't think we've really looked into this. But again, I'm going back to this idea that species can recover, or organisms can recover. If you are under continuous predation risk, you have a couple of choices, right? Or any stressful situation, you can endure it and try to get through it, or you try to find a solution, right? Maybe you want to move to a different habitat or hide. And so you have to also, that's also one reason you have to look at behavior. So how do organisms respond to potential stressors? Maybe they move to a different habitat. In addition to the physiological adaptation, right? If you feel stressed out, you have social stress, your body will adapt, you will cope, okay? And, but yeah, that's predation. Going back to the kind of like how the now perch boom has more stabilized and how some cichlid species have come back. That's of course a fascinating story. I recommend actually referencing also Ole Seehausen's work. He did some really amazing work on that. But yeah, thank you so much for all the questions.
Thank you very much, but
and
allow me please to give a last chance of question to my DG.
Yeah.
I saw your hand.
Thank you.
No, that was not a hand. Sorry.
Thank you very much. Thank you very much for your time, participation, and engagement in this keynote session. Let us give a round of applause to our presenter. Thank you so much.
And let us extend the same to Priska, the moderator. Let's give a warm round of applause as well. Distinguished participants, ladies and gentlemen, we are now going not for a tea break. Not for a tea break, but we're going to parallel sessions. And I'm going to project the QR code so that we can all scan and know where exactly we want to engage. Can we have the QR code? Yes, can we all please scan that? I will take you through quickly the flow of the day today, but after you've scanned the QR code. I would also wish to remind us that later this afternoon, we have our colleagues, our Muslim brothers, probably and sisters, who will come here to observe the Maulid. It's Mtume Muhammad's birthday today, what Christians we call Christmas. So it's Christmas for the Muslims today. So we will need to vacate the premises as earlier as we can, so we'll not continue to drag time. We're already 30 minutes behind schedule. So right now we're moving on to the parallel session. Saadani will be looking onto biotechnology and fish breeding. Please take a note, and as we synchronize, it's now 10:36. We shall break for lunch at exactly 1 o'clock or 1:45. So respective chairs, session chairs, moderators, kindly take a note that by the time we clock 1:45, 12:45, please allow your participants to proceed for lunch. So Sadani will be looking onto biotechnology and fish breeding. Udzungwa, sustainable fisheries and biodiversity conservation. Mikumi, social economics and fish value chains. And then Mafia, mixed themes. So all the people that will not have a room to go on to those specific rooms will convene at Mafia room, where there will be different topics. But again, Saadani We'll have a parallel scientific session and special session. This is session 2, looking onto biotechnology and fish breeding, session 2. So kindly take a note as I have announced onto the respective rooms, and in the next 1 minute, believing that we all have the schedule, we shall proceed onto the respective rooms. Please, this is not a break, okay? It's a transition. You know what happens when you drag your time on transit? You can be left by the aeroplane. So please, this is just a transit window period for us to move from this room to other rooms, except for those who are staying back.
Proceed with this in mind. I will have the Korea-Tanzania presence in turn. I kindly ask Dr. Kimire to introduce Tanzanian presence in turn, okay? Then I will introduce the first presenter from Korea, Dr. Hyop-Seung Lee, Managing Director of ITEC Tanzania. ITEC is Innovative Technology and Energy Center in Arusha. His theme is technology collaboration between Tanzania and Korea in oceans and fisheries sector. Dr. Lee, please.
Ladies and gentlemen, Abrejos Bui, it's my privilege to be a presenter at this conference. Thank you, Dr. Kimare, for inviting me here. Now, my talk is focused on the collaboration between Korea and Tanzania. So the collaboration history between Korea and Tanzania is relatively short, only 34 years. However, over the last 3 decades, Korea and Tanzania has been doing a lot of close collaboration and technology transfer and advisory and infrastructure development and all those things together. And with very visible results. Now, over the last 3 decades, especially since 2024, when Dr. Hassan, President of Tanzania, visited Korea 2 years ago, Korea has committed more than $3 billion in terms of soft loans and grants until 2028. 2 years ago, $2.5 billion was committed again. So altogether, it's almost $3.5 billion in ODA projects. Now, recently, the commitment and ODA project has been focused on AI, and ICT, and green tech, and marine technology. Some of the marine technology project will be discussed later in this session. Korea is a very successful case which was helped by ODA program of the last half a century. Korea was in complete ruin right after the Korean War about 70 years ago. And the Minister project was one of the key reasons why Korea was able to rise from the ashes. The Minister project was initiated between the collaboration between Korea and United States, and main focus of that project was to modernize the technical education and technical training for Korea, especially for Seoul National University, which is the top university in Korea currently. And you are looking at the example of that project the results. I was born in Korea, got education in Seoul National University and College of Engineering, went to Silicon Valley, stayed there for almost 40 years, got education and got experience in startup businesses and work for semiconductor equipment companies and other companies. Now I am here, and I am conducting some Korean government supported projects and do startup businesses, helping startup companies and individuals here in Tanzania, and I'm seeing very tangible results. Now, if we work together, I'm very sure, I'm very confident that even though it took 40 years for Korea to rise from the complete ruin to the current status, I think it will take less than 20 years, probably 10, 15 years for Tanzania to rise from the lower middle income country to the upper middle country. I'm very sure about that. And that's why I've been here in Tanzania for the last 10 years. Okay, now. So, here's some potential technology and collaboration areas I can present today in this marine and fishery sector. Yesterday, during a keynote speech, cold chain was the most important gap in the whole ecosystem in fishery. And I'm a specialist in mechanical engineering and energy sciences and renewable energy sector. And today I'll be talking about mostly the cold chain, how to bridge the gap for the cold chain area. So there are two major gaps in cold chain. One is the cold room, the lack of cold room, lack of storage space. Those are two main gaps we have here. Because one, we have lack of electrical power. Like a couple days ago, President Samia inaugurated the 2.1 gigawatt hydropower plant, right? So we have now a lot of electrical power. However, we still need to have the distribution lines and those lines, which will take some time. All right, so until then, we are lack of available electrical power in those point of views. And also, to maintain the low temperature in those cold rooms, you need not only the power, but also the facility and technology. So I'll be talking about that technology mainly today. So I've been using ROS, reactive oxygen species, which was mentioned during the talk about 1 hour ago. Now, during that talk, ROS was mentioned as a detrimental thing. But here, we have been using reactive oxygen species, which is a free radical, essentially, into good use. And also, for the power sources, especially in remote areas, remote fishing villages where those cold rooms are needed, the electrical power is currently, a lot of in those places not available. So we can use a distributed power sources, solar power, everyone knows that, or wind power, or biogas. I have a technology and I've been using biogas in Arusha area, and also hydrogen technology, hydrogen engine. The Korean company, the big conglomerate, has been developed a hydrogen engine, has been using hydrogen as a power source for electrical vehicles and for power generation. Reactive oxygen species, I'm not going to go into technical detail, but it is very powerful. It can essentially destroy any DNA it sees. That means it can kill microorganisms. Those are fungus, and those are the live microorganisms. So the free radicals are very unstable, but very powerful to kill those. So now, if you pass a air, which is about 20% oxygen, through a very small gap, and then put a voltage, high voltage across it, some of the air is activated into ozone or hydrogen peroxide, all those very active species, and you can use those to kill those microorganisms. You don't need any raw material. We have it here. And after you use, because it's very powerful, but it's very unstable, so it comes back to oxygen within 30 minutes. So there's no residue. It's free material, no residue. So it's very eco-friendly. It uses very small power because the element is about size of the coin, thickness of the coin. It's very small. If you want a lot of RS generation, you just stack those elements together. That's it. And a company in Korea called CAST has developed that technology, and I am using it in Arusha right now. We built a crop storage facility, processing facility with COICA funding, and it's been in operation over the last more than 1 year. It's very actively. Now, this is exploded view of the element. The size of the disk is about size of your 500 trillion coin. The thickness is about 1 millimeter. Okay, it's very small, so it's very stable. It operates like 10,000 hours without any failure, and uses very small power, and it's and also the size is very small. Now, so you use that to operate. So if you put this in the cold room, you can lower the temperature. I mean, you don't need to use a very high power to keep the room at a freezing temperature. You can have a little bit higher temperature, but more than that, you can augment the freezing temperature effectiveness with this RS species, so that you can keep the environment, storage environment very clean. And also, sometimes you wash fish after the harvest, catching fish, you wash with chlorine water or water, right? But you don't want to use chlorine anymore. Now, if you bubble this RS gas into water, so it's essentially ozonated water. If you wash your fish with ozonated water, it kills all the bacteria and microorganisms on the surface, and then you dry, and you put it in the storage. It's a very effective way. But it does not reflect the freezing, because freezing is the most effective way. But it can reduce the power requirement for that. These are the equipment that the test company developed, and the ones we are using right now in Arusha. And also to use the organic water, this is a system they developed. And also you can make it a mobile containerized cold room system with this. And if you use a solar panel on the top, you don't need to have it connected to a Tanesco. Now, this is the cold room processing facility we built last year in Arusha, and it's been in operation ever since. Daily, about 70 to 100 local women are working here. So we provide 100 jobs for the local women, but also the exporters of those crops use this facility to process and pack those crops, and then they are exporting to Europe 2 to 3 times a week. And they're just shipping those through the air right now. There's no reason why it cannot be applied for fish also. These are some photos which inside that processing facility. And this is the artistic view of the facility. So it's all the rust-based, and we don't have a solar panel yet, but we will put a solar panel later when we have more funding. So it's all IoT-based, AI-based monitoring and control system, all installed, which can be used for the fishery application also. So with that, we need to, so we can build the system, we can operate it, but most important thing is we want to train local manpower, local young adults, so that they can make these systems here. And then also they can make it and maintain it, operate it. So I established a mechatronics and material engineering degree program at Arusha Technical College 6 years ago, and the project ends next year, and I'm teaching some courses there. And okay, I go back to, so this is Arusha Technical College, which I created, and we have a lot of good equipments there. Now, in addition to the technical teaching and technical training, we also do the business startup entrepreneurship training, because all the technology you have, all the skills you have should be connected, should lead to the work, the business. So we focus very hard. So we've been doing this for the last 10 years at Nelson Mandela Institution, now at Arusha Technical College. So we do the entrepreneurship training. Every year we have a business plan competition. And one of the winners are doing a very good business of cage fishing in Lake Victoria. This is the photo I took last December. So he has 5 cage fish, about 6 meters in diameter. Each cage generates 1,000 USD per month. Half of it is a pure profit. He is still a college student at SAUT, SAUT. Okay. So, and I have some other examples, but now she was the winner of a business plan competition 6 years ago, 2020. Okay, okay. I'll be done in about 3 minutes. Now, she raised BSF, black soldier fly larvae, and she raised it and she uses as the fish feed. Last December, after 6 years of incubation, she won the competition in the entire continent of Africa. The prize money was 300,000 USD. Even I haven't seen that kind of money, but she's using it to build a factory right now. So this is obvious. We have sustainability is the key. One of the best ways of keeping sustainability is empower and make it into a business. Oh, okay. So now, in addition to the cold room, we have a couple of other technologies which we can collaborate. Obviously, drying. I've seen a lot of fish drying in Lake Victoria area, but the environment of drying is not that clean, right? So I saw one poster session which uses the greenhouse to dry the fish. Is a very clean way. So we can use those. We can use solar power, but also you can use biogas and other energy sources. We can work together to make it a industrial scale. Okay. Now, canning. Canning is a very effective way of utilizing most of the fish products because you don't waste them that much. If you can, it's very easy to ship, very easy to store. And because the size is small, you can make it at a very low cost for each item. Everyone knows that. And the cannery is not that expensive to create here. So I think canning can be a very good potential solution. The Dongwon Group, which is the biggest tuna can company in the world, they can come out here if you invite them, okay, and create a factory here. And the power generation, solar power and also hydrogen. The Korea one of the biggest Korea group is operating hydrogen engine for vehicles and factories and those things. We can use those to power these facilities. And biogas. I am part of the group which put biogas plants in 2 beta schools in around Asia. We plan to expand it and then make it into a source of energy not only for the kitchen but for the vehicles and also to generate electricity. So we have a lot of good areas to collaborate with. Thank you very much. Okay, so because of the time, I can entertain questions later.
Please understand that due to time constraint, we cannot receive many questions, and we'll summarize, finally, wrap up the one by one, and individual follow-up will be arranged after the meeting. Thank you.
Good morning. I'm Ismail Kimirei, DG Tafiri. I will be introducing the Tanzanian presenters. We will have, I think, 4 presentations from Tanzania, but we are going to be alternating them between Korea and Tanzania. And now I wish to bring to you Mr. Dr. Muhongole, who is the manager of the National Fisheries Laboratory, which ensures the quality of fish in Tanzania and those that are exported. No, it's Muhongole. The laboratory presentation, please. Oh, it is with you? I sent it to you. Okay. Yeah, will be the second one. No, bring Dr. Happy, please. Okay, so while we sort that technical glitch, Dr. Happy Peter is a researcher at Tafiri, and she's been involved in the development and actually research on the PFZ, the potential fishing zones, using satellite data to identify areas for fishing. And during 2024, when the President of the United Republic of Tanzania visited Korea, there was a MOU signed between the Ministry of Livestock and Fisheries and the Ministry of Oceans and Fisheries in Korea, between the two countries, to collaborate on blue economy. And along that, we also had an MOU with the Korea Institute of Ocean Science and Technology, and developed a project to establish a satellite application center in Tanzania, which is going to be in Tafiri. It's in the advanced stage now of, we are going to be installing the processing facilities, and we have conducted, I think, 2 now trainings. We'll do the same for the rest of the period. And now the technology that is going to be established here is going to add value to be used for research in oceanography, fisheries, and all the stuff. So I wish to present to you Dr. Happy Peter. Please take us through. You have 15 minutes of presentation. We will give you 5 minutes of Q&A, but please, if you can make it shorter, it will save us time. Can we get a round of applause for her?
Good morning, everybody. Good morning, ladies and gentlemen. I think I've been introduced already, and the screen there has my name and everything. I'm glad now to present to you the PFZ. How do we communicate with the desk there so that I can get the forward slides? Okay. This one is forward. PFZ itself is an application that fishers are using to determine the potential fishing areas where they have chances of getting fish, at least, not at least, for 75%. And then when fishers are fishing outside the potential fishing areas, the chances is 80% that they will miss fish. So before having this kind of application, there should be a challenge. The application should solve the problem that the community or the people are facing. So at the moment, the fishermen were having difficulties to identify the area that they can catch fish. The fishers were just doing the fishing in a hunting kind of system, where the fishermen, for instance, they get the information like on this time of the month, this time of the year, you can go maybe location A, B, and C. It's just in hunting. And then in most cases, they were missing the fish. Another challenge, because you are just going randomly or hunting, then the fuel will be a problem, because you are using the fuel going to fishing. And then you are just going to the places that you don't know, and then you are not aware that you are going to catch fish or not. And then the rising in the fuel cost also was a problem to the fishers. Probably if the fuel were just getting free, maybe it would have not been a problem for the fishers to just go roundering around the ocean or the lakes. And then we're having also the problem of ocean dynamic in ocean conditions. Sometimes based on these climatic changes, the fishers cannot catch enough of the fish. And then the longer searching time. A fisherman is just going there, going around, without knowing where they can get a potential area, so they will use a lot of time to search. For instance, they can get they can leave the landing station at 15 hours. And then the intention is to go starting setting the net at 19 hours. So you find that they were using a lot of time from leaving the landing station to where they're supposed to set their gears. So these were just the challenges, and then we need to help the end users, the fishers who are depending on the ocean itself, the community that are depending on the ocean, at least to get the importance or to get the benefits out of the resources that is around them. So now, having that kind of a challenge that the fishermen were facing, that's when we come up with this, the Earth observation that could help us in finding the possibilities of helping the fishers. And then from the Earth observation system, we get the satellite information, and then the satellite information that we were using, we use the sea surface temperatures to determine the potential fishing areas. From the sea surface temperatures, where we're having the location or from the mixing up of the water in the ocean, there will be a chances when there will be an abrupt shift in the temperature difference within the water masses. And then if this condition stays for 3 to 7 days, that's when we have the ocean fronts, where we are sure that the fishermen, when they're given the coordinates of going into those locations, they'll be able to get fish for 75%, as I've introduced before. So for our case in Tafiri, though we are getting the information of chlorophyll and then sea surface, but this for the ocean, the sea surface temperature is a parameter that we are using for developing this PFZ application system. How PFZ works? As I've introduced, we get the information from the satellite, where we get this the sea surface temperatures information, and then from the sea surface temperatures, we can get the potential area, the potential fishing zone map, where we have different coordinates within there. As I've said, the difference in temperatures within those water masses, when they stay for that 3 to 7 days, we can have the map at our e-station in our office. And by then, we were getting the satellite information from the GMS in Africa, and then we could have those maps. At the beginning, the fishermen were given the coordinates of where they should go fishing. We get the information from the satellite station from the e-office from the satellite station in our office, but at times we are not at the office, so the fishermen could not get the information of the coordinates of, for instance, today, the coordinates of potential areas, it's as A or B. So the potential fishing zone, we had to come up with the application. Now the fishermen at their comfort zone, they can just request from their phone, instead of just waiting for the potential, for the coordinates to be received from the fishers, from the fishing officers at their district. Through the process, I didn't want to go through the process of how it was working before based on time. That's why you can find me, I'm getting back and forth of trying to filter which information I can deliver here. But the information is, we get the map, which is having the coordinates of those potential fishing areas. And then from those maps, that's when the fishermen are requesting from their, after installing the application in their phone, that's when they can be able to request those coordinates. And the mobile itself, the mobile app, can be found in both the the Android system together with the iPhones. And then at the moment, I just wanted to give you the statistics of how many people have been registered or how many people have been using the application itself. Then you can see from the numbers that the fishers, together with the researchers, or the users who are using the system, we're having more than 800 people, users that are using the application at the moment. Because the application is not only for the fishers. The fishers are using to go to get the information of where they're supposed to go fishing. But us who are going to buy fish, we can get also the information of the market from the same application. So the fishermen are posting what they have caught. Okay, I'm told that I'm remaining with only 1 minute. So I'm just have to rush, like the number of the people that have been registered is 800. And then the request of the fishermen, yes, the fishermen request, like today they are requesting for the potential fish areas. We're having more than 2,000 requests. And then the sell post, because the fishers are buying fish, are selling fish, and us who are buying fish, we can also have the sell deposits posts which are showing the, what we call them, the transactions. It's 200, more than 320. And then by having this kind of discussion or presentation, different meeting, we get some interest from different countries. For instance, at the moment we are having 4 countries which have been interested in having the same application. Because as we know that fish don't have borders, and then we are working with this, especially the migratory and pelagic fish. So if we are not being caught by our fishers in Tanzania, they can be caught also in Somalia or in Mozambique or Mauritius. So the other countries have been interested that I have just shown there. And then in some of them, we are also in the memorandum stage of starting also to coach them or to give them this kind of the system. Because so since the PFZ is a satellite thing, so we can use all the water bodies. And I think based on the time that I'm having, this is what you can get when you go to the Play Station and then the App Store, you can install. And then when I'm counting people here, I think we are 50 of us. So if you can also try to install the application, that number which I've said, the 800, will go higher. And then maybe we can discuss more on the time of question and answers based on the chair. Thank you.
Thank you, Dr. Happy. Actually, you saved me 2 minutes of your presentation time. That is 7 minutes in total, plus 5 minutes of question and answers. I was a bit inventive. I didn't want to tell her the whole story so that she can finish in time. So, ask questions if you have questions. Asante, karibu.
Yeah, thanks for a nice presentation. My just little small question, didn't worry. How will we ensure an intensive fishing or overfishing? Thank you.
You got the question, eh?
Yeah, I like that question.
Yeah, I know. It has been asked many times.
Yes.
Another question? Oh, you answer that? Another question? Okay. Okay, you can answer that. Thank you.
Okay. Thank you for the question. Maybe based on the rushing of how I was presenting, sometimes when you say potential fishing zone, we expect maybe they are just static. They are just changing based on the different water movement, upwelling, ocean activities that are happening there. So they are not static. So by the time I'm giving you the coordinates here, tomorrow when you ask the question or the coordinates, it won't be on the same spot. Sometimes it can give you at 6 kilometers, and then sometimes 50, sometimes they are not there. So they are not static, like every day the fishers are going to go on the same spot. And at the same time, the potential fishing areas, they are not the very small, very minute overlay areas. It's a very huge areas. And at the same time, as I've mentioned before, the fishers that we are targeting with this application, they are migratory fishers. So when you are not fishing them with the worries of overfishing, they will be fished somewhere else. So the intention was at least to make sure our fishers are benefiting from the resources that they are around them. Yeah, so don't worry about the overfishing. Thank you.
Yeah, thank you. Thank you, Dr. Happy. Now, I would like to introduce second Korean presenter, Dr. Kwang Sun-pa, Korea Coastal Management Association. He is a research associate. KCMA is the governmental funding association under the Ministry of Ocean Fisheries. Dr. Park, please.
Good morning, everyone. My name is Kwang Sun-pa from Korea Coastal Management Association. First of all, I would like to thank Dr. Geun-young Kim and the Korea Institute of Ocean Science and Technology for giving me this opportunity to speak. Today's my presentation is closely related to ongoing Tanzania Marine and Fisheries Satellite Application Center project. During my career at Korea Institute of Ocean Science and Technology, I led the development of the Korea operational oceanographic system surrounding waters of the Korea. Based on this experience, I would like to introduce ICT AI-enabled operational coastal forecasting center for coastal hazard reduction and the blue economy in Tanzania. This center can be used not only to reduce coastal hazard, but also to support fishery, port, maritime transportation, tourism, and the various coastal activity. Now introduce the vision for Tanzania. Okay. Tanzania has a long coastline along the Indian Ocean. Many people depend on coast for their daily livelihoods and economic activity. All activity related to fishery, marine transport, tourism, and coastal community require a safe coastal condition. However, better coastal information is needed to support this activity and improve coastal resilience. This is why operational coastal forecasting system important in Tanzania. Tanzania already has valuable coastal resource, but operational information service remain limited. Observation is available. Forecasting information service and decision support are still limited. Tanzania's long coastline support fisheries, shipping, tourism, and coastal community. However, a coastal hazard and marine risk continue to threaten lives, infrastructure, and the blue economy. Therefore, Tanzania needs an operational coastal forecasting system. Operational coastal forecasting support safe, efficient, and sustainable coastal activity every day, not only during disaster. It supports many sectors, including fisheries, aquaculture, marine transportation, coastal tourism, coastal construction, and marine environment protection. This is why it is essential for blue economy in Tanzania. Okay. Korea has numerous coastal hazards and maritime accidents, including typhoon and peril disaster. So, there were great demand for reliable coastal information to reduce coastal hazard and support various coastal activities. So, at the request of the Korean government, the Korea Institute of Ocean Science and Technology carried out 9 years national research and development program to develop a Korea Operational Oceanographic System, or AQUACULTURE. At the time, I was project manager of that program. We successfully developed and transferred the system government agency in 2017. Since then, government agency has been using that system to provide operational coastal forecasting service. As shown here, coastal system consists of 3 main components. First, observing component collect real-time data from various observation platform. Second, modeling and forecasting component produce reliable coastal forecast using forecasting model. And the third, service and support component support various coastal activity and emergency. It has played an important role reducing coastal hazard and supporting a wide range marine activity. This is the experience and the expertise that we would like to share with Tanzania. This is an application example of the operational coastal forecasting system. In April 2014, passenger ferry sank off the southern coast of the Korea, and more than 300 people died because the tidal current is very strong in the accident area. Accurate 3-dimensional current information were essential for underwater search and rescue operation. Using CUS, we provide the real-time and forecast information on wind, wave, 3-dimensional current, temperature, and other coastal conditions for search and rescue operation. Okay, this slide shows an example of 3-dimensional coastal current forecasting for search and rescue operation. The figure on the left represent predicted 3-dimensional current at the ferry sink site. The figure on the right compare forecasting result with the observation. You can see a red dot represent observation and the solid line in predicted current. It is very important for such a team to know available diving time, available diving time. So we can determine available dive time and plan underwater search and rescue operation. Okay, this slide shows a daily predicted search area after passenger ferry sank. We use the CUS system to define search area for such surface, drifting survivors and support search and rescue operation. Okay. This slide shows the oil spill prediction case from 2007. Oil spill accident on the coast of the Korea. Operational forecasting system could provide information on wind-driven current, wave, tidal current, temperature to the oil spill model, together with oil spill information and observing system. Then, oil spill model can provide the movement of the spread of the spilled oil and support response operation. Okay, this slide is a daily coastal and offshore forecast service by provided Korea Oceanographic and Hydrographic Agency. It provides information on surface wind, wave, sea surface temperature, current, and so on. This service is operated based on cruise system, Korea Operational Oceanographic System. Okay, we have shown how operational coastal forecasting system works in Korea. You may then ask, there are already global forecasting system as well as OPERA in Africa. Why? Does Tanzania still lead its own coastal forecasting center? Global forecasting system provides useful forecasting information, and OPERA support regional capacity in Africa. But global forecasting system and OPERA cannot replace Tanzania's own operational coastal forecasting system. Tanzania needs localized forecast, and warning and decision support for various coastal activity and emergency. So Tanzania needs its own coastal forecasting center. Okay, from now, I would like to introduce the project overview, our proposed project. Title of project is Establishment of the ICT AI-enabled operational coastal forecasting center for coastal hazard reduction and blue economy in Tanzania. The project is planned for 4 years with the estimated budget of about $6.5 billion US. The target area, Tanzania coastal waters. This slide shows the target area. This project covers entire coastal water of the Tanzania. This region includes the country's major coastal city, port, and marine economic activities. Objective of this project to establish an ICT AI-enabled operational coastal forecasting and decision support center to strengthen coastal hazard preparedness and support the climate resilient blue economy in Tanzania. To achieve this objective, the project consists of 4 major components. Like this, I will explain each component in detail. First major component is the comprehensive data integration. It integrates data 3 major sources: national ocean observation data, global ocean meteorological data, and satellite remote sensing data. This integrated data foundation provides reliable, diverse, and high-quality information for operational coastal forecasting and decision making. This slide shows the important role of satellite remote sensing in operational coastal forecasting. Satellite provide actual ocean condition, such as sea surface temperature, sea level, ocean color, surface wind and wave, and so on. A numerical model use this information for initialization, verification, and data assimilation. So, satellite observation improve the performance of the operational forecasting system and the forecast accuracy. Therefore, the proposed project can be closely linked with the Tanzania Marine and Fisheries Satellite Application Center, currently established by QST. If implemented as a follow-up project to the Tanzania Marine and Fisheries Satellite Application Center, this project will create a strong synergy for operational coastal forecasting and decision support in Tanzania. Second major component is a numerical coastal forecasting system. It integrates atmospheric forecast, ocean boundary condition, and the satellite and in situ observation. Numerical model produce reliable coastal forecast on wave, storm surge, coastal inundation, current temperature, and the sea level. These information support early warning, marine safety navigation, fisheries, aquaculture, and coastal management. It will help people make better and faster decision make also. Okay, this slide saw the major component. ICTI enable forecasting and decision support center. This center serve as operational hub of the coastal forecasting system. This center integrates satellite data, numerical model, in-situ observation, and the historical data, and AI analyzed data, provide decision support, the assess coastal hazard, and deliver timely information to government agency, coastal community through web, mobile, and alert service. Okay, this slide is the first major component. Component is capacity development and institutional strengthening. Technology alone is not enough. Sustainable forecasting system also require skilled people, strong institution, and long-term operational capacity. This component focus on training, education, knowledge sharing, institutional strengthening, and sustainability operation. Through this activity, Tanzania will be able to operate and continuously improve the system in the future. This slide shows the proposed cooperation framework. Korean partner will provide technical support, training, and experience, while Tanzanian partners will participate in development and operation the center. Through cross cooperation, technical exchange, training, and pilot operation, we aim to establish a sustainable ICT AI-enabled coastal forecasting and decision support center for Tanzania. Okay, this slide illustrates the operation room of the Coastal Forecasting and Decision Support Center, Tanzania. This center integrate global data, satellite data, and Tanzanian observation. It produce localized and higher resolution forecast for Tanzania, and the assess the coastal hazard and support decision support for coastal activity and coastal hazard and maritime accident response. Ultimately, the center will serve as the national hub for the coastal hazard management and the blue economy activity in Tanzania. Okay, this is the main 5 outcomes. This project will strengthen coastal hazard preparedness, establish operational ICTI forecasting and decision support center, and improve safety of the coastal community. It will also support the climate resilient blue economy and strengthen Tanzania's national capacity. Finally, I would like to leave one simple message. Better data lead to better forecast. Better forecast create safe community. Together, they support a stronger blue economy. We look forward to working closely with our Tanzania partners to maximize the value of the Tanzania Marine and Fisheries Satellite Application Center through an ICTI-enabled coastal forecasting and decision support system. Thank you very much for your attention. Asante sana.
Thank you very much, Dr. Park. So as I mentioned to you before, if you have any question, I will summarize the end of this section. Please understand. So it was very surprise proposal for Tanzanian side, but before our departure from Korea, for information, concerning Tanzania is a priority country, partner country for Korea, group of experts from various fields, including coastal erosion, ocean disaster prediction and response, like Dr. Park, satellite was formed to make project concept paper and the details of the project in Korea. And hopefully this proposal will be considered as one of the possible candidates supported by Korean Ministry of Oceans Fisheries with a special interest and support in the future. Thank you, Dr. Park.
Thank you very much, Doctor, for very illuminating presentation. And I can see now how what you're proposing aligns with the application center and provides more information to the application that Dr. Happy provided, from the fish to safety. It's a lot. It's a lot. I think we are seeing, and I think we are going to add the debris tracking system at the same for microplastics and all this other stuff. Muhongole now, please. I want to introduce Muhongole, who is the manager of the National Fisheries. No. Okay. National Fisheries Laboratory, currently based in Mwanza, but we are developing labs in Dar es Salaam and moving to Bagamoyo, and this is supposed to align with the new to be constructed fishing port. And so, Muhongole, please take it on.
Good afternoon. Thank you for having this opportunity to say a word on collaboration with the Korea and Tanzania in the area of quality control. I will talk on establishing Tanzania Seafood Quality Control Center in Tanzania, of course, under collaboration with the institutions from Korea. One will ask, do we have or we don't have quality control services in Tanzania as of now? But the answer, we do have it. We have inspection services and the laboratory services in place. But for the laboratory is centered at Lake Victoria and Mwanza. And now as we see the fishing activities is scattered across the country at the coastal, Lake Tanganyika, Lake Nyasa, and Mwanza. And we have now numerous stakeholders, fishers, whom we want to bring the services close to them so that you can ensure the quality of the products. So the talk would include, as you see there, shortly, and under there we have inspection services of Lake Victoria, and here the laboratory services in Mwanza. Basically, our institute is alliance or follows, you can see, under national fisheries policies, Fisheries Act, regulations, and other programs, which we work to comply with those. This, I think, is known to most of us, but you will see that is a processing for the smoking or salting of fish. It is artisanal processed. This is a processing in the west coastal area of Indian Ocean. We see this is the current boiling pans, and see here is boiling, and then drying. Some they dry. This is Lake Victoria, drying on the Raxi. And here is already dried, they are sorting it. You see these are products, different types, and dusts. This processing ends up with dividing the fish, sardine, the head part and the body part, and the dust. So actually we have like mainly two products. We say the dusty daga and the body part, which actually we are losing a lot from this science of daga, which provides a lot of nutrients. So when you aim the boat and you leave a part of the head, it means you lose a lot, which could be consumed by the human. Similarly, these are also processing. This one, we are trying to study on sensory evaluation to see if we can preserve the small sardines from Lake Victoria, be stored for a while to be consumed or packaged as a fresh. And this is a sorted one. This is the barbecue tilapia. This is smoked tilapia, and this is the rose from the Nile perch. And we see small-scale processors. We are conducting training, teaching them on how to process this process I have mentioned. So, quality control seafood Tanzania, as I mentioned earlier, it is present under the implemented through the inspection services, which started in 1995, and laboratory which began in 1997. And the lab is accredited was earlier accredited by SANAS, South African National Accreditation System, and was taken over by the SADC, Southern African Development Community Accreditation System from 2018. And till to date, it is accredited in 20 parameters, as you can see there, with the 13 personnels. One as a management signatory, two nominated and deputy nominated representatives, and 10 signatories, 5 each of the laboratories for the chemical and microbiological laboratory. This one is incubation where we grow to enumerate the number of bacteria which as a general or specific identification. Why then establishing the seafood quality control laboratory, as I said, or the need of this collaboration is Tanzania to borrow science and technologies on fish inspection, laboratory services, as well as research and development aspects, so that we can strengthen the area of the quality of the products or processing and distribution. Also, we have the need of expanding the accreditation. As I mentioned, we have 20 accredited parameters, but most of them in microbiology, and a few, 6 of them in chemistry, but mainly in water. So we think if we collaborate, we will further expand to accredit other parameters like proximate analysis, pesticide, mycotoxin, such as aflatoxin, antibiotic residues, hormone residues, and even microplastics. These ones are the main contaminants which is costing the country. At times, it's supposed to take the samples to abroad for testing those. So also we have weakness on quality control, which we hope will be strengthened. We have limited certified small and medium enterprises practitioners, horizontal and vertical linkages, and of course we have limited support to the small scales who have shown adding value to the products, which would need a support so that it can be scaled up to produce more, and especially fulfill the market demand or even the export. The need for Sea Quality Control Center is to enhance science, as I mentioned, technology and knowledge-based transfer, equip laboratory and inspection services with the modern equipment, facilitate compliance on fish and fish product standard specifications, and expansion of the scope again. These fish and fish products standards, they are in a place, but maybe some awareness and the need for the compliance of each standard, like for dried daga, sardines, for smoked daga, for frozen fish, and other fishery products, which if we equip the lab, it means can be able to ensure full compliance of these standards from microbes, chemistry, and other contaminants I've mentioned above. So also we'll establish advanced fish quality control and traceability system and eliminate fish possible losses. As the earlier presentation on satellite tracking the fish, it means we'll be catching more fish, and those fish should be traced. And this will help even to illegal and regulated fishing. Okay. This expand its National Fish Quality Control Laboratory, as mentioned by director was introducing me, that now the accredited laboratory is in Mwanza, but we hope we are expanding Dar es Salaam laboratory, which will be extended to Bagamoyo, in line with the fish harbor, which will be built. And of course, it will further be extended to the southern part and northern part to control the fish and the fishery products. Benefits we expect with the collaboration with institutions, especially with Korea, will be able to establish Tanzania fish and fishery product quality management system, strengthen and accredit the National Fish Quality Control Laboratory in Dar es Salaam, establish fish and fishery traceability system, and strengthening national and international cooperation through joint research projects. Of course, this will be done jointly with the inspection services, research with the TAFIRI and other institutions in the area which the institution will be competent. So, in conclusion, Tanzania Seafood Quality Control Center will play a great role in ensuring the compliance of the stakeholders along the value chain from fishing to marketing for general public, and it will advocate on the wholesome post-harvest handling, compliance of stakeholders on the national and international food safety standard specification. As well, it will facilitate certification in SME stakeholders all along the value chain, of which now is a challenge that is like they are hanging. No one who can guarantee them in the full scope of standards which will guarantee them to be certified and be comfortable with the trading with their products. Thank you for your attention.
Actually, Muhongole have saved me 8 minutes. That means we can, of course, overflow this. But thank you very much. You will note that the vision, Tanzania Vision 2050 that we have started implementing this July actually deals specifically with export market, and his labs and whatever infrastructure we're going to put in place builds towards building that capacity. So it's a really huge issue that we need to tackle. If there is a question, I will take one question so that we save enough time. Karibu.
Okay, thank you. My name is Akin Matola. I'm working with the Tanzania Fisheries Research Institute at Dar es Salaam Center. I will ask about what is the priority parameter for example, between histamine, for example, for small tuna, and also microplastics, and in modern laboratories, these are very essential, especially in the tuna industry. So in your laboratory, what is your priority species for targeting the hygiene condition?
Thank you for the question. As you mentioned, the priority parameter, I would say, is like all, but we'll go for the affordable one. Like histamine is the most important in the tuna fishing, and of course in line with the harbor which are built. That is part of hygiene, but also microbiological, which we will extend the labs to the point of harbor, so that we monitor it from the landing and to the laboratory, and that will be certified before is traded domestically and for export.
Thank you very much.
Thank you very much, Dr. Mongole. Another round of applause. Yeah, this morning I get the good news. Tanzanian government issued official letter of intention for this project to Korea. So the button is in my hand. So I will do my best. It's very feasible to see the real project in 2 years. Please keep your fingers crossed. Thank you very much, Dr. Mongole. Now, third Korean presenter. Now I'm introduced Dr. Keun-Yong Kim. Ho-Gun Jeon. He's the research director of the UST 21st, which is one of the leading private oceans and fishery research institution in Korea. Please, Dr. Jeon.
Good afternoon. Habari gani?
Mimi naitoa John Hogun. Today I want to make a presentation with the title of Eyes in the Sky, combating marine debris with a satellite and drone and AI. So actually this turn is maybe quite different before presentation, but how about converge to this theme? So today I want to introduce about my project, our company's project about marine debris, and I want to focus on what kind of data we archive and how we do use the data and what kind of result we have made. So these three is very important for you guys, I think. Then let's go. Oh, I'm sorry. Okay, this is presentation. So the plastic debris problem is not only one country's matter, but this is very global matter. So as you can see, this graph, this map shows how much the plastic debris occurred in each country. And how about Indonesia, Bali? Yeah, as you can see, there are a lot of PET and plastics along the coast, and also on the right side, there is the site of resort and hotel and tourism spot. They are damaged by this marine debris. So they are very support and they offer or suggest us to participate in satellite monitoring about the Indonesian coastal region. How about Korea? Now, Korea is also not unavoidable in the plastic debris and marine debris matter. South Korea also faced plastic debris on the coastal, especially from the fishing gear. Actually, the PET and household plastic debris is nothing much, but when it comes to fishing gear, it damages too much of the cost. So Korean government have this kind of marine debris management scheme. So there are 4 kinds of marine debris. From the left, coastal debris, and floating debris, and submerged, and uninhabited island. So there needs some on-site collection and also some center of fishing gear deposit and recycling. So today, what things UST21 have worked for is marketed as a lead triangle. So from now I want to introduce 4 projects which is especially held by the government Korea. We had one R&D project and 2 operationalization project, and last one is a BIOLA project. The first one is a source analysis marine debris on inhabitant in island. To investigate inhabitant island marine debris, we use satellite, aircraft, and drone. These 3 platforms collect the information as imagery, and those data is saved in the database. And this data is analyzed by the AI platform. Then the result can be used for policy making support. But it is very also difficult to understand how much satellite we must use to analyze this kind of result, this kind of marine debris consistently. We have to use a lot of satellite images, 1,783 scenes, and so it was too expensive. Yeah. Next. And how we use the satellite and area imagery. The left one is satellite imagery, and right one is area imagery. As you can see, the left one has wide area, but on opposite, area imagery has very lower area, but the resolution is very finer than satellite. But nowadays, it become changed. Because according to technology development, the resolution of satellite imagery also become more finer, less than 1 meter. It means the satellite imagery can be good alternatives for aerial imagery. It's much cheaper and more efficient. But in this project, we use the 3 kinds of data sources. First one is a commercial satellite, national satellite, and aerial and UAV. The reason why I use the 3 kinds of data is also the problem of the cloudy days. Because cloud hide the remote sensing, especially from satellite. So we needed to analyze what is the anomaly degrees along to the cloud day. So we derive from satellite and drive satellite and drone imagery. The first one is the marine debris heat map in the center. So it indicates how much the marine debris focuses on the region. And the right one also one of the heat map and the report. So by making this kind of report, we can support and make decision making. Also, we analyzed about source analysis. There was 4 source vectors as in the red triangle: traffic volume, fishing hours, catch volume, and precipitation. So as you can see on the right map, there are priority the region sources. So it is divided to layer 1 to 4. So by using that, we can provide some this kind of map to support decision making. Second project is AI-based marine debris management system. So this is one of the system scheme infographics. So we needed to make a realistic marine debris investigation system that purpose was making or implementing clean fishing village. So as I mentioned before, drone and satellite, in additionally underwater acoustic, underwater vehicle, those kind of platform additionally considered in this project. So the data first collected from process from the utmost left, and then second marine debris big data construction, and thirdly AI-based meta-analysis predictions have implemented, and finally the decision making support will be happen. According to debris type, the data source also must have difference. So when it comes to the coastal debris, we use the satellite and drone, and the floating debris we use satellite because it is very far. It is very difficult to find using the drone. And when it comes to submerged debris, we use the sonar. So those kind of 3 kind of platform were utilized to investigate marine debris. This is drone RGB image. As you can see, this RGB image can be used by their shape and color and make a result to the classification and detection. However, there is also difficulties only color and shape. Because of each material have their optical spectral characteristics. So we use the hyperspectral imaging for the plastic, orange V, and styrofoam, as you see. So we finally made such kind of time series graph after detecting the marine debris, for example, white styrofoam and orange V. So we found there is some seasonality, the marine debris occurrence. Next is A-based satellite data sub-expansion project. Why we should consider satellite in this time? That is because of number of satellites by orbit. Nowadays, the satellites more and more increased. So that means the more frequent, more cheaper satellite can be used in the future. And become higher resolution satellite imagery, as I said, it can be a good alternative for the aerial imagery. The light one also satellite imagery, but has a resolution less than 1 meter, 0.3 meter. And the light is about the case we apply the satellite. The first one is marine debris, and the light is port monitoring. When it comes to port monitoring, because the ship is very fastly moved, satellite detection is not much effective. But how about the marine debris? They mostly stable in their position, so it is very effective to use satellite, and because it is very periodical and regular. So we needed to make the smart marine debris monitoring in Iceland. So we needed to made smart marine debris monitoring in Iceland. The bottom side picture is about the Korean one island situation. There are a lot of the fishing gears. And we also made some sargassum expectation using the satellite and the numerical model. And the light one is the format of the report. So also we are made those kind of aquaculture maps, and the light one is detecting illegal fishing area and change detection by using comparing official announced data and the difference imagery. So our vision is to making that kind of subscription-based administration support, especially for Korean local government, Shinhan-gun. So now only they check their environmental condition manually by visiting the place. But we are now making this subscription service, and they can monitor those kind of the regions by AI-based smart marine management without visiting them. Also, we are now creating environment and economic value stationary to support sustainable ocean and carbon neutrality. So this is the last marine debris project. So we have started the pilot project until last month because they had a strong demand on a commitment and limited effectiveness national policies and lack of quantification national data. The right one is provided by the Indonesian professor in the Udayana University, Indonesia, Bali. And we received this information because he said they visit the place and check quantity in ice and make the record. So the map is made by during 1 year or 2 years, whole years. So it requires much time and human resources, money. That was some problems, so they asked us to help them. So now plan to support marine debris response. So we are very relieved and looking forward to cooperate with those kind of items with you guys. And so firstly, I just mentioned satellite-based marine debris source and the system and the marine source, marine litter interpretation facility, and 2 autonomous marine litter collection robot, and marine litter treatment facility, and 1 carbon reduction impact qualification system. I think those kind of items is very attractive, and I'm looking forward to cooperate with you guys later. This is a little bit different theme, but I think it is very interesting for you guys. We had a POC pilot project of the concept with Samsung Logistics. This is about the African logistics between the Democratic Republic of Congo and Zambia. We found there were many truck congestion in abrupt. So the Samsung Logistics asked us to find out the reason of why the truck congestion happened. Because these kind of things, dispatch delay on the return route and at the border due to congestion. So analysis of congestion causing needed as truck congestion status and rest time. So we targeted our truck parking lot near the border checkpoint close to Kashumbalesa. So the Kashumbalesa truck point was key border crossing and the frequent truck congestion and delay. Lot expanded 26 to 2024 to ease congestion. So we checked and the region how the truck parking lot is, the dimension is increased. The left one animation is about that, and we also found that the article in our newspaper in the website. So there was some contract between Zambia Revenue Authority and Zambia National Service. So that article was very coincident with the left animation. So our hypothesis was truck congestion border will make a parking lot stay full for longest period. Parking lot expansion work will make delay relieved after completion. So on the below, the left side, Kashumbalesa, and the right side, Kashumbalesa before expansion, but right one is after expansion. And rightmost image, 2 images also describe before parking lot expansion and after parking lot expansion. So you can see the left side has many trucks congested, but right side there is no. So our vision is also have the monitoring, the global monitoring logistics by using satellite from end to end, from source production to customer. Closing. So we are qualified the skills for investigating marine debris with satellite and drone. So I want to be a partner, you guys. And additionally, we have 4 core patents among 20 satellite patents. Also, we have a global network. Surely, we have a network in Tanzania, and also we have Asia and other countries. Our company has vision from the underwater to space and marine data analysis. Thank you. Asante Asana.
Thank you very much, Dr. John. His company name is UST 21st, Underwater Technology 21st. So, as I mentioned to you, Korean presenters, with regard to the Korean presenters' question, I'll summarize end of this section. Thank you very much, Dr. John. And for NEMCP4, so he made a very humble the presentation, but frankly, he already made a real PCP for NEMC project. Only 1 page is very big. They have a very big vision and people departure from Korea, they have a connection with the KOICA and the Ministry of Oceans Fisheries. I think I cordially advise you to have a sit together after the session. Okay? Thank you very much.
Thank you, thank you. These are all good stuff. I remember to have posed a question to someone from Ghana, I believe, 1 year ago, on whether we can, if someone throws plastics at sea, whether with the satellite we can pinpoint where that debris was dumped, and then triangulate with the currents and wind speeds and determine where it is going to land so that it can be collected before it lands. And I think that's a legitimate question, and I believe it can be done. Now, please allow me to welcome Mr. Zahor from Zafiri, who is going to present to us, just a moment, who is going to present to us about seaweed, seaweed nursery as a solution to address seaweed seed shortages in Zanzibar. And Bwana Zahor, please take the stage.
Good afternoon. My name is Zahor Malim. I'm from Zafiri, Zanzibar Fisheries and Marine Resources Research Institute. I'm a researcher. So I'm here on behalf of my colleague, Abu Bakar Norman, to present our work. So our work was seaweed nursery farms as a solution to address seaweed seeds availability in Zanzibar. Before going further, I would like to extend my profound gratitude to KOICA, Korea Republic, for their support of this project from 2025 to 2026, almost in a year. Yeah, thank you. Seaweed farming is a vital livelihood activity in Zanzibar, supporting coastal communities and contributing significantly to the blue economy. So currently we have a lot of initiative from Revolutionary Government of Zanzibar to ensure that we are going to boost our economy through this very important sector, especially for seaweed farming. So currently the government has been initiated a lot of efforts, such as extensive training for the seaweed farmers, distribute the seaweed raw materials such as ropes, seeds, ropes and boats in order to support this sector. But however, there are a lot of initiative has been done currently by the government, but still we have confronting with a huge challenge of seeds availability in Zanzibar. So this study, conducted to ensure that for the seaweed farmers there is seed availability and continuous supply for the seaweed farming. And this was done by collaboration with the Korea government through the KOICA program. Yeah, This is a seaweed production trends in Zanzibar from 2020 to 2025. You can have a look. There is a significantly increasing in production from 2020 to 2025. Now we are here. We are producing 20,075. This is data from 2025. We expect for the 2026, the production will be highly increased. Yeah, because not only Korea, but also we have another international organization like TNC, the Nature Conservance, IFAD. Those are international organizations we are collaborating in Zanzibar in order to boost the blue economy sector, especially seaweed farming and other blue economy program, especially fisheries. So objectives of this study is to establish the nursery farms to address the shortage of seaweed seedling availability for the seaweed farmers. To achieve this, we had 4 objectives, specific objectives. The first one was to identify the suitable sites for establishment of seaweed nursery. And we also have a specific objective about the issues of monitoring seaweed growth performance, water quality monitoring, and assess the health of seaweed in the farms. Also to prepare the seasonal production calendar aligned with the local climate condition. And the study area. This study was conducted in North District A in Unguja, which is also found in North Region in Unguja. We had 4 villages selected in this program, which was Tumbatu, Bwereu, and Kandwi. And the selected village, it was Pwani Mchangani. Materials and methods. So this study, before operate this study, we conducted a baseline survey to identify various challenges facing seaweed farmers. So we did interview 150 seaweed farmers interviewed to assess the challenges, especially the area where they are farming, if there is any problem such as maybe disease, ACS disease, temperature rise, salinity. So those are the kind of question we interviewed the farmers, and also we assess the willingness for the seaweed farmers, if they are really and capable, to intervene or to cooperate with this program. And for site selection, before starting this program, we also identify a base site for establishing of nursery farms. So for the site selection, we're capable to identify the water qualities of the area, especially temperature, salinity. Also, we observe the issues of social interaction, because you can't identify, you can't establish the seaweed farms in the areas where there is various ocean users, such as fishers. So you will experience a lot of challenges, such as maybe wiping the ropes for the fishers when they are passing the areas where there is farms. So we also identify area which is user-friendly for establishing these farms. So based on these 4 villages, select both on for these 4 villages, Pwani Mchangani was selected areas because it surpassed other 3 villages based on our study. So for nursery establishment, 4 nurseries established. In each nursery farm consists 90 meters square. And because the area of Pwani Mchangani characterized by having a strong ocean current, so we decided to establish the farm with at least short kind of rope with a meter, 60 meter. But each farm, we make 6 ropes per each farm. Because this is also, it was a pilot study. So we didn't make a big areas for farms. We established 4 farms, but in a small areas. And we also used 2 kind of ropes. First one, it was ropes for tying the pages, which was 80 millimeter. And another one, it was 1 to 80 millimeter for tying the seedlings. And before planting our seeds in the ocean, we managed to measure the ropes to see, and after harvesting, to see the growth performance. So in each ropes, we measure 5.2 kg. So for the monitoring of the farms, we make a specific routine for monitoring our farms by visiting during the spring low tides. So we also record the temperature, salinity, dissolved oxygen, and pH. And these were the result ranging from temperature ranging from 24 to 36 degrees, and dissolved oxygen from 5.2 to 9.3, and salinity 27 to 50, and also pH 6 to 8.4. So for every week during the low tide, we have visited the areas and to see this progress and growth performance of our farms. For the production result, we managed to harvest about 5 tons, approximately 5 tons, for all 4 farms established. But we observed during the phase 3, the production was significantly increased. You know, because in Zanzibar, some of these there is issues of environmental changes. So the nutrients in the ocean sometimes differ. So even the production sometimes also differ based on the nature of our ocean current and nature of the area we conducted this activity. So for the phase 1 and for the phase 2, the production is a bit slow compared to phase 3 and phase 4. After monitoring, we also managed, and harvesting, we also managed to distribute our seeds for the farms because that village is working in the groups, CVD clusters. So 395 farmers across 24 groups received the seeds in that region. And the distribution were done based on the production. For example, in phase 3 and phase 4, we distribute 5% for the production. We distribute the CVD farmers, and 50% also were remained for restocking for the continuation of the CVD farming activity. For the phase 1 and phase 2, because the production was not high, was very low, so we distribute only 25%. The outcomes. So based on this project, we observed a lot of benefit and some kind of challenges. One among the benefits is the issues of seeds availability. North region of Unguja is the one among the area where there was a very huge challenge of seeds availability. So due to this project, currently, what I'm speaking now, there is a continuous and availability of seeds because of this program. And also the issues of marine ecosystem health, because as you know that seaweed farming normally attract various organisms and provide a lot of organisms for the habitats, nursery grounds, and breeding areas. So it has been reported that these areas has significantly improved with their marine health, and also the knowledge and the skills transfer. Currently, there is a spilling over effect significantly increased. Not only the area initiated of this program, but also in adjacent areas in other villages. And environmental conservation as well has been significantly also improved. Apart from benefit, we had experienced some kind of challenges, such as the weather and the seasonal variability. That's why the production cycle, not the same. Some of the cycle improved and some of the cycles was not improved. And also the issues of distance and the transport cost. Some of the seed farmers came from different areas and villages, so experienced the cost of transport due to the distance where they were living and the area where they are established their farms. So recommendation and the way forward. So we recommend to sustain seed production, to encourage seed farmers to continue nursery-based seed production, because we believe when we establish these seeds in a specific farm, you will ensure the seed farmer will get not only accessibility and availability of seed, but also the issues of quality seed, which is well managed. Not only that, but also the issues of keeping, investing in training. Still, we need to spilling over effect the issues of this knowledge, because some of the seaweed farmers in not only Tanzania, but also in Uyo region, especially in the areas of Mombasa, they don't have that kind of knowledge of establishing of these nursery farms for improving their seaweed farming activity. And then an issue of replicate across, we encourage to replicate this activity, not only Zanzibar, but also in beyond. By saying that, I would like to say thank you so much for paying attention.
Asante sana, Zahor, and you have saved me 4 minutes, which is good. I will accommodate only one question. And while you do that, please remember all the presenters, we will have a group photo after we have done. So please don't go away. Any question? 2050 is around the corner and the blue economy is shining. You cannot discount seaweed.
Hello, thank you very much. I'm late, but it was very interesting to listen to this presentation. But I have one question. I want to know the source of parental stock which you are going to do this, in nursery. I mean the parental stock you started to do this nursery of seaweeds. Parental stock. You get me?
Parental stock?
Yes.
Okay, the issue of parent stock. We get this stock from the seaweed farmers. You know, before establishing these nursery farms, normally there is a seaweed farmers in that regions. So the stock in a initial stage we get from the seaweed farmers.
But I want to make a comment. If that's so, for me, I just advise if we can do the hatchery-based production, because if we source the parental stock from the farmers, the same story we have, we are sourcing from the past harvest, or we are sourcing from the neighbors of seaweed farmers, because for the issues of the quality biosecurity measures, because currently our main challenge of this production of seaweed to transfer from the sourcing from the local or from the past harvest, and then to come with the hatchery production or to do the tissue culture so as to get the quality seeds. Yeah, that is only my concern.
Yeah, to come up with your comment, actually, currently Zanzibar, we have established the laboratory, and we are coming with your point that we are now focused on the issues of tissue culture. And as I speak, I have two colleagues from Zafiri. Now they are in Philippines for the training of tissue culture. So we know that if we completely depend on this kind of stocks from the farms, normally the seeds, the same seeds when you cut and then for farming, it reaches the time the quality, removed. So that's why now we have that kind of a strategy to establish our tissue culture program in Zanzibar, and we expect to start very soon.
Thank you very much. Okay, let's have another round of applause. Okay, now we have another important agenda. So lunchtime is already started. So concerning that, so we have to finish this session by 1:30. So I would like to quickly introduce last Korean presenter, Dr. Gunyoung Kim from Korean Institute of Ocean Science and Technology, to make a present. Dr. Kim.
Good afternoon, everyone. My name is Gunyoung Kim from the Korean Institute of Ocean Science and Technology in South Korea. Today, I would like to introduce our Korea-Tanzania cooperation project and share the vision we hope to achieve through it. The title of my presentation is From Partnership to Sustainability, Building Tanzania's Marine and Fisheries Satellite Application Center. Tanzania has rich marine and fisheries resources and a very large exclusive economic zone. But at the same time, monitoring such a large marine area is challenging. Satellite technology can provide an effective solution. Satellite data can support fisheries management, marine environmental monitoring, and surveillance of illegal, unreported, and unregulated fishing. Therefore, our project aims to establish a marine and fisheries satellite application center, and more importantly, to strengthen Tanzania's own capacity to use satellite technology. This is a 5-year project running from 2025 to 2029 with a total budget of approximately $4 million. The project is supported by the Ministry of Oceans and Fisheries of Korea and led by KIOS, together with our technical partners, USC21 and Miresheum. On the Tanzania's side, Tapiri is our key partner in implementing the project. Our project consists of 4 major components. The first component is planning for the establishment of the Marine and Fisheries Satellite Application Center. We assess the current technological capacity in Tanzania, identify user requirements, develop a satellite data application plan, and prepare a master plan for the center. The second component is capacity building. Every year, we organize training programs for local experts. We also support academic exchanges and conferences. As you know, this is our first conference in Tanzania and Korea cooperations. In addition, 2 Tanzanian professionals will be supported through master degree program in Korea. The third component is strengthening the technical infrastructure and satellite applications. This includes the design of satellite data collection and management systems, technical support for research infrastructure, and enhancement of operational capacity. The fourth component is the development of an in-regulated satellite-based marine and fisheries monitoring platforms. The slide shows the overall roadmap of our 5-year project. We started with planning and capacity building, and we are now moving towards system development and infrastructure establishment. In the latest stages, our focus will shift to stable operation and strengthening Tanzania's capacity to manage the center independently. Ultimately, our goal is to build a sustainable system that can continue operating after the project ends. Now, I would like to introduce some of our key achievements in 2025. The first is the production of chlorophyll A information. A major challenge is optical satellite observation is missing data caused by cloud and differences in satellite observation time. We applied a similar approach to sea surface temperature. SST is one of the most important variables for both ocean monitoring and fisheries applications. Again, rather than depending on only one satellite, we combined information from multiple satellite platforms. This allows us to reduce data gaps and produce more continuous SST field. The third application is ship detection. For this purpose, we are using SAR satellite data. SAR has a major advantage because it can observe the ocean both during the day and night and under cloudy conditions. In the future, ship detection information can be combined with AIS or other vessel information. This has potential applications not only for fisheries monitoring, but also for IUU fishing surveillance and broader maritime monitoring. Another important part of our project is training local experts. Through training programs in Tanzania, we aim to develop satellite application specialists who can use satellite data for marine and fisheries monitoring. Ultimately, these experts will become a key people who operate and further develop the satellite application center in the future. So far, 23 participants have successfully completed our training program in last year. For us, the number is more than just training statistics. Each trained participant represents a future capacity within Tanzania. As these experts gain more experience, We hope they will not only use satellite data themselves, but also train other people. This is how capacity building on becomes sustainable and continue even after the project is completed. In addition to the local training programs, we also invited key Tanzanian personnel to Korea for more intensive and practical capacity building. In 2025, we organized 2 invitational seminars in Korea. The first seminar was for key stakeholders who are expected to contribute the establishment of a satellite application center. We discussed how satellite data could be used in Tanzania and what kind of system and services would be needed for the future center. The second seminar was more practical. We invited staff members who are expected to be directly involved in operating the center after it is established. This slide shows how the satellite information produced by our system will be provided to users. Users will be able to access satellite products such as chlorophyll A, sea surface temperature, and silt detection results through a web-based service. To provide these services continuously and reliably, we also need a stable system behind the user interface. Our end-to-end platform consists of 3 main systems: automated data acquisition, satellite information production, and data management. These systems work together to support the entire process, from receiving satellite data to producing and managing useful information. Ultimately, we want the satellite products generated by our system to be used in the existing potential fishing zone application. In the past, Tanzania provided potential fishing zone information to fishers using satellite debris SST and chlorophyll A data. Our goal is to reconnect this service by supporting stable satellite data from the Satellite Application Center. Through this integration, the results produced by our system can be directly used in a practical service for fishers. So this slide shows the final step of our system, turning satellite data into information that can be used in the field. But technology alone is not enough to make a project sustainable. We also need people who can operate, improve, and further develop these systems in the long term. For this reason, we are also supporting Tanzanian professionals through master degrees program in Korea. The first training, Mr. Juberi Harunam Juma from Tapiri, has been selected and will begin his program in the fall of 2026. The second training is planned to start in the spring in 2027. Along with training experts, we also need physical spaces for them to work and operate the system. This is the outside design, and I will show the interior. This slide shows the proposed interior of the Satellite Application Center. The operating and monitoring room will be main working space of the center. Here, staff will monitor satellite data, check system status, analyze marine and fisheries information, and provide operational service. Finally, I would like to share our long-term vision for this project. At present, our project focuses mainly on marine and fisheries satellite applications, and TAFIRI is at the center of the effort. However, we hope that capacity developed through this project will not remain only within the marine and fisheries sector. Once, Tanzania has trained experts, satellite data infrastructure, and experience in operating satellite-based service. This capacity can be expanded to other areas such as environmental monitoring, agriculture, disaster management, and climate change. And in the future, Tanzania could share its technology, knowledge, and experience within the neighboring countries and play a leading role in satellite application in Africa. So our long-term vision is from TAFIRI to Tanzania, and from Tanzania to Africa. This is what we mean by from partnership to sustainability. Thank you for your attention.
Thank you very much, Dr. Kim. As I mentioned to you, due to the time constraint, I will arrange the individual follow-up after the section. Please understand.
And finally, Changoma, yes. Yes, please. The final presentation, please, if you can make it quick, lunch is looming, and then we finalize. So Changoma is from the University of Dar es Salaam. He will be presenting on behalf of the University of Dar es Salaam plus the IMS on the proposed collaboration between Korea and Tanzania on marine research. And so he has some ideas on how we can approach this, and I believe you are ready to listen. Thank you very much. Ino. Oh, okay.
Hello, my name is Changoma Francis. As I said, let me take you straight to the presentation. Actually, as I said, we have the proposed collaboration between us and Korea. In this case, we maybe I will start. This is the title, Joint Situation of the Western Indian Ocean between Korea and Tanzania, and I'm presenting my team, as you can see from the number of authors. The title is Assessment and Monitoring of Environmental Parameters. As we may know, why an integrated fisheries information system? As we may know, we have this very productive part, and currently a lot of information is available in terms of fisheries data, environmental data, fishing activity, and national institutional data like TAFIRI, TMA, UDSM, and partner systems. And these are the examples that can be found in this area. So from the satellite information. But we need to do something. Tanzania has the building stock, how many blocks? Currently we have it, but they are dispersed. We have a SOAFI station, we have AUE station, we have Global Fishing Watch that can be also used. We have Korea TAFIRI collaboration, and we have TMA and the UDSM training that we can offer training and capacity building. But now the problem is data silos. So the applicated info and slow synthesis and also limited resource alerts. So these are the things that we need to intervene. So currently we did some simply international benchmarks which focus on the same thing, like you can see from PINA, AFIMS. They also focus on the information that can integrate management platforms. But also the information streams that they use, like vessel, position, electronic catch reports, observed information, and etc. But also they have the management value that you can see. But what can Tanzania learn from this? Is to build around interoperable data streams, shared identifiers, automated validation, and also listening products that has to be done, not just a database. So another benchmark was this one from the United States, multi-stream ecosystem information. And also it integrates the VMAs, e-reporting, electronic monitoring, surveys, and ocean data. The focus is to do integrated assessment and management, which at the end of the day, it can also inform about stock assessment, spatial measures and bycatch controls, catch limits like management measures, and also improve timeline and data quality. The third one, but not least, is the European Union, which is about control, traceability, and interoperability, which also use VMS tracking, ERS or e-logbook, lending declarations, and which include lending sales, and also central national systems. But also it has management compliances. And in this case, what do we learn from this? It's like we can use common data standard that we have, also we can do automatic cross checking between different data sources, make outputs which are useful for management, but also design traceability and governance into the system that from the beginning. So this is what we want to move from isolated database information. The other one is Australia, digital monitoring and data quality. What do they have? E-logs, VMS, GPS, cameras and sensors, and also central review. At the end of the day, they want to have independent verification on fishing activity, which is, of course, take them to better data quality, stronger stock and ecological assessment, as well as more defensible management decisions. So these are very important when we talk about the integrated management approaches in fishery resources. The last one is the Fisheries Transparency of Initiative. This is about the governance and public information. In this case, FIT standard, it actually focuses on, example, catches, fish population health, the vessel authorization, law enforcement. It also considers trade and consumption, fiscal management, and small-scale fisheries. But what we can learn, or what are the reasons to the Tanzanian concept, is that the shared information standards increase consistency across institutions, and we have to do public-facing information that improve accountability and trust. But also we have to make a multi-stakeholder governance, which provides a model for institutional coordination. Not only that, but also we can do a national fisheries information system that can combine an operational data layer with a transparent governance layer. At the end of the day, we can get this kind of a comprehensive approach when it comes to those kind of information. So what do these international examples have in common? From the first one to the fifth one. As you can see, there is data integration. They have the integrated data into multiple ways, and also they do digital reporting instead of doing manual reporting. They move from paper, manual workflow to structured electronic data. But also they validate, cross-checking independent sources to improve confidence. Not only that, but also the decision products, the near real-time capability, and also governance. These all take part in what we can learn from the example that we shared. So in our case, the proposed Korea-Tanzania architecture is we can utilize fisheries-independent data like catch, effort, landing, and logs. And also we can utilize the fisheries-independent data from surveys and research and biological samplings, but also integrate this with remote sensing, together with the fishing activity information, ocean water and weather and socioeconomic information that can, at the end of the day, have an output with a national integrated fisheries information system. This is what we propose from the Korea-Tanzania collaboration from our side. And the proposed analytical workflow can go like this, from raw data to management intelligence, like what we acquire, automatic feeds and social data, but also what we harmonize, like the standards, metadata, and the unique IDs, and also what we control the quality of what we get, and like the validation, gap checks, and cross-matching. All these require to take us to the analysis of the data, which can include stock assessment of the resources that we have, spatial, environmental models that can take part in with those kind of data, which has been recovered from our sources. But also interpret what it means and how it can be used, like the reference point, the status, anomalies, and trends that can also be compared to support resource management. At the end of the day, it can deliver an annual report. Also, because it's digital, we can have dashboards and some alerts. And this is how we try to show the flow. from raw data to management intelligence. And also a core assessment layer is stock status, fishing pressure, distribution, and reference indicators. But also at the environmental layer, it can be the SST, chlorophyll, currents, width, and anomalies that are very important to understand, which are integrated with stock status and productivity. But also the decision layer. From all the staff now, what we can decide based on the annual status, alerts, and management advice. Co-products for Tanzania, the proposed system should generate a small number of high-value products that the chain makers can actually use them. This can be annual fisheries resource status, like stock trends, exploitation pressure, uncertainty, spatial distribution, but also environmental condition report that can, like a key oceanographic and meteorological indicators, anomalies, which are very important. It's also a seasonal outlook. Not only that, but also the fisheries and the environmental alerts, which are very important, like threshold-based alerts and use of fishing pressure, or fishing activities into the area, and environmental anomalies or rapid spatial changes. And for the management dashboard, like we can use interactive maps, indicators, time series, and data provenance along with the user access levels. So from this now, fragmented information, we can get a routine national intelligence that can support resource management. Institutional collaboration. Okay, this is just to show how we expect this Tafiri, Korea, UTSM, TM, and other partners come up with a joint governance and cost sharing mechanism. So let me take you fast because of the time is so fast. So we have these expected outcomes and value proposition, like the operational and data integration and annual fisheries and the environmental status reports. We have the early warning capability, capacity development, and the long-term collaboration platforms. This is the end. In conclusion, we could just say that this is a practical opportunity for Korea and Tanzania collaboration. The concept built on existing data, institutions, and technologies, and focuses on making them work together. So Tanzania has already important asset, international examples we show, and also we can now go to number 3, that we can develop a platform and connect data and decisions. Thank you so much.
Asante sana, and thank you for making it. I know you had another presentation, but you made it to the room. And finally, I think Professor Kim, you need to do your finalization, right? So that people can run to lunch. But we will need to take a group photo of all the presenters with us, your superb moderators. Thank you.
Thank you very much. I'd like to thank you for your time and efforts to make this meeting meaningful and rewarding to each of us. I want to declare the side event over. Thank you. Asante sana. Everybody come forward and make a group photo.
work the IT.
In the meantime, I want to make sure that we capture all of the attendees. So I think there was a sign-in sheet that was moving around. Excellent. Yay, thank you.
So yes, if you're here for the workshop, please add your name to the sign-in sheet that's going around and come and sit forward so we can talk. Okay, great. For anyone who might be online, can you please unmute and let me know that you can hear me?
Yes, we can hear you.
Thank you, Lisa. Okay, well, why don't we get started? And thank you everyone for your patience and for being here today. It's a great pleasure to be at the FAR4ViBE event and to be able to offer this workshop. And so if you're here for that and you're up front, that's great. What this workshop is really designed to do is to provide some education around the types of techniques that we commonly integrate with limnology to get a better understanding of ecosystems and ecosystem processes. The instructors here are many, and many of the folks in the team have many years of working in this discipline. And we hope to communicate to you here today just the power that exists with integrating ancient data with modern data for thinking about ecosystems and fisheries. That's really the major objective for us as part of this process. And so by way of introduction, my name is Mike McLew. I'm a professor at the University of Kentucky. You can just call me Mike. That's the culture in my discipline is first name, so it's perfectly okay to just call me by my first name. And I'd like each of the folks who are participating as instructors here to stand up and say who they are and a little bit about their background so we can get to know one another.
Hello, everyone. My name is Tricia Spanbauer. I'm at the University of Kentucky. I'm an associate professor there. I study the intersection of ecology and geology, so aquatic ecosystems on temporal scales from days to thousands of years. I'm really excited to be here with all of you, and I will pass it on to Laura.
So my name is Laura Stribe. I'm a postdoc at the University of Kentucky and the University of Haifa. I work with diatoms that we're going to talk more about today a lot, and geochronology that we'll talk about. And I've worked on Lake Victoria and Lake Nyasa and Tanganyika.
Hi everyone, my name is Tumaini Kamlali. I'm a paleolimnologist. I also do hydrodynamic modeling. On the side of paleolimnology, I use diatoms TEX86 for temperature reconstruction, and I also did fish fossils.
My name is Joel Ogwang from the University of Wyoming. I study fish. I use genetics to study fish, especially for applications of conservation. So I will be taking you through some hands-on exercises on genomic analysis. Thank you.
And for the instructors who are online, if you could unmute and say hello.
Jeffrey, why don't you go?
I'm Jeffrey Stone.
I'm a professor at Indiana State University, and I study diatoms.
Hello, everyone. I wish I could be there in person, but my name is Lisa Park-Bausch. I'm a professor at the University of Connecticut, and I study astropods and ecological and biodiversity processing.
Great. Thank you, everybody, for those introductions. It would be great if the participants of the workshop could also stand and introduce themselves so we can get to know everyone here today. We can pass the mic around to do that. So if you could state your name and your institution and your research background.
Good afternoon. My name is Gaudensia Mgen. I'm from Tafili, Kigoma, laboratory scientist.
Hi everyone, my name is Irene Mhagama and I'm a laboratory technician from Tafiri, Kigoma.
Welcome.
Idris Arioba, lab technician from Tafiri, Mwanza, biotechnologist.
I'm Clever Nation from Tafiri, Mwanza, laboratory technician.
I'm Julius Kimario. I'm from Tafiri, Kigoma, a researcher. Greetings to you all. My name is Msengi David Gabriel from Tafiri, Kiela. I'm an aquaculturist.
My name is Dastan Godson from Tafiri, Kigoma, research technician.
My name is Zuberi Haruna. I am from Tafiri, Dar es Salaam. I study fish biology, but also stock assessments.
My name is Chande Mazola. I'm a research technician in Tafiri, Kigoma.
Hi, my name is Ignas Karupawe from Nojesville, Tanzania, Aquaculture Project Advisor.
Hello, the name is William Faraji. I work at Nkaba Biotech. So we offer different life science solutions as well as NGS service using Park Bio and ring technology. Thanks.
Hello, my name is Salum Chamwange, marine engineer, Tafiri, Kigoma. Interested to learn new things. Thank you.
Hi everyone, my name is Innocent Mwaka. I'm a socioeconomic researcher. I'm not using genetics. I'm not using biotechnology. I'm not using diatom to study anything. Rather, I'm using socioeconomics tools, methods to study the socioeconomic dynamics of biodiversity conservation and resource management within the context of fisheries and aquaculture in Tanzania. I don't think if we're in the lighted press, I don't know.
Hello, my name is Yona Mwakiluma. I am from Tafiri, Kigoma, as a researcher. The rest of my biography is like my colleague here.
Good afternoon, everyone. My name is John Daudi, and I'm a researcher based in aquaculture from Tafiri, Kigoma.
Good afternoon, everyone. My name is Priska Mzirai. I am a researcher working with Tafiri based in Kigoma. Thank you.
Hello everyone, my name is Jonas Nyandwi. I'm a researcher from Tafiri, Kigoma. I specialized in marine resources and ecology, but I do everything at Tafiri, Kigoma.
Good afternoon, everyone. I'm Alfred Mohongole. I work with the Fisheries Laboratory as a scientist. Thank you.
Good afternoon, everyone. My name is Tause Kitentia, an aquaculture and conservationist of fisheries resource, working with Tafiri at the Sota substation.
Hello everyone, my name is Jamila Ibrahim Kalambo. I'm from Tafiri headquarters. I'm a researcher working with aquaculture, but specifically mariculture. So I'm interested to study this because I'm interested in issues of breeding, so as we can know how to improve our farmed species breeders through genetics issues. Thank you.
Thank you. Did we get everyone? Everyone for the workshop is here. So just as a reminder, if you could put your name and email address onto the sign-in sheet, we can make sure to send you information as time goes by that will be relevant for you. So good afternoon and welcome, and thank you for coming. We hope to accomplish a number of things with the workshop today, and we have a brief outline here that just sets forth what we hope to do. And again, this will be very informal. We've had a couple of time changes and scheduling changes already, but the culture of our research tends to be fairly informal as well. So we can step through the material of the workshop at the pace of the group and talk together and discuss as much as we do lecture and exercise. And so this is the general format that we hope to follow for today. We'll start by talking about ancient data in particular, and how we can use sediments to better understand fisheries and limnological systems like the lakes that we have here in Eastern Africa that support the fisheries. Our project, the project that we're working on together collaboratively, and I should say, our team is from 5 different institutions in the United States, plus Tafiri collaborators. And so we are working together from a number of different institutions, really to use modern and ancient data to understand how the food web of Lake Tanganyika, and particularly the pelagic food web, has changed over time. That's our main objective with the project. And if you've been at some of the talks that our group has presented here at FAR4ViBE, you have perhaps a sense of the type of work that we do. And we hope to unpack that more here for you today, to think about how we think about these data in more detail. Because of course, 8 minutes for a presentation is hard to do anything, so now we have a bit more time and we can talk more about how we do it and why we do it. And so this is the general format that we'll follow today. We'll step through the background and the basics of lake systems and how we harness sedimentary data from the lakes and integrate that with modern insights to think about changes to the food web. And then we'll step through aspects of the food web progressively. So we'll look towards the bottom of the food web with their algae, and step through then our benthos, getting into the pelagic food web reconstructions using different types of DNA tools, and then we'll work on fish genomics and data analysis, and Joel will lead that aspect of the workshop and the seminar here towards the end of the day. And throughout, we can be talking about professional development and building our networks together. Okay, there may be some here who are interested in doing different career things, perhaps coming to the United States for studies. Part of the goal here with the workshop is to help understand that process and to meet people who are knowledgeable about it. And so in that way, we can help one another. We'll take a couple of breaks as we go. We realize it's a long day for all of us here, and we have perhaps different constraints of things happening here in the convention center. So we'll do our best to follow the schedule, but with the necessary breaks as we go. Okay. Any questions as we get started? Okay, great. So I'll ask Laura to come up and begin. And so if we can switch Tuma over to the next talk, the Lakes talk. That would be great.
Okay, so I'm going to start and just give sort of an introductory part to core science, sediment core science, as we get going. So we'll start with sort of the motivation behind studying sediment cores. Okay, great. So some of this might be review, but we've got different audiences here, so we're going to kind of start at the beginning. we good? Is it good now? What?
You want someone to share that screen?
No, I don't. So someone said there was a problem. I don't know what the problem is.
They can't see your screen.
Okay, I don't have a screen. That's not mine.
Somebody's sharing.
Yeah, so whose computer is that?
Yeah, that's your talk, isn't it?
Yeah, it's my talk, but it's not my computer. They want us to share the screen, but I can't. I don't have that computer.
Yeah, the IT will have to share the screen.
Can you guys see the screen online?
We can see it, but it's clipped on the sides.
Yes, that little.
Are we good? We good now? I think this isn't. Okay. All right. Sorry about that. Starting again. For why we study lake records. In any lake or ocean basin, organisms living in the lake and inflowing terrestrial material are being continuously buried. So when we take a sediment core, each layer represents a snapshot of the environment, and we can go back in time studying those changes. The vast majority of most of what we know about paleoclimate over the last 170 million years comes from ocean cores. We've got a plot here of marine. Comes from marine cores. But the limitations of those are that in the marine systems, sedimentation's rate tend to be lower. So if you're concerned about recent changes to systems, they're not very good for that. In marine systems, sediment sources can be unclear, and people live on the continent. So if you're concerned about climate or environments where people live, you need to study lacustrine systems rather than marine systems. So the vast majority of lakes that exist on the planet today are only around 15,000 years old, and they're right around our northern hemisphere, right? Because they were all formed during the last glaciation. So most lakes are only around 15,000 years, but our very oldest lakes tend to be formed in rifts, like the East African rift lakes we have here. And there's sort of sporadic volcanic lakes and meteorite lakes that can be much older as well. There's Lake Basumptwi in Western Africa is a meteorite crater that goes back around 1.1 million years, and there's sorted rift lakes around the world that can go back several million years. So what I'll do now is give sort of an introduction to all the different proxies that you can study with lakes, and some of the talks going throughout the session are going to cover these in more detail. So within a sediment core, there's a number of different types of proxies. The first one is biological proxies, right? So we have what organisms were living in our lake at the time. If we know what kinds of conditions those organisms like, we can then infer what the system must have looked like in the past. We can use those organisms to infer things like water depth, temperature, pH, salinity. And we can do that in a number of different ways. Sometimes just the organism itself, and sometimes we can measure the composition of those organisms' remains. Right? So a number of these different proxies. There's a typo there at the top, but pollen. The pollen produced by trees and grasses, they deposit in sediment cores. So you can use sediment from a lake to look at what kind of terrestrial ecosystems looked back through time. You can use data there to say, how did, say, our fishery change as land use around our system changed through time, if you were interested in that sort of thing. We have diatoms. You guys are probably familiar with those, but they're silica algae that preserve well in sediments and are used commonly in lake cores. We've got chironomids. Those are used more commonly in sort of high latitude or high northern or southern hemisphere lakes. They're flying insects whose head capsules are preserved. Ostracods, we'll hear more about those. They're tiny crustaceans. We can use the composition of their shells and the composition of their assemblage to study lakes, study the environment in the past. And then in addition to our biological proxies, there's a number of physical proxies that you can get from a lake core. You can look at things like the size of sediment grains, the rate of sediment accumulation to study what was happening terrestrially, like how much precipitation there was, how intense it was, those sorts of things. And lastly, we have geochemical proxies. There's quite a number of these. You can use them to study things like ecosystem and nutrient source. There are proxies for precipitation and temperature. Okay, so the next few things that we'll go through are sort of the basics of collecting a sediment core, how we make those decisions before we move on to everything else. So how do you decide where to take a sediment core? So generally, as people start to study a lake, they'll do a seismic survey and get a profile like this to find an ideal location for coring. Usually with coring, you want to look for, if you guys were in Mike's presentation earlier, if you remember, he got a question about why the cores are only 1 meter long. When you're taking a sediment core, you want to think about what time interval you're interested in, and to think about how far back in time you'll be able to go. Often you have to sort of find a balance between how deep you can core versus a resolution that you want. In different parts of a basin, sediment's going to accumulate at different rates. And so with things like seismic imaging, you want to try and get a handle on that before you take your sediment core. So I just have some pictures here. This is a seismic profile from Lake Nyasa, where they took a long 300-meter core, and they selected it using this data here. These are seismic lines from Lake Victoria, where we're thinking about a coring location. And that's the sort of equipment used there. And you drop it in the lake, and it pings, picking up those sub-basement profile. Okay. So next we'll get into the basics of how you collect a sediment core. The way that works, it's going to depend highly on the sort of length of record you're thinking about. You can take short cores of less than 1 meter, pretty inexpensively, just by hand. And it goes all the way up to really big, giant rigs that can cost millions of dollars and take big teams to collect. At sort of the smallest end, there's hand coring. If you're in shallow water, there are some tools that you can use. You can just walk right into the water and take the core with like a hammer and hammer that guy in there. And if you're interested in really recent changes, that's a perfectly fine way to go about it. As we get to bigger scales, there are gravity cores, like the one pictured here. This is us on Lake Tanganyika last summer, where you hand drop into the lake, and the gravity forces your tube down, and you kind of just collect as much sediment as the core is heavy to go in. We can scale up. We can get into piston coring. For these, you have sort of a floating platform, and it starts to get a little more complicated. It's not just gravity involved, but you have a hammer that you have to control to get deeper. This starts to get into the more expensive end of coring. And then as we get to here, these are on a lake that's liquid. But as in places that get cold in the wintertime, you can core right on an ice. So you have a solid platform. They'll drill a hole in the ice, and they go down through the lake in the wintertime. But at the very largest end, we have big drill rigs like this. Even with a massive drill rig, cores tend to be pretty small. You're going to only get a core like that big around. The larger the core, the more it's going to take to drill. But these can go quite deep. So the deepest are like a kilometer or so, I think, from lakes. And then with ocean drilling, they go even deeper. Okay, so the last bit here we'll talk about before we get on to the rest is chronology. So we've talked a bit about different kinds of records you can get from lakes. That data on its own can tell you about how that particular lake varied through time. But oftentimes what you're trying to ask is how your lake compared to others, how it compares to other systems, what's the driver of your system. And to do that, you have to have it on an age model, right? So if you have your lake and you want to compare it to another one, they both need to be in accurate time, right? So we're going to talk kind of briefly about the different methods that you use to data sediment core and a tiny bit about age modeling. All right. So there's a number of different dating methods. Some of them can be done on sort of any kind of sediment, and some of them require specific materials to be present, like volcanic ejecta and carbonates. Some methods, you're dating sediment directly, and there's some that are relative dating, where it's compared to pre-existing records. We'll just go through a few of the common ones. For people who study really recent stuff, there's cesium-137 dating. That's where nuclear testing and the Chernobyl incident leave kind of global spikes in cesium-137. And if you're studying just the last 100 years or so, there are these really clear spikes in records where you can date the years 1986, 1964 quite perfectly. But that's only useful if you're looking at really recent material. As we go further back in time, people are usually familiar with radiocarbon dating. With radiocarbon dating, that's useful from, say, 100 years to 40,000 years ago. It works in most lake systems where there's generally enough organic material, but sometimes in really low organic environments, it's not very useful. And the way that radiocarbon dating works is that living things, plants and animals, were in equilibrium with the amount of radiocarbon in the atmosphere. But when a plant or animal dies, it's no longer in equilibrium with the atmosphere, and our radiocarbon starts to decay. And to get an age, you look at a ratio through time of carbon-14 and nitrogen-14 to get your age. So there's luminescence dating as you get farther in time. This is useful from around 1,000 to around 500,000 years before present. This one's sort of newer in the lakes world. Luminescence dating works by any given mineral grain has imperfections in its crystal lattice. And any given sediment has radiation in it. As sediment is buried through time, that radiation is going to impact those sort of imperfections in our crystal lattice, and it causes what are called free electrons to get stuck in electron traps. But what basically happens is that when you expose sediment to sunlight, all those electrons snap back into place, and it releases light. And you can measure that light to figure out how long your sediment was buried for. We've got tephrochronology. You can do this, sometimes that's from volcanic ejecta. Sometimes this is locally, and sometimes there's really big eruptions that spread volcanic ejecta around the world. These eruptions can be well known, and they can have a known chemistry. So where you take a core, you can study the chemistry of the tephra in your core. It's a layer like this. Under a microscope, it'll look sort of like broken glass. And you can, if it's a well-known eruption, you can know that age exactly from seeing that volcanic eruption recorded in other places. But there's also ways to date volcanic ejecta directly through argon-argon dating. That tends to be more useful when you're studying scales of 10,000 years or more, with the argon-argon dating part. As you get into deeper time, there's paleomagnetic dating. Through time, Earth's magnetic field has flipped and changed, and those flips and changes are recorded in rocks. This one tends to be most useful deeper in time. People will try and use it on sooner time, but the most recent flip was around 700,000 years ago, so that's useful when you're thinking on really long time scales. And we also have tuning. There's a standard curve for interglacial transitions. And what people will do most often in marine cores, but also in lacustrine cores, is you can try and identify those changes and match it to this curve to sort of relatively date your sediment core. This gets more common in deeper time, where there aren't many ways to directly date a core. And they end up with this. All right. But when you have a long sediment core, you'll end up with something like this. What we have, this is a core from Lake Nyasa that goes back, it's around a 300-meter core that goes back around 1.4 million years. And when you have a long core, you'll end up with something here where, depending on the different parts of the record, you have different ages. We talked about some of these. So we have depth here on our x-axis and age on our y. And as we move through our core, we have different kinds of ages. Right at the very top, we had radiocarbon and luminescence. And at the bottom of our core, we have paleomagnetic ages and argon-argon ages from tephras, right? So you have a whole mix of different age types. In the past, what people had to do to try and get an age model from this, in the past, before there was technology that we have today, right? You could maybe try and you can try and connect them here, right? But what that's going to do is put sort of false changes in your sedimentation rate. Or you could kind of try to draw a line straight through, but then you're not taking in the full assessment of each of your ages and you're not studying error. So what we have today are there are a number of widely available programs where they'll use sort of probability to run 1,000 or so potential intervals. So for this, what we have here is an age model, sort of median in red, and a program has run 1,000 possibilities through each of our ages. You get sort of a cloud of potentials. And then when you have a record, you can use that error as information. Right? So today, if you get into coring, there's a number of publicly available modeling packages that people can use to get something like this, where you have a set, these are radiocarbon ages, and you can run them through. All right. So we'll move on to the diatom part next. Are there any questions on that? Sort of general sediment stuff?
Any questions? One important thing to know about harnessing the sedimentary record for fisheries management or biodiversity and conservation work is the last points that Laura was making about the chronology. Okay, the reason we emphasize chronology in the work is the constraint of knowing time, right, allows us to know how fast or how slow ecological processes are happening. Okay, the rates of the processes are really important and fundamental if we're to implement management strategies around them. So for example, if there were changes happening to Lake Victoria or Lake Nyasa that only happened every 1,000 years or every 5,000 years, those are not changes that are easy for us to practically address. If, on the other hand, the changes are happening every several years or every decade, well, in the course of our lives, we can prepare for those outcomes. Okay, and so from the perspective of this conference and how we think about using the sedimentary record, the highest resolution chronology is really what we seek to achieve. Okay, we really seek to understand the sediments in sites that are accumulating very, very rapidly, and therefore we can get very tight constraints on the amount of time contained within the information. Okay, because that really opens up the possibility to understand the rates of change in the system. Okay, so that's a really kind of fundamental aspect of this process. Any questions on sediment? Any questions on how we collect it? One thing for everyone to know is that we have a lot of tools now at the Kigoma station for a sediment program and for coring. And so these types of techniques can be applied quite readily here and now. And those sorts of tools could be replicated elsewhere in the Tafiri system. Okay, it's relatively as Laura mentioned, it's relatively easy to core different aspects of the lake as long as you don't try to achieve something very, very long. Okay, if it's a shorter core, it's quite achievable. Okay, yes, sir.
Right, I would like to know, we have seen several methods that are used in the process of sediment collection. What I would like to know is what method amongst the mentioned method is most commonly used, and also which one is the easiest one to be used or adapted to.
So there isn't really an answer, I think, to the ones that are most commonly used. It depends on your question. There's people who specialize in all sorts of things. We've got a few paleoecologists here that study diatoms, but geochemistry is very big. There's kind of pros and cons to each proxy you use, and it depends on the system. So like diatoms are quite inexpensive. So if you have a microscope, you can do diatoms. There are lots of geochemical proxies now, but they require equipment that can cost millions of dollars. So depending on your resources, they might not make sense. But there are lots of paleoecological proxies that you can do on sort of a small scale. And like grain size, there's a number of things. It depends on what you're doing. Like grain size, paleoecology, they tend to be more accessible perhaps and easier to get going with. And they can still tell you a lot about the system. There's a whole lot of geochemists now, but that's it takes maybe a larger project to do. But yeah.
Yeah, there's many different options for pursuing paleolimnology, and as Laura correctly said, it really depends on the question you're trying to pursue. For example, we see in the Lake Tanganyika watershed quite a bit of deforestation on the landscape. If we want to understand the nature of the deforestation and the effect that it has on sedimentation, we really only need to know things like the rate of accumulation, the grain size, and perhaps some fairly simple chemistry of the sediments to know specifically about the erosion problem and what effect it may be having on the nearshore ecosystems. So the technique that you choose really depends on your question. But for conservation and for management, there are a lot of very easy to access tools that can tell you a lot about how the ecosystem is changing. Inevitably, the hardest thing to achieve is the chronology. That's the most time consuming and perhaps the most expensive. But through our partnerships, we can provide some of that information as well. So that's, I think one goal that would be great for Tafiri is to grow the sediment program, and we can work in collaboration to help facilitate those analyses that you don't have at the lakeshore. So, any other questions? Yeah.
It's not a question. So simply we can say that sometimes the nature, features, and the characteristics of an area of interest where you want to pick or collect the sediment may decide on what kind of a tool you're supposed to use, right? Yes. All right. Thank you.
Okay. Okay, why don't we move on to the algae component of the workshop today. Could the IT people please bring up the stone talk? And I'll go there just to help. And Jeffrey, as soon as you see the slide, you can unmute and start, and we can support you here.
Okay. Also, if necessary, I actually have a copy of this slide here. I could just share it.
Okay, we can see it on our side, Jeffrey.
I can't see it though.
Can we project it to the online group as well? Share.
Yeah, that'll work. Okay, perfect. Is it okay for me to start, Mike?
Yep, go ahead, Jeffrey. Thank you.
Okay. It's a good introduction to why we need to look at lakes that Laura just gave us. And one of the things I wanted to say is there's a sort of a high density of diatomists in the room over there, and also me here in the online version of it. But diatoms are really important tools that we could use in lake systems, and one of the reasons probably so many of the people there and here are diatomists is that they're sort of a cornerstone for the tools that we use in lake systems, and one of the reasons for that is because they're very sensitive to sort of a wide range of environmental conditions. And I'm going to show you some examples of that. If I could get the next slide. So one of the things I like to tell people about diatoms is they're generally invisible. What I mean by that is you can't see them without a microscope. But if you get enough of them together, you actually can see them. And oftentimes when you can see them, they don't look very attractive. So a typical mass of diatoms like this Didymosphenia geminata from a river system kind of looks like really gross when you're looking at it in the large scale. And this is one of the only diatoms that actually has a common name because it grows in these giant mats that are so thick that basically they cover over all kinds of surfaces in river systems. The common name for this diatom is rock snot, which is a pretty accurate description of probably what it looks like, kind of gross looking. But if you were to look at them a little bit closer in a microscope, one of the things that you'll find is they're actually quite charismatic. So next slide. Here's some examples of what they actually look like on the in the sort of green colored version over there on the right is what it looks like in a microscope. In a living condition, you can see that it has chloroplasts and grows on a stalk for this particular type of diatom. But on the left-hand side are some images from the light microscope and also an image from a scanning electron microscope. And I think one of the things that you'd find is looking at them in the microscopic scale are actually typically very charismatic. They come in a wide range of shapes, and this is actually the cell wall that we're looking at in the sort of black and white images, and the living organism on the other side of the screen, the color. Go to the next slide. So what are diatoms? They're a type of algae. People usually say they're golden brown algae or a brown algae because of the color. In nature, they kind of have the color that you could see in that image, a kind of a gold or orangish color to them, not the green color that you usually think of when we think of algae. And what's diagnostic to sort of recognize that you're looking at a diatom versus something else is they have a siliceous skeleton. So the cell wall is actually made up of silica, of biogenic silica, and it's precipitated over the organism in their living state, and then when they die, the skeleton is left behind. Like other types of fossils, this actually makes them very useful. One of the things that's sort of characteristically good about diatoms is they reproduce very quickly. They have very short lifespans. They're highly sensitive to whatever environmental variables that they're growing in, whether that be things like depth or light conditions or nutrient conditions or pH or water chemistry or heavy metals. And so because of their extreme sensitivity to all of these different environmental variables, it means that that they're useful for reconstructing something when we look at them in the fossil record. And so the other thing that's nice about them is because their skeleton, this cell wall, is made out of silica, that's a very resistant material that preserves for a very long time. And it means that we actually get diatoms in the fossil record that go back millions of years. And so, again, valuable for all of these reasons. Sort of what I've described to you is an ideal tool that we can use for analyzing what past lake conditions might be like, because they'll give us some sense of all those environmental variables just by the type of diatom that's growing in those different environments. The next slide. Diatoms have an interesting life cycle. Unlike most organisms which start small in their juvenile stage and grow to be larger and larger, diatoms actually in their juvenile stage are at their largest point, and because their skeleton is made of rigid silica, and they form new cells inside of the old cells and basically just divide in half, giving one of the valves from the parent to each of the children. What that produces is an organism that gets smaller the more times that it reproduces, because it's reproducing asexually and it's just cloning itself. So most of the populations, and the reason that they can become such a large part of a lake system is that asexual reproduction or cloning yourself lets you actually grow very rapidly into these environments. The problem with being, an asexual organism that is cloning yourself is that it's pretty hard to adapt to the conditions in the environment if they change. So if that's a good condition, that's fine, but if conditions change or if there's something that you need to do to evolve in that system, sexual reproduction is the pathway people or organisms generally prefer for that sort of approach. And so what's interesting about diatoms, when they get to very small sizes, they can switch over to sexual reproduction, and that's the way they typically regenerate to their larger size. So this is sort of a unique pattern where they start in their large form, they get smaller and smaller. They get to some minimum size, and then something in the environment gives them a cue that it's time to undergo sexual reproduction, like their size plus some environmental condition. And then basically they will switch to sexual reproduction. And this allows them to colonize environments very quickly, but also allows them some capacity to evolve and adapt to those conditions. Next slide. One of the things that I mentioned about diatoms is they come in a wide variety of forms. They also have different types of life habits. In other words, they can live in colonies in terms of like clusters like that Didymosphenia that we saw at the beginning, or they can live as solitary organisms. They're found in basically all types of aquatic environments, and the number of species of diatoms is unknown, but estimates are somewhere in the range of 2 million possible species. And so a lot of the diatoms that you might encounter in the fossil record or in the modern record may not be described as species yet. Despite the number of diatomists in the room, there's actually not that many diatomists that are on the planet. And people who focus on this sort of field kind of limit the amount in terms of our understanding of the speciation of diatoms. And so that's one of the questions that we all work on, on this topic a little bit. So can you get the next slide? I'm going to go through some environments just to give you an example of where diatoms can be found. And I mentioned that they can be found basically in any environment. But I want to kind of highlight exactly what I mean by that. So this is an example of some diatoms that are found in Mono Lake, California, a lake system that Mike has worked on in the past, actually. And Diatoms can be found in this system in the modern environment, even though the pH of the system is toxic to humans and most organisms can't live in that environment. The pH is usually in the range of 10 or 11, so extremely high alkalinity and saline systems. And there's diatoms that can live in those conditions pretty easily, and the ones that do are usually diagnostic of high alkalinity systems. Next slide. On the opposite end of the spectrum, this is some diatoms that were found in a small creek that's here in Indiana, very near where we are right now. And the pH in that creek goes typically from somewhere around 0.5 up to about 4 pH. So it's on the other extreme end of the pH system. And they're living in the current system in the stream that comes directly out of an old coal mine. And the pH in that water system is very acidic as a result. And this is a diagnostic diatom found in acid conditions. And again, as an example, they can live in this range of salinity, of pH anywhere from basically 0.5 all the way up to 11, which is an extremely wide range, and most organisms basically don't have that sort of a span. So next slide. Also, if you're thinking about what about the water temperature, there's diatoms that have been found in thermal streams and thermal hot springs, and they're living on environmental conditions that are really at the limit of photosynthesis. So around 75 degrees Celsius, organisms that photosynthesize can no longer photosynthesize at all, like the physical process and chemical processes of photosynthesis start to break down. But diatoms can live in these thermal hot springs, and there are communities of living diatoms have been found in all kinds of very hot systems where most organisms can't live. When it gets a little bit hotter than where diatoms can live, it switches over to things like archaea, which are very simple organisms, relatively speaking, that can manage these conditions. So next slide. Again, on the opposite end of the spectrum, there are diatoms that currently live in sea ice, in the ice shelves off of places like Antarctica. And sea ice is a really interesting condition because when the seawater freezes, it actually purges the salt from the water itself, meaning that the diatoms that are living in between the ice crystals, and they're still living, you can see they have their chloroplasts and stuff in the image there, it's like a brownish color. Those are living in things that are in conditions that are colder than normal freezing water because it's salt water, and then it's cold enough to purge the salt out of the water, so it's also a hypersaline environment. So diatoms living in these really extreme conditions where it's not only below freezing, below what we think of in like normal sense of freezing, but they're also in hypersaline and freezing condition. And so this is basically the other end of that crazy spectrum where diatoms can basically live from one limit of extreme cold to extreme heat in aquatic systems. Next slide. Diatoms also live in many habitats what we would consider to be subaquatic, and what I mean by that is they're not wet all the time, and many examples of this are things like lichen and mosses, which basically live in these subaqueous environments that get wet when it rains and then basically maybe sit in a shady area, but can completely dry out and then get wet again. We have some current work where we're looking at diatoms that live, for example, on the backs of turtles. The turtles are only in the water some of the time, and then basically they're out basking in the sunlight, drying their shells and stuff. And the diatoms that are living in those systems are living on the backs of the turtles. So they can be in these sort of temporarily wet environments, and diatoms can be found living in things like tree bark and moss and lichen and the backs of turtles. And in fact, the sides of buildings where there's a rain spout or where water pours down the side from a gutter, for example, often lead to short-term blooms of these diatoms that can live in these really extreme dry conditions even. And will still be preserved in fossil records even in lakes if some of that material is carried into the lake system. Next slide. Just to sort of prove a point in terms of the temporary types of environments that diatoms can live in, these are diatoms that were found living in tank bromeliads. So these are plants that live in tropical areas where the rainfall is captured in a small sort of cistern-like shape in the center of the plant. And that water has almost no nutrients. There's no measurable nutrients in most of those water systems because it's just rainfall, basically, that's collected in the plant. And people have found diatoms living in these conditions. You can see some examples of the diatoms that are there, were collected from tank bromeliads in basically unmeasurable low nutrient conditions. Diatoms will also be on the other extreme example in places that have been enriched by people in eutrophic conditions where there's a lot of extra nutrients. Diatoms can easily grow in those settings as well. So this is sort of just giving you a broad range of where diatoms can be found in sort of any condition where there's water. And not only that, in each one of these cases I've given you, the diatoms that are living in those conditions are indicative of either low pH or high salinity or cold temperatures or warm temperatures. So those environmental variables actually play a pretty large role in shaping which type of diatom is growing in that environment. The next slide. This is another example of this sort of subaqueous diatoms that can be found living even in places where there's barely any light. So on cave walls that get wet, for example, where there's openings where sunlight come in. They've also found diatoms living in places where man-made lights have been installed into caves for tourism and stuff. If they get wet and there's light there, basically diatoms will grow in those environments. The next slide. So again, what I'm hoping that you take away from this is that diatoms are valuable for us in fossil lake sediments because they have this sort of excellent potential for being preserved. They have these extreme wide ranges of basically any aquatic habitat where they can be found. They're very common fossils that are found in lake systems. They're the base of the food web, so they're basically what the medium-sized organisms and some of the larger organisms like snails and fish eat. And so they're basically, like sort of the grass of lake systems that many organisms depend on. Another advantage of diatoms over most other proxies is it does not require a lot of sediment. So if you're collecting material from a core, the smallest piece that you can pick off with a toothpick or like the size of your fingernail or something is enough sediment for us to analyze and clearly identify what kind of environment was there in the past. They grow very quickly, and also as we've seen looking at their evolution through lake records, they can evolve in a matter of thousands of years, so much more quickly than people probably assumed evolution was occurring. Many of the species have sort of a cosmopolitan nature. In other words, we find them in wide distribution around the world. Although in places like Africa, there's actually some narrow distributions for things that are only found in the continent of Africa, for example. They have very short lifespans. They have rapid ability to colonize environments, and they're known for their sensitivity to these environmental parameters. So, what I've given you is basically an ideal fossil that has the capacity to be captured in any system across any type of environment and will be a pretty good, reliable tool for us for basically backing out what those conditions likely were. Next slide. Another thing that's kind of interesting about diatoms is that they're, I sort of view them as being like excessive organisms in that they will just try to consume as much nutrients as possible. They typically can outcompete, if there's silica present, basically all the other types of algae. And a consequence of this has been observed even in really long-term records. We know diatoms likely evolved in the Cretaceous near the Jurassic period. And one of the things that we see is that for a very long time, the amount of silica that was found in the ocean was a little bit higher. And then when diatoms evolved, effectively, their skeleton and their growth basically stole all of the silica out of the ocean water because they were one of the first organisms to come along and grow very quickly and rapidly colonize the ocean. And one of the things that we see is that there are other organisms that lived in the ocean that grew more slowly that used silica skeletons, but their skeletons became lighter as soon as diatoms evolved in ocean systems. So this is just an example to sort of highlight how rapidly diatoms can grow, how quickly they can take over an environment, and how much of an impact that can have on other organisms in those systems. So next slide. Diatoms have a number of different strategies that they use to grow, and we utilize this when we're trying to figure out what do they mean, and it's actually really critical for us if we're looking in environments that don't have a living example. So if I'm looking at something that's 1 million years old and there's no living example, we can use basically their skeleton to give us a sense of how they live. Many diatoms have no ability to crawl around, although some of them do, and we can utilize the sort of shape or the elements of their skeleton to tell us kind of a lot about how they were living. One major way that they grow is that they're just plankton. In other words, they float in the water. As long as there's nutrients present and the water has some circulation, the diatoms will stay afloat. And then they sort of resist sinking by creating sort of like structures. They're sort of like parachutes, basically, that baffle their descent or slow or regulate their descent. Many times they can actually make this adjustment so that they don't fall out of the light completely. They stay in the upper part of the water system where the light is available, because they need light in order to grow. And so we often see them hanging out in places like the boundary between the upper waters and lower waters in lake systems, where the water is a little bit denser and they can resist sinking. Next slide. Another type of strategy that diatoms will use is the sort of life habit called miroplanktonic. In miroplankton, diatoms basically want to be in the water column when there's high nutrients, but they don't want to be in the water column when there's low nutrients, because they can't live under those conditions. And so they're very sensitive to how much silica is in the surrounding water. They can create these very long chains that are denser than growing by themselves. And if the water is circulating and there's a lot of nutrients, they'll be in the water column. If the water stops circulating, then basically they sink out of the water column. And they intend to sink out of the water column because they can build these structures that basically allow them to survive being in anoxic conditions through a process called encysting. And when they do this, basically they can survive in darkness for a while, and then when the lake starts to mix again through wind disruption or something, they're carried back up into the light, and then they're available when there's nutrients available again. So they have this sort of distinct strategies that are a little bit different than true plankton, where they just want to be in the water when the water's mixing. And what that tells us is that we could use these diatoms to indicate when lakes are mixing, and they actually give us the ability to reconstruct things that other organisms can't, like how windy the environment is. Can I have the next slide? Another major way that diatoms live is attached. As I mentioned, many of them can kind of crawl around or move around on substrates, so they can be attached to things like plants, rocks, mud, other diatoms, other types of algae. Many times those things are actually diagnostic. So I'm showing a picture of the genus Cuccanese here. Cuccanese actually has, characteristically lives on other types of plants or algae. And their body plan or the structure of their skeleton is actually designed to be attached to something like a plant. And so we know that if we find them, that there are probably other plants in the environment. Just to give you an example, they occur in shallow waters in relatively high concentration where there are plants occurring. But other benthic organisms might specialize in rocks or sand or mud or something very specific, and then we can utilize that to reconstruct what that environment was like in the past. Next slide. Just really quickly, a tool that I usually use for diatoms is using them to reconstruct things like lake level change. Using those big broad categories of whether they live attached or whether they live free floating in the water is a pretty good tool for us for indicating what the lake level was like, because stuff that lives attached has to be attached where there's sunlight available, which means necessarily they live in shallow water where the sunlight can reach the bottom. In the deeper water, the sunlight can't reach the bottom, so they can't live in those environments. And things that are floating in the water column basically can live anywhere the water's deep enough for them to float. So next slide. So I approach this in a number of different ways. Usually things that are floating in the pelagic environment produce these specific sort of euplanktic organisms. Things that are in the littoral zone, in the shallow water, are very specific shapes and very specific types of diatoms. They indicate basically that the lake level was lower. If we take a core in a lake, in this example, the little sort of rectangle on the bottom of the lake is meant to represent where a core would be taken. When the water's deep, basically, we're going to capture a large amount of plankton. When the lake is shallow, those littoral zone diatoms basically move closer and closer towards where the core is collected, meaning that there's going to be a higher percentage of them. And we use this in something called plankton to benthic ratios to sort of figure out, like, how deep a lake was, and we can see the lake's response to changes such as drought that might affect the lake level. So next slide. Just to highlight this, you can kind of see that concept, the high lake level, there's going to be a lot of planktic diatoms and not very many benthic diatoms reaching the bottom of the lake. So the blue stuff is plankton, the orange stuff is benthic. And as the lake level lowers, we're just moving those environments a little bit closer towards the center of the lake, which means we're capturing more benthic diatoms in that setting. And then the proportion that we would see in the sediment would be then biased towards more benthic diatoms when the lake level is low. Next slide. Here's an example of that from a lake in Montana that I've worked on. Under normal conditions, when the lake is relatively deep, we get plankton. You can see this sort of round diatoms that are in the top panel here in the picture. The bottom panel, you can see these sort of kite-shaped diatoms or diamond-shaped diatoms have completely taken over the sediments. These live in shallow waters that have slightly elevated salinities, and so for this particular lake, it's a good indication that we went from normal conditions to drought conditions, and we can capture that in the long-term record. Next slide, please. Just looking at the proportion of the two different types of diatoms. Here's an example of that. You can see the green line is the planktic diatom index, showing us where we had a ratio of high to low diatoms, and the brown lines that are on there are actually the Palmer hydrologic drought index, which give us a sense of shallow versus deep lake systems. You can see the diatom response to the drought. Basically, when the drought is low, we have planktic diatoms in abundance. When the drought is high, we have benthic diatoms in abundance. Next slide. I also used some three-dimensional tools to try to get a sense of these different things by looking at diatoms in different types of habitats. So this is an example of using lake level change using three-dimensional modeling of the lake, just using the bathymetry and trying to map out different types of habitats. So next slide. Here's an example. The blue areas on the top are where benthic organisms are living under different depth conditions. So as the lake level lowers, the amount of benthic habitat changes based on the bottom shape of the lake. On the very bottom, you can see a row of all the different planktic habitats, so basically where water is deeper than a meter. And I can just use the model to change the lake level height, and then I can compare the amount of planktic habitat versus the amount of benthic habitat, just using some basic modeling tools. And it will give us some sense when we're reconstructing things like lake depth from diatoms to try to get a sense of whether there would be more planktic or more benthic habitats available. Next slide. Diatoms also can measure things like changes in salinity. So this is an example from some work from my PhD advisor, Sherry Fritz, looking at a lake in North Dakota where she reconstructed the salinity of the lake using diatoms. So as the lake starts to become shallower, one of the things that happens is the salts get trapped in the lake, and so they can't escape when the water evaporates. And as a result, the water becomes saltier, and the diatoms will respond to that. So this is just showing us the sort of transition that we see in the diatom inferred salinity versus the changes in the measured lake level. Next slide, please. So this is on the left is one way that we use diatoms as a tool. We figure out what their optimum condition is, which is the dot, and the range in conditions that they might live in for salinities. And then basically we can apply this in sort of like a weighted averaging technique. So if I find a lot of things that have the sort of like euhaline or polyhaline signal, it means that likely with that system has become more saline or euhaline. And if I find a lot of stuff that might have that sort of oligohaline system or low salinity system, it indicates that the lake had preferred lower salinity conditions, and you can see how people will use this tool to actually reconstruct not just that salinity has changed, but also the actual amount that it's changed by. Next slide, please. Diatoms also pretty good indicators of things like nutrients and stratification. I don't want to dwell on it too much on the next slide. So diatoms require a lot of nutrients in order to do well in conditions. They have to have salinity. Typically phosphorus and nitrogen are the other primary elements or nutrients that they need. We use this oftentimes to reconstruct stratification in lake systems, because when the upper waters are stratified, it typically means there are lower nutrients available for organisms. When it destratifies, when lakes mix, basically they mix nutrients from the deeper water into the upper waters. And so this pattern allows us to actually reconstruct what the stratification of a lake system was like using diatoms. In the past, even when we don't have diatoms, even if we don't have those diatoms living in the lake system anymore. Next slide, please. So in a dimictic lake system that we have, in temperate regions, it looks more like this. The lake completely mixes in the spring and the fall. In summer and winter, it stratifies in different ways. This is not the pattern that we typically see in places like Africa, of course. But it doesn't really matter what the pattern is so much that we might utilize the system. So when you have a windy season, a monsoon season, then basically the lake will be mixed. There are usually times where the wind doesn't blow very strongly, where the lakes will become stratified. And so if there's a long periods of stratification versus long periods of mixing, we'll see different types of diatoms growing in those lakes based on the nutrients. Next slide. We use all kinds of tools for this. So some work that I've done in the past, we actually can try to use some modern ecology to sort this out. This is some work I did together with colleague Jasmine Saros. And we looked at trying to reconstruct things like how much light, temperature, or access to nutrients affected different types of diatoms, just by looking at the diatoms that live in a specific lake. This is Hidden Lake in Montana in the US. Next slide. What they did is we grew diatoms in cultures at different depths to try to get a sense of how much light and how much nutrients affected, and then they put different types of nutrients in bags that were grown on these different floating sort of platforms that are here to give us a sense of the change in nutrients and the change in light, and incubated the diatoms or grew the diatoms in these different conditions. So next slide. What we found is that depending on the type of diatom, they might have grown better in the upper water or in the lower waters. They might grow better with some combination of nitrogen and phosphorus, and then the C is just a control. So this is an example from our growth experiments. This is the first one, a diatom called Discostella salidra. So I can go to the next slide. What you see is that depending on what type of diatom it is, how it grows and what depth it's growing at actually plays a role. So stratification and the amount of nutrients actually can be combined together with light to affect how well diatoms can grow. This is another diatom, Comensis, for example. Here's the next slide. Here's another diatom that was also in those same systems, and you can see that there's no interaction between depth and nutrients for this diatom, for example. So the nutrients stimulated the growth, whether it was in shallow water or deep water, relatively equally. So we use these together to give us a sense of how well the lake stratified, what kind of nutrients are produced. And then what's cool about this particular project is then I collected a core from this system, and we used these same diatoms in that core material to actually reconstruct what the conditions were like. So next slide. What we determined was basically when the lake was completely mixing, we for how deeply the water needs to be mixed in order for us to reconstruct what the conditions were like for them. And it's based on some combination of light, nutrient, and depth that they grow at. Next slide. This has been applied basically to a very wide number of different environments to show how climate has changed through time. This is some work from John Small's lab where they showcase basically systems that where Arctic lake systems are basically being converted from their natural conditions, or more recently natural climate change in these Arctic settings where things that normally would like to be well mixed. So an example in the bottom panel here from Lake of the Woods, Ontario is showcasing a transition around 1980 where the lake jumped from Olocosiro, which liked the lake to be completely mixed because they're miroplankton, to things like Cyclotella, which like the lake to be mostly stratified. And so we can see these transitions across a number of different lake systems that basically give us a sense that climate's changing and the diatoms are responding to this through how they mix in the water. So next slide. One other thing I wanted to mention is that diatoms actually are good indicators of eutrophication. So as people modify landscapes, it'll actually change the types of diatoms that can grow. And you can see they can do the same kinds of tools where we find an optimum, which is the dot and the range of the diatom, by looking in their modern settings, and then use the weighted averaging technique to actually reconstruct how much phosphorus is in an environment or how much nitrogen is in an environment. These are pretty common tools that people use diatoms for, to reconstruct and put a number on things like how much nutrients have been added to a lake system. Next slide. This is just an example showing that diatoms that like elevated nutrients become more abundant as we move towards the top, showing some cultural eutrophication or addition of nutrients to the environment as the landscape changes. Next slide. One last thing, diatoms can also be used as an indicator of pH, as I mentioned. We can see them in across a wide range of those. So you're going to get the next slide. Again, the same model. We use different types of diatoms from modern environments to reconstruct the pH that they prefer, and then we can use these to basically figure out, like, as the lake becomes more enriched with acids, for example, it increases or decreases the pH, and then the diatoms will respond to that. So they're extremely sensitive to things like acid. We use them in North America largely for reconstructing things like acid rain or acidification of lakes that happen as a result of industrial practices that put a lot of acids into the atmosphere. This also has that's fine, you can move forward. It also has some minor drawbacks for diatoms. When the pH gets very high, they're not very useful tools. Okay. So when pH is above 9, diatoms typically don't preserve very well. And so there's oftentimes heavy dissolution. It's at those times that we usually switch to some other tool that we might find in the lake record. So next slide. That's it for using diatoms in lake environments. If you have any questions, I'll be happy to answer them. Also, Tuma, Laura, and Tricia could also answer those questions.
Does anyone have any questions for Jeff about that?
Any questions on the diatoms? Yes.
Thank you for telling us about diatoms. And I want to know, because diatom seems like to have a lot of things which can support the productivity. So can we also use the diatoms record to estimate how fisheries were there before?
Yeah, I would argue that people often want to find the link between them and fish, because many fish actually, if they're planktivores, are going to require diatoms. And so it's sort of following the food web forward. If you add a bunch of nutrients, or if there's nutrients available for diatoms to grow, they're a food source, which means everything else above them will also have that food source. And so there is a link between food security and the growth of diatoms in lake systems. If you limit the growth of algae that is a consumable food for fish, it limits the fish that can live in those systems. Another thing I wanted to point out in this sort of response to that comment is diatoms are sort of like a nutritious snack for organisms that are hungry. And if they're replaced by something like cyanobacteria that can have toxins or are difficult for organisms to digest, that can create problems. So, there are no toxic freshwater diatoms, but there are a lot of toxic freshwater cyanobacteria, for example. So if you add too many nutrients, if the silica becomes limited, you actually will switch to systems that are dominated by things like cyanobacteria, and can be problematic for fish and other organisms. Thank you for the question.
One more question, maybe?
Thank you. Yeah, actually, I'm interested in these diatoms. So, considering how rapidly they are responding to the environment change, like they reproduce quickly, about up to 8 times in a day. They have short lifespan. How can their sediment records help us understand the long-term evolution of lakes and distinguish natural changes from human impacts? Thank you.
This is a good question, so thank you for that. One of the things I would say is when we look at a chunk of sediment from a lake record, for example, what it does is naturally average the sediment record for the however long of the time frame that we look at. So if conditions become more nutrient enriched, for example, over time, in 5-year intervals or 10-year intervals, what that means is that we will see different types of diatoms. So the assemblage or the types of fossils that are present will actually change. And the average state of the lake changes. That means that the average types of diatoms that we'd see in that lake also change. So if it stratifies for longer periods or more frequently stratifies, we'll find diatoms more frequently that would indicate stratification. So largely we just look at the ratio and think of it as being time averaged by the sediment record itself in the way that we collect it. If you're not looking at lake sediments, if you're just looking at modern systems, you can also just look seasonally from one season to the next, where they might grow. Even if they have relatively short lifespans, you could look every spring and say, what's blooming in that system this spring? What's blooming in the system next spring? Will give you kind of a sense of time averaging from modern organisms as well. So they can be used to monitor lakes and stream systems as well.
That's great. Thanks, Jeffrey. That was a great presentation. I think in the interest of time, because it's now 4:45 here, why don't we take a 5-minute break? We'll do a short break, and then we can return and we can resume with the workshop. Okay, so let's take 5 minutes and then come back, okay? Hopefully the online folks heard me that we're going to do just a quick break here and then resume.
Mike, is the benthos ostracods unit next?
Yes, why don't we do that one next, please?
Okay, are we going to do the same, you'll project the slides and I'll just tell somebody to advance them?
Yes, if you would just, like Jeffrey did, say it next slide, we'll sort it out.
Okay, and this will be probably, abbreviated, right? Because we're short on time, is that correct?
That would be great. If you could do 20 minutes, 15, 20 minutes, that'd be awesome.
Yeah.
Great. Thank you.
And then the last thing is my internet connectivity is a little wonky. So if I drop out, you know this stuff, so you'll just have to.
Yep.
I did drop out a couple times on my own, so just saying.
Okay, will do.
Hopefully it'll be fine, but.
Yep, no problem.
All right, great.
Hey, Jeffrey. He's good?
Yeah, I'm still here.
All right, just had a simple question to ask, at least for me to know. Like, I've seen that we have several diatoms that are adapted to live and survive in different conditions, from alkaline saline lakes, acid marine drainage, hot springs and streams, and sea ices, and so on. I would like to ask and get to know, have you ever encountered a certain group or kind of species of a diatom that exists in more than one adaptable environment? Like the one that is found in alkaline saline lake is also present maybe in a hot spring and thermal streams.
Yeah, so that's actually a challenge for diatoms in some level, is that, they might be growing in an environment, but you don't necessarily know what the control is that is causing them to grow in that environment if you don't know enough about their ecology. So I would say that, fundamentally, they probably can and often do represent multiple sort of extreme conditions or multiple variables that could play a role. Some people try to exploit this in a way, like if you were to look at the diatoms in a system, you could potentially reconstruct the nutrients and the pH, for example, from the same group of diatoms, because some of them have different controls for pH and some of them have different controls for nutrients, right? So the question is, what is the controlling component? And of course, understanding their ecology is the solution to that question. We don't have a lot of diatom ecologists. So, you know, like a lot of our understanding of diatoms and the way that we utilize them is through inference. So we know they're found in that environment. It probably means they do well in those conditions. And we have to look in the modern lake systems that are around a lake in order to use those tools.
All right, Jeffrey, thank you.
Test, test. Lisa, can you hear? Testing, testing. Lisa, if you hear me, let me know.
I can hear you.
Okay, great. Do
Can
you
you
see
hear
the?
me?
I can hear you.
you hold on one moment? It seems we're having a bit of an audio issue. Okay, you should be able to go now. Sorry about that.
No problem. Can you hear me?
Yes, we can hear you. Thank you.
Great. So thank you, everybody. I'm looking forward to talking to you about octocods. I'm going to focus this presentation primarily on octocods in Lake Tanganyika. So I thought it would be more relevant to what we're doing today. And this presentation will also build on Jeffrey's presentation on diatoms, in that octocods, also being living proxies in the lake, have some of the same features and things that can help us interpret what the lake is doing in terms of pollution studies and climate reconstructions and so forth. So, next slide. So, octocods, particularly in Lake Tanganyika, are part of the benthos, and if you're not familiar with that term, it just means something that lives in the bottom of the lake. So they're in the surface sediment and also in the subsurface layers, although octocods in particular only go perhaps in the, maybe in a few a centimeter at the most. They're typically not within the sediment, but on top of the sediment. So, so they're typical bottom dwellers, along with things like cichlid fish and gastropods or snails, bivalves, crabs, which are also crustaceans, and other invertebrates, as well as octocods. And for octocods in Tanganyika, they are particularly restricted to the upper 100 to 200 meters of the water column, where dissolved oxygen is available. So because they're restricted to this upper oxygenated zone, they're really important then perhaps for understanding changes in lake level and understanding changes in the what we call the oxycline, or that zone between oxygenated and less oxygenated water. So this is going to be, again, a bit of a focus for what I'm going to be presenting today. Next slide. So in Tanganyika, we know that one of its great features is the high amount of endemism. So those are things that only live in Lake Tanganyika and are not found elsewhere. And here you can see a chart that compares Lake Tanganyika versus some of the other African lakes like Malawi, Victoria, Edward, Turkana, Albert, and Kivu. And here you can see that well, Malawi has a lot of cichlid fish. It has fewer things like gastropods and bivalves, and about a comparable amount of octocods. However, Tanganyika is particularly special in the amount of endemism, particularly with respect to some of these other groups. So that is one of the reasons why we like to study octocods within Tanganyika, because they are so special in that they're evolving within the lake watershed itself. And so octocods live in rocky shores, sandy substrates, some of the deeper benthic zones, and they also live, in open water. And because of their specific habitats that they can inhabit, they're very good in interpreting changes in those habitats. Next slide. So what exactly are ostracods? They're extremely special microcrustaceans because they have a bivalve calcite carapace, and this carapace can be preserved in the fossil record and in the near fossil record. In addition, they have soft parts, much like a shrimp. So they're kind of like a shrimp in a shell, if you're not familiar with them. And here you can see pictures of various ostracods. And on the upper right, you can see different types of ostracods that have various noding patterns on their carapace or their cells. So this makes them easily identifiable down to the species level. The pictures on your left show living ostracods, and you can see that their typical way that they're living is with their valves relaxed so that their appendages can be out, and typically they're crawling or swimming along the bottom surface of the water. And the diagram in your in the middle there shows the different parts, soft parts of the ostracod, which I'll get to in the next slide. So next slide. Okay, so I mentioned that ostracods are small crustaceans, and I also mentioned that they have a bivalve carapace of calcite, but particularly they're low magnesium calcite. And there have been studies in the past where you can take their shells and analyze them with their magnesium content. You can compare the magnesium content to the calcium content within the shells and get a better understanding of the salinity of the water and the alkalinity of the water. With respect to their appendages, they have anywhere between 5 to 8 pairs of these limbs. You can see that on the right-hand side. They're very small, but not as small as diatoms. So you can see them with the naked eye. You can see them with your naked eye. They look like little flecks of dust, but you can see if they're there or not. But you do need a microscope to study them. Within all ostracods are found everywhere, from small little ponds and puddles to large lakes like Tanganyika. And overall, there's about 20,000 living species, and almost triple that or quadruple that species overall throughout the fossil record. They have a long fossil record, all the way down to the Ordovician. And in general, people do not know this, but they are the most abundant arthropod fossil in the fossil record. So they're extremely useful, whether you want to go in very deep time or whether you want to go in shallower time to use them as proxies. Next slide. So I like this slide because it shows not only some of our largest ostracods. So on your right is a Silurian ostracod. So you can see how big they can actually get. And then on your left, you can see how they can live interstitially between the grains of sand, just below the sediment water interface. So they're definitely interacting with the water column and interacting with the environment. Next slide. So if you look overall at ostracods worldwide, like I said, they occur almost everywhere, from lakes to ponds to swamps to streams. They can be in brackish water conditions, but also freshwater conditions, and they can be even at hypersaline lakes, like the one that you see down at the bottom right. That picture from the Bahamas, where the salinity gets up into the 300 parts per thousand. So they're very, very useful, and like I said, because they have calcium carbonate shells, they can be very useful, not just in looking at their abundance, but also you can use their shells as means by which to get oxygen isotope records, as well as magnesium, calcium, and strontium, calcium trace element ratios. Next slide. Okay, so once you get an ostracod, how do you know what it is? So I mentioned before, one of the things that is very useful for ostracods is their shells themselves, because their shells not just have ornamentation on the outside, but they also have a hinge at the very top, which we call the dorsal margin, that can be used to diagnose what family the ostracod is in, and sometimes you can get it down to the genus based upon the hinge elements, which are at the very top. They have a tooth and socket hinge structure. So one side of the shell has the teeth and the other side has the socket. They also have various features like muscle scars that are useful for also diagnosing what species and what genus it has. So there's lots of features on the shell itself to tell you what it is in terms of taxonomy. Next slide. And so here you can see that these are the different aspects of the outside of the shell. So I mentioned before the different superficial features. There's something called reticulation. There's things like growth like alae. There's pores. There's spines. There's little eye tubercles. There's sometimes there's a little sinking thing at the very top of the shell called the sulcus, and marginal pork mouths. So these are easily identifiable features that can be compared when looking up these shells. Next slide. I think, how's my connectivity? I'm kind of, is that okay?
Yeah, we can still hear you.
Okay, next slide. It should be the soft part slide.
We're on hard parts. Is that next?
Next one.
Great.
Okay, so if you have soft parts available, so if you're collecting living ostracods, the soft parts have a lot of different features as well that can tell you what species you have. So they have antennula, antenna that are up at the head area. They have both mandibula and maxilla, which are those are the feeding mouth parts of the ostracod. And then they have these limbs, and these limbs can be modified for swimming or for crawling, and those are sort of in the middle of the body. And then they have a number of appendages for mating, because these are, there are some asexual ostracods, but for the most part, they're sexual. So they have a furca, they have a hemipenis, and they have this thing called the Zenker's organ. So all these are readily identifiable. Again, in modern ostracods or living ostracods, you can see these features and be able to tell what species you have. Okay. My screen just went blank. I don't know if... Next slide, if you can. I just have colored lines on my screen. Not sure.
Lisa, we can see you here, but there appears to be some online technical difficulty.
Okay. So if you see a slide with a bunch of, it says classification at the top.
Yep.
Okay, great. So ostracods can be classified into different subclasses and orders and these things called superfamilies. And for the most part, in freshwater systems like Tanganyika, they're all something called podocopids, which are order. And within Tanganyika, there's about 4 or 5 superfamilies. I'm going to get to that in a moment. Next slide. Now, importantly, and very interestingly, ostracods have what we call isometric growth, meaning that they grow the same direction, and they do something called molting. So they have what we call 8 instars, so 8 versions of themselves. So it's the adult version that has all of the appendages that I showed you earlier, and it's also the adult shells. You can do cell chemistry. You can do the magnesium-calcium ratios and the isotopes. It's really important if you're working with ostracods in a geochemical setting that you're only working with the adults, because there's been some studies done that show that as the cell is progressing and it going from the lowest instar, the A9, all the way up to what we call the A1, which is the last juvenile, there is a shift from high magnesium to low magnesium in the calcite lattice. So it's very important if you're doing the work yourself, or if you're working in collaboration with somebody doing shell chemistry, that you're only working with the adults or the last molt of the ostracod. And you can know that by, there's a selvage margin on the anterior margin that only occurs in the adult stage. Next one. All right, so as mentioned before with the diatoms and some of the other proxies, ostracods can be used in a lot of different ways in sediment cores to reconstruct things like climate, to reconstruct pollution, lake level, and things like that. Here is just an example of, I'm going to go through this study in more detail from 2003. Ramon Allen and Andy Cohen did this study on lake level in Tanganyika, where they used ostracod abundance, which is on the right-hand side, to show changes in lake level in Lake Tanganyika. And as I mentioned before, we can do this through abundances. We can look at isotopes, trace elements in the ostracod shell themselves. All right, next slide. I mentioned Tanganyika has a lot of different ostracods in it, and there are likely about 142 species, which is a very high amount of species for a single lake system. We think that about 80% of them are probably endemic. And so there's 88 confirmed species, and they come from 7 families and about 17 genera. And so I color-coded these different families to see, so you can kind of see where the diversity lies. If you notice that a lot of these ostracods have sort of the genus name and then new species number 10 or 14 or 15. Some of the work in my lab in the next few years will be to reconcile some of these species and give them proper names. So this was work that was started by Andy Cohen. Some of you knew him, some of you knew him very well. This was his legacy that he started when he started working on Lake Tanganyika several decades ago. And so this is work to be done, and if people are interested in doing this kind of work with us, let Mike know, and it would be lovely to connect with some of you that would like to help out with this. As you can tell, there's a lot of work to be done. Next slide. So in the coming next four slides, I'm going to just show you some of the stuff that we've been doing with ostracod distribution in Lake Tanganyika. And here you can see the picture of the ostracod themselves, and then the blue dots represent their abundance or relative abundance at certain depths. And so the depths themselves go on the left-hand side, they go from 0 all the way to about 100 meters of water. And so you can just see that most of these species that are portrayed on this slide are fairly shallow water. They like shallow water. So if you're looking at this upper one on the upper left, Macinus Irascae, it likes to live in the upper 20 meters of water. So you'll see in a minute how this can be translated then into interpreting water depth from cores, because we know that this is in fact optimized for shallower water. Next slide. So these are some more ostracods, and you can see that some of these actually have a greater preference at slightly deeper water. If you look at the right-hand side in the middle, that's Gomphocystri alate, and you can see that that one is more optimized towards 20 to 30 to 20 to 40 meters of water, and can be found all the way down into 80 meters of water. So this one is more broadly distributed within the lake. And it's sometimes dependent also on the type of sediment, whether it's sand substrate, or if it's rocky substrate, or shell lag. But for the most part, these are depth dependent distributions. Next slide. This is just another example. And here you can see some of these, if you look, are like deeper, even deeper water. And then the next slide are more of the ones that really like the deeper water, particularly Gomphocystri cohenii, which is in the middle, and then in the middle on the left, and in the middle of the whole diagram is a species that we've been focusing on, Gomphocystri downingii, which, as you can see, is most abundant in very deep water. And interestingly, these things have sort of alae, or little wings on their ventral margin, the bottom margin of the shell, that we think allows them to stay afloat in deeper, soupier sediments. Okay, next slide. So getting more practical, if you're interested in ostracods and collecting ostracods, you can do it in a number of ways. At the very top of the slide are sort of 3 basic collecting techniques for modern and subfossil ostracods. So there's a collecting net, so you can just use a regular plankton net. You can have an adaptation where you can have a sample jar at the end of the net to help you capture the sediment. But you can use a collecting net, you can use scuba. There's a variety of ways you can collect using scuba, mainly with just a sample jar, sample bag. And then there's the sonar, which can get you in deeper waters greater than 15 meters. Once you have the sediment, We sieve them through two sets of sieves, 63 microns and 260 microns to capture the adult or the bigger ostracods, and then the smaller ostracods. And then we sieve them, and then we pick through them using just a petri dish, and we put them on microslides, so you can see in the middle there, using very low technology, a camel hair brush that picks them out and puts them on the microslide. Much like 4AMs, if you're familiar with 4AMs, we use the same type of material. And again, fairly low technology. You do need a binocular microscope to be able to work with these things. Next slide. Okay, so if you're interested in learning more about ostracods, there's 3 resources that I've listed here. One is Neotoma, and we will be putting in the Lake Tanganyika ostracods that are described in Neotoma, hopefully this fall. And then as we are working on more of the species, we'll be adding them to the database. There's also something called the World Ostracod Database, which is pretty good. You can type in any species, and it'll come up with a variety of resources for you to use to identify them and learn more about them. And then also the British Geological Survey has a fairly good website about ostracods in general. So those are 3 online resources that might help you to learn more about them. All right, Mike, how am I doing on time?
I think we're good, Lisa. If you want to wrap up there, we can maybe take some questions from the group.
Sure.
Okay. Any questions from Lisa about benthos, ostracods, and methods?
There's a hand raised in the chat, by the way.
Okay. I've got to walk across. There's a hand raised, but there's no message in the chat.
Okay.
I think it's unintentional. Any questions from the audience, from the group here participating for Lisa?
Hello, how are you? My name is Dr. Makame, and I just raised my hands a little more back. So, can you actually hear me now?
Yes, yes.
As I told you, my name is Makame, and I'm from Zanzibar, and I'm a director of fisheries in urban district. And I would like to know 2 things. Can I use ostracod as a bioindicator for making the database survey for if I want to make the fisheries stock assessment fisheries, or maybe there is a specific species of ostracod that indicate the stock assessment of the fish?
That's a very interesting and good question. So, so from what I understand, you want to know if you can use ostracods as bioindicators for fisheries stuff? Is that right?
Yes. Could I use the ostracod as a bioindicator if I want to make a database survey of marine fishery? I mean, stock assessment of the fish?
So we know, for example, Jeffrey had talked about diatoms as a major component to the food web. Ostracods are also part of the food web, and we know from my own work, actually, dissecting fish, is they are fish food. So fish do eat ostracods. So we also know from pollution studies on Lake Tanganyika, for example, that there are certain ostracods that do better in more heavily polluted, particularly sediment polluted areas. And so those would be the ones that are more living in more shallow habitats. So I would say that right now I couldn't point exactly to a specific ostracod to tell you what the indicator species is, but I think if you had a research question where you're trying to find out if there is an indicator species, I think that would be a really good project, a really good thing to try to understand. We do have data that show that certain ostracods do like disturbance and tend to be in a certain grain size, for example, or certain, they tend to be more prevalent in more disturbed settings, like a lot of runoff from rivers and so forth. So I think it's possible. I don't think that there is currently any ostracods that would be used as indicator species yet. I hope I answered your question.
Thank you very much. Thank you very much.
Thank you. I think maybe in the interest of time, we'll move on to the next presentation, which is the DNA presentation. Thank you again, Lisa. That was great. And let's move forward.
Thanks for hanging in there, everyone. Great. I'm excited to be here to talk a little bit about some of the work that I'll be starting on Lake Tanganyika. This is work that's collaborative amongst a lot of the people that you've heard speak already. And unlike a lot of my colleagues, I don't have a lot of data yet, but I'm going to kind of walk you through our project, then talk a little bit about ancient environmental DNA, how it came to be. It's not that old of a proxy. And then circle back to giving you some resources if you're interested in this field. They are also in that USB drive that was handed out, so there's a lot of literature there as well. So that's sort of mapping out what I'm going to be talking about today. I just wanted to recognize the USNSF that has funded this research project and also this workshop today. So thank you to them. And I just want to start off by talking a little bit about environmental DNA. Now, a little bit of caution here. I put in a bit of a description of my talk, and I had Gemini draft a figure. And when Gemini drafts figures, there's little tiny errors all over it, but it still is a very, very nice figure. And it really shows what the power of doing environmental DNA from the water column and coupling that with sedimentary DNA from the sediments at the bottom of the lake. And why we use these two together is because the environmental DNA we collect from that water column gives us a ton of information about what we might find in the past. We can get to those littoral zones, collect samples from there. We can go into the deep pelagic zones, collect samples from there, and use those samples to reconstruct the past environment. And over the past 2 days, we've heard a lot about the fisheries, some 60% drop in the fisheries from this is like a quiz for me, 98 to today, and that was a really alarming statistic, and disentangling why that is part of this research. And of course, there's overfishing, there is land use change, and then climate change. And climate change is the real impetus of our research in this group. And my colleague Mike McGlue talked a little bit earlier about a lot of how climate has changed in the Lake Tanganyika area. And so that was really the big impetus for this proposal that was put forward and funded through the NSF, and brings together lots of different proxies that you've heard about today, and then also ancient environmental DNA. All right, so just a little bit about environmental DNA. That's been on the scene for a really long time, so that top part of the graphic. I'll walk through just a schematic of the process. And I looked at a couple posters today, and there's already a lot of really cool environmental DNA going on in the region. So you guys are already engaged with this sort of process. But one of the things you can do is you can go out and collect water, and you can collect water from almost any type of environment, and then filter that water onto a filter. Then you process that filter, bunch of purification steps, and try to get the DNA cleaned up and ready for sequencing. It goes onto a sequencer, then you have all these A's, T's, G's, and C's. You've got to sort that out bioinformatically. And then you have a biological inventory of what's there. A very powerful tool. And this is just NOAA's depiction of it. They've done a lot of this in the marine environment. Okay, so moving on to the ancient environmental DNA. So a little bit different than the water column. This is not a sort of instantaneous snapshot. It gives us time by going back through the layers of the mud. One just quick note here is that ancient environmental DNA is also very fragmented and is not as intact as modern environmental DNA. And just a reminder to what you heard about before, paleolimnology collects all sorts of environmental proxies into the sediment. And we heard about some of those. Some of them can come from watershed, atmosphere, and lake inputs. And today what I'll be talking about is that DNA fraction. So a little bit more about modern DNA versus ancient environmental DNA. If you were to go and extract DNA from a plant, or even the water-filtered extracted DNA, those are going to be really long strands of DNA. As time and exposure to the elements, especially UV, so light, and then other processes lead to ancient DNA molecules being very short fragments. And not only are they very short fragments, they also start exhibiting damage patterns. Some of those C's turn to T's, and this is really helpful. It's a challenge, right? It's a lot better to have that really long genomic DNA. But when we have these little short fragments, we can easily, or more easily, start to partition what is ancient from what is modern, just by size. And that's what you're seeing here. You have DNA fragment length. When you're looking at ancient DNA, it really hangs out in that 25 to 65 base pairs. Now, when you're talking about genomic DNA that you might extract from fishes, those are usually thousands of base pairs to tens of thousands of base pairs. So this is a really good way of knowing you're dealing with something that's ancient, is the degradation. But also, on the 5' end and on the 3' end, we start to have deanimations. So this damage process happens on the little fragmented ends, and that can also help us differentiate what's modern from what's ancient. Because when you sequence sedimentary ancient DNA, you're getting a lot of modern microbes. They're still doing work in the sediments. So you have to differentiate those things that are active and living from that is dead in the past. And where do we collect ancient environmental DNA? All sorts of different environments. Now, our project, we're working sedimentary environments preserve DNA in the same fashion. So you have to find the right place to get sediments from to recover this ancient environmental DNA. And here's just a couple examples of coring. We've already talked about coring at length, so I'll skip this and move on. But that's what we're after, is that sediment. And then once we get the sediment from whatever depositional environment we're working in, we take it to an ancient environmental DNA lab. These are highly specialized labs that are clean room facilities. This is just sort of a diagram of how you would maneuver through this lab. Starting at the bottom, you would go into a gowning room, get fully dressed in a Tyvek suit, and then you would move into progressively cleaner and cleaner spaces within the lab. And here's what that looks like in pictures. Everybody here is in this proper PPE. We have to keep it hyper clean to make sure that we're not contaminating any of that ancient environmental DNA with modern DNA. And here you can see even that while I deal in sediments, a lot of people deal in fossils, and they will handle these fossils in these same clean room facilities to get the ancient DNA from them. All right, let's talk a little bit about sediment subsampling. Once you have your core, there's multiple ways to kind of get into that core and start to look at it. When you start to subsample, you have to make sure that you're not using any of the core material that touches the barrel. And so usually what we do is slice it down the center and then take a little like razor blades and cut out little sections, or you can see those circles there. That's using just a little syringe to really aseptically subsample the core. And then once we have that, we do extraction. And it's the same thing. If any of you have done extractions in the audience, same sort of process. What you're really doing is separating the DNA from whatever substrate it's in, and then several purification steps. But you have to be aware of sediment types. When we are coring, we often core through lots of different types of material, and all of those different types of material interact with DNA in a different way. For instance, clays really bind DNA very well, so you have to use a different extraction technique when you're dealing in clays versus something in a more sandy substrate. And there's also different kinds of DNA. I talked a little bit about those clays. That's usually the free DNA in the system, the extracellular DNA. There also can be intracellular DNA still intact in the cores. You can think of that as the pollen or plant materials or any organic materials still contained within the organism. So we attack both the intracellular and the extracellular DNA by using different extraction processes. And here's just running through that. A lot of times we use kits, or we will modify a kit. So this is just based on a Kyogen kit, where we have a bead beating step, and then we move through protein precipitation, start removing inhibitors, and then the ultimate goal here is to extract total genomic DNA. All right. Then the fun begins, right? You have your little DNA sample, a couple nanograms, maybe if you're lucky, 100 nanograms in that little tube, and you send it off for sequencing. And we always include controls. Negative controls are really important when you're dealing in small amounts of DNA. And then we take and send it to the sequencer. But there are many different sequencing strategies. The most basic is we just do library preparation, and library preparation just means getting it ready for the sequencer. There's some certain ligation steps that preps that DNA to go on the sequencing machine. And when we do that direct from the total genomic DNA, we can recover anything that's in that sample. But it's a bioinformatically more challenging output. And that I just depicted by this shotgun sequencing here. Now, most people take a more targeted approach when they're sequencing because shotgun sequencing is so bioinformatically challenging. So they will use a target. One of those targets is 16S or 12S or RBCL, and all of these codes just mean that they're targeting a specific group of organisms. Like 16S can target mammals, the TRNL loop targets plants. So you start to use some targeted region of the genetic code to pick out a group that you're interested in studying. And instead of sequencing everything, you're just sequencing that short bit of DNA. Now, a little bit more sophisticated and kind of a hybrid between total shotgun sequencing and targeted approaches is target capture. And target capture, instead of just using some barcode, maybe that would be a couple hundred base pairs, you could target the entire, say, mitochondria of the mammoth. And that gives you much more genetic information about what you're trying to reconstruct. Or you could do the entire genome of a fish. And then go into a substrate, even environmental DNA or ancient environmental DNA, and specifically only target those, that genomic DNA that comes from a fish. All right. So with lots of different types of sequencing, there's going to be some drawbacks and advantages to them. Let's first talk a little bit more about metabarcoding. Metabarcoding is really inexpensive. It amplifies just a short section of DNA. So that section tells us, like pine from like maple. And this locus is flanked by conserved regions, and it allows us broad taxonomic inference from a specific group. Now, some drawbacks of metabarcoding is that sometimes the DNA is too degraded to recover that barcode. Now, those barcodes can be anywhere from like 75 base pairs to 400, 800, 1,000 base pairs, depending on what region you're targeting. And the issue with that is that, remember that graph I showed? Most of the ancient environmental DNA is between 25 and 75 base pairs, so you're missing a large section of your DNA. And here's just a depiction of that. Here's that 25 to 75 base pairs, and most of the barcodes are much longer. So what if it's too degraded? People started moving towards, and there's where 16S is depicted compared to ancient DNA. Most people moved away from metabarcoding and started using shotgun sequencing or target capture. So again, some pros and cons of all of these methods. Metabarcoding is very sensitive and cheap, but it's unable to authenticate because you need that entire intact template, which ancient DNA often can be too degraded to recover. Shotgun metagenomics, this is really great in the breadth of organisms it can recover, but it's very hard to sequence through all the live microbes in the sediments. Target enrichment also is sort of an intermediary in both cost, but it is very specific to a certain organism or set of organisms to do. And a drawback to all of these is sometimes authentication, authenticating that what you're dealing with is in fact ancient. But with shotgun sequencing and target capture, you can use those damage patterns to authenticate them. And one drawback to almost all environmental DNA, ancient environmental DNA work is the reference databases that this all relies on. We really have to have a well-vouchered organism that's been deposited in some reference database to even be able to tell if you're dealing with one type of cichlid or another. And just to underline what I'm talking about when I'm talking about shotgun sequencing being not very efficient, here you have several very early shotgun sequencing studies from ancient environmental DNA. And you have the sequences generated in the millions of sequences. The sequences used for interpretation were in the thousands, so you're losing some 90% or more of your sequences when you do shotgun sequencing. This was a really big deal in 2015 because sequencing was really expensive. It is increasingly becoming less of a big deal because you can get billions of reads in a lane now, and now the trade-off isn't as extreme. When you're losing 99% of your reads, but they're so cheap to get, it becomes less of an issue. So shotgun sequencing has sort of come on the scene as being the most popular way of dealing in ancient environmental DNA. Okay, so I'm going to go through these really, really quickly. Again, these are on your USB, but each one of these studies has either that sort of shotgun sequencing, the target for target capture, or metabarcoding. And I just want to highlight this timeline because we're dealing in time that's not that long, right? We have 1998 being one of the very first ancient environmental DNA studies that used sediments to recover organisms in the past. 1998, like, some of you were probably still alive in 1998. That is not a very long history for a proxy. And they looked at 7 samples or something, and all purple sulfur bacteria. So we're talking about just one tiny group of organisms and a very few samples just some 20, 30 years ago. Fast forward a little bit, and they started working on caves. And they did this with pairing both bones and sediments using metabarcoding. Again, when you go through these slides, if you're interested on your own, anytime you see those barcodes, it's a metabarcoding study, and all of the references are included. Molecular caving became a really big deal. Going and targeting specific megafauna in caves and recovering from metabarcodes the organisms that lived in those caves. Fast forward a little bit more, we start reconstructing entire forests from Greenland. Fast forward more, and we have the last survival of mammoth and horse in Alaska. Still all metabarcoding. So now we're at about 2009. 2014, people are reconstructing livestock from lake sediments. 2016, more mammoth extinctions, but here we're starting to see shotgun sequencing. And this study was really interesting in that it precisely timed the extinction of an organism in the sedimentary record. And then shotgun sequencing started to be used to look at exploitation and hunting in the past. Target capture starts really becoming a really utilized technique because we started having whole genomes of organisms. So we could use that target capture to recover humans as well as mammal whole genomes. And it really became one of the more common types of sequencing strategies because a lot more whole genomes became accessible. And a lot of studies would pair metabarcoding with shotgun sequencing to really give a fuller picture of organisms in the past. And this is another forest example. Most of the ancient environmental DNA to this point has really been terrestrial in nature or focused on mammals. And then more DNA work up in sort of the Arctic and the subarctic areas. And you will notice a lot of DNA work is done in that region, and there's a good reason for that. DNA preserves much better in cooler areas, but more and more folks are recovering ancient environmental DNA from the tropics. And then I just wanted to highlight this study as one of the sort of last shotgun sequence ones because this was an Ice Age algal population that they could actually track some of the changes in the haplotype groups through time using shotgun sequencing. All right, so what are some of my approaches for doing work in Lake Tanganyika? I will be using both a shotgun approach as well as target capture. There's been a lot of really lovely work done on gut contents of fish that has really nice molecular databases, so that will be what we use in our target capture to start looking at the food web dynamics. And then pairing that with shotgun sequencing just as a way to make sure that we're not missing any of the biodiversity. And really what our focus is going to be is reconstructing food webs during past warm periods. Mike talked a little bit about that earlier today, using multiple lines of evidence. In these two schematics, we have a food web during warmer periods and a food web during cooler periods. And if you think back to Mike's talk, that has everything to do with stratification and nutrients within the food web, and whether or not there are more nutritious algal groups that then permeate up through the food web. And then we're using multiple proxies to kind of tackle that. And just like we're doing today, part of that work is training the next generation of students here and abroad. And really the impetus for all of this is to inform management. And so with that, I am all finished with my talk. I'm happy to take any questions you may have. I know I breezed through that really quickly, but it is getting late in the day. Yes?
Thank you. My question is on sample collection. You mentioned that you have to find the right place to collect the sample. I want to know that's...
What's the right place to collect a sample? That's a really good question, especially when talking about genetic information. Do you mean for environmental DNA or ancient? Okay. I think that is going to be really highly driven by your research question. Environmental DNA doesn't have as many constraints as ancient environmental DNA because it hasn't spent as long in the environment. When we're talking about collecting ancient environmental DNA, we have to think about that sedimentary environment and whether it's a good place to preserve DNA. With environmental DNA, modern sampling, you really have to be kind of question-driven in where you want to sample and make sure that you're capturing the organisms in their life cycle and in the right places at a right time. So just after this, we're going to be doing some water column sampling, and I've been thinking about this a lot, about exactly where to sample. And for my questions, I want to have the photic zone, focus on that, but I also want to be right underneath the photic zone to start understanding what's raining out of the photic zone. So that's sort of just an example of one sort of design for a research question.
Thank you. So just to get clarification on your last point, you mentioned that your work would, you think would be informed because there's very good data from gut microbiome from a fish. So my question is, to target the microbiome from the eDNA, or is to target the information about actually the population for the fish in there?
It's a fantastic question and really leads into our next speaker, who works a lot in understanding populations of fishes. I will include in my ancient environmental DNA bait set, so that's what's used for the target capture, some sections from fish. However, capturing, the structural variants and the SNPs needed to reconstruct populations sometimes isn't always accessible with the areas that we know are going to be good at differentiating in a bait set. So that has often been inferred from sort of modern genomics, is that population structure through time. However, moving towards understanding sort of evolution in situ could potentially be a possibility if you were to collect the right data and then get the right strata through time to track those populations. I mean, I think that's a really fascinating and interesting application, for sure. Fish DNA is hard to get, though. I've had colleagues who've tried. From sediments.
Thank you, Trish. You are collecting samples in a massive water body where you don't have reference libraries, and you have to collect DNA. How do you deal with this kind of situation where you have a sample, you are getting a signal, but you really don't have a reference to that species, or you don't have a physical sample that you can sequence?
That is a fantastic question. And when I started this project, or possibly even when I started talking to Mike before we were funded to do this work, I was very concerned with that very thing. There wasn't the references. It's getting better. There has been some of that gut contents work that I've kind of discussed that has laid down sort of some information. And you're exactly correct that a lot of that information doesn't have an exact match to an organism. Sometimes it's at a family level. But that's still pretty informative for us. And then there has been some more detailed studies where they actually went out and made a reference genome of a lot of the food that the fish eat. So that's going to also be useful to us. But then our field season just coming up right after this conference is another way for us to ground truth what's in the water column to what we're finding in the sediments. And sometimes we won't be able to name things, and there's statistical ways that we can use. We found this sequence here that can help us be, and the pH is this, and the chlorophyll is this. Then we can start matching at least environmental signals to some of those things that don't have taxonomic assignments. So that's like the least satisfactory, but at least some more information for those sequences that have no specific identity. But great question. It still makes me worried, and I think that you're not alone in the lack of reference genomes. I also work on the Laurentian Great Lakes, and I'm continually frustrated with, especially in the lower food web, how little genetic information there is. More questions?
Am I allowed to pose a question to Lisa? I believe Lisa is still online. Yeah, there was a question from Zanzibar on the use of ostracod.
I'm here.
Yes. So my question is, can we use multiple indicators and then form a proxy that can be used to indicate productivity and somehow answer the question of, I think the Shirley or from Zanzibar? Maybe a combination of diatoms, ostracods, chlorophyll, or whatever, and then make an algorithm that you can use as a proxy to indicate productivity and stocks of fish that this person was referring to.
Very possible. I mean, at least for the ostracods, we're working towards having a fairly comprehensive understanding of where these species live, not just with depth, but also in the different environments. And so the fish, it would be really interesting to see if that could be done. I think we're moving towards that in an optimistic way.
Yeah, thank you. My name is Salom. Yeah, I want to know which is the best way of using when you want to detect the invasive species, like maybe tilapia or other species, by using modern DNA and the environmental DNA, which is the best way to use.
That's a fascinating question. I didn't talk about a different sequencing strategy today, just in make time a little bit shorter on this. But another thing you can do just after extraction is use quantitative PCR or droplet digital PCR. Fascinating to think about sediment cores and invasions, and whether or not invasions that are detected, sort of by eyewitness and verification of taxonomy really coincide with detection via DNA, and whether or not one might precede the other. So that's a fascinating idea.
In many aquatic ecosystems, microbial loop has a greater contribution in primary productivity, especially the picoplankton. Were you able to incorporate them? I saw in one of your slides, you showed only the reconstructed food web, and you only showed the cyanobacteria. So I was wondering if you were able to incorporate the picoplankton. Thank you.
So, yes. It's actually quite fascinating. I don't have any data from Lake Tanganyika yet, but I do have data from a couple US lakes, including one from the Laurentian Great Lakes. And picoplankton have an insane contribution. And a lot of the unsung protists, very small green algae, very common. So there's a lot of hidden diversity within that extreme lower food web, the smallest of the microbes, that I think we're going to start to unlock a lot more of what they're doing through time as well. So I think that's a great point, and one that I've seen some evidence for. You're welcome.
Thank you. Actually, I'm an aquaculturist working in the aquaculture project, but my interest in Lake Tanganyika is profound because there is where I was born and raised. I was born in Lake Tanganyika Basin. So my question is, what will your study, actually environmental DNA, added to our current understanding of Lake Tanganyika biodiversity that conventional fisheries and ecological survey may not capture?
I think with each ancient environmental DNA study, and thanks again for that question, it's a good one. What do we offer? I think what we offer is baseline information that happened prior to monitoring. It gives us some context for trajectories and maybe thresholds of ecosystems through time that I think adds valuable context to our understanding of how things change. And that starts to be helpful to prediction. I don't want to promise prediction, but I think it starts being helpful for understanding what to expect. I think that's a good stopping point, and giving it over to my colleague Joel, who's going to talk to you about fish genetics and walk you through a bit of an example of some coding, right? Excellent. All right, so we'll just do a quick change over here, and he'll come on up.
I think I should be able to share here so that I have more control. So as we are waiting for the setup, we yes. Okay, I think that's excellent, right? Yeah, so as already been introduced, I'm Joel, and I'm a PhD student at the University of Wyoming. Today, I'm going to be talking to you about the applications of genetics to fisheries management. I think a lot of you probably are interested in genetics for different applications. We are going to have time to discuss most of them. And my original intention was at the very end of this small presentation to walk you through a hands-on coding exercise. But when I look around, can I see by show of hands how many people have their computers here? Or how many have R installed, R Studio? Just 2. So that means I am going to quickly show you how to run a few codes, and I've already shared the tutorial with you. I think a few people have got some flash disks. That tutorial is in there. Sorry. But the data sets to run, I'm going to share a link where you can download them. Okay? So before I forget, I want to acknowledge the people who fund my research. The National Science Foundation, Nord University, and several departments at the University of Wyoming, including the School of Computing, the Center for Global Studies, and my program, which is Ecology and Evolution. And not forgetting my lab, my supervisor is that one with a hat or a cape. All right, I just want to get... Okay, right here. And this picture is unique to me because this day we had gone on an expedition with the department to show people what we do in the program Ecology and Evolution, and we were showing people how to collect cocopods. I know nothing about them, but yeah, my supervisor does research with them too. So, on the application of genetics in fisheries management, we now know that there is wider global awareness of the role of genetics in natural resource management, and this is well recognized in major global frameworks, including CBD, So in its resolution, that is COP15 in 2022, they resolved to track genetic diversity in wild species and obtain genetically based estimates of effective population size. So you can take notes of what questions you want to ask. And then the FAO and other bodies. The FAO has a lot of tutorials online where you can just register and get certified. For a long time, they did not have genetics, but as of 2022, they introduced genetics as a course for fisheries managers. There are a plethora of examples of how to apply genetics in fisheries management. I'm going to highlight a few that may be of interest to you, including revision to extant stock boundaries. If you do not know where your stocks are located, you can use genetics to do that. I mean, by stocks, I mean different groups of the same species, you know, that occupy a particular sort of habitat. But you can also use it to revise species boundaries. If you're not sure whether this is one or two species, you can use genetics to ascertain that. Where morphology is hard to do. It's one of the things I'm doing in Lake Albert with the Nile perch. You know that Lates niloticus, which is like the most common perch, exists with another species which is Lates macrophthalmus. It's very hard to differentiate between them morphologically. So genetics is one way to do that. Detecting illegal fishing is one of the ways you can use genetics. You can do real-time management of stock components. I'm going to give you an example on this. You can do rapid assessment of species diversity and species richness. I think this is one area that eDNA excels. Yeah, environmental DNA that she just talked about. Estimation of effective population size. That is the center, rather the subject of my PhD, really. So just to help you define effective population size in the simplest term, imagine all of you, you are like 50, but I do not think all of you are willing to pass on your genetics to the next generation. There is an aspect of being willing, and there are many other aspects. So those who eventually pass on their genetics to the next generation, if we head count them, they become the effective population in this group. Assessing genetic diversity, which is like the most common things you know. So I will give you a real-time, like a real-world example of how to use genetics to do stock component assessment. So in Norway, there is this, the Atlantic cod, two stocks, one which is very abundant and then one which is rare, but they share a breeding ground. So the problem here was that when you go fishing and you catch, you know, this, you do not, like you catch this fish, both stocks will be in your catch, but you don't know the proportion of, you know, which one is the most abundant, which one is the least, because they look alike. But thanks to genetics, there is just a single gene that can distinguish between these two stocks. They are not two species. They are the same species, but different stocks, and they differ in one gene, and that difference arose because of an inversion. So using this gene, you can quickly detect the proportion of catch between the two stocks. So the outcome was, as you can see in the graph, an increase in the proportion of the abundant group when fishermen went fishing, while keeping the proportion of the rare stock down, rather in their catch. This way you're protecting that stock from, you know, further decline and depletion. It's a beautiful example that, I mean, this experiment was carried out of between 2017 and 2013, and they achieved that remarkable result. Another application is predicting future impact of climate change. There is something called genomic offset, which is the mismatch between genetics and the environment today. Rather, if you have a population in its environment today, how well does this population match this environment? And how will this match look like 100 years to come? Will there be a mismatch? So that prediction is called genomic offset. Just last week, I did a workshop to take people through this kind of analysis. So I have the resources I can share later. Now, assessment of stock structure is one of the most common things that we do. Whether you're doing conservation or breeding, it's something we all do. And I'm going to take us on a hands-on exercise on population structure analysis, and I'll be using the sardines or the dagger of Lake Tanganyika as an example. Well, before I go through this hands-on exercise, I want to hear from the audience which application of genetics in fisheries. I don't know where. So, for this hands-on exercise, the basic question we'll be asking is, how many stocks of dagger are there in Lake Tanganyika? And the basic analysis we are going to do is, a population structure analysis based on principal component analysis, which is a very common, dimension reduction analysis done almost with any kind of data. To access the data that I will be using in this example, you either enter this link or scan that QR. And if you had your computer and you have some experience with R, you could open the tutorial that I just shared and, just save it in the right directory, and you would be able to run the tutorial that I have here without any problem. So right now I will pause. I will give you some time to scan the QR. Okay, I think everyone has access to the data now. I will share something different. Okay. Yes? Oh, it is GBS data, which is targeted. Well, it's not targeted sequencing, but in a way, yeah, it's not whole genome sequencing, but it is reduced, what we call reduced representation, where we exclude some portions of the genome, like, the repetitive parts of the genome. Yeah. So still you get a lot of data. You get, millions of sites that you can work with. Yeah. I want to increase this window. How visible is the screen? It's good, okay. So In doing this kind of genetic analysis, if you are familiar with R, if you are not, then I will refer you to a tutorial where you can start from the basics. And I have that somewhere. I will share it. If you don't mind, I think I should include a QR on my presentation and share it later. Yeah. So what you do when you're analyzing data in R, generally, you'll need to load the packages that you're going to use. Yeah, so when you're doing this kind of analysis, you need to load these libraries. VCF is a library that has all the packages for reading genetic data, which is a .vcf file. And then Adegene and then Pegas all have functions that will help you to manipulate genetic data. And generally, a VCF as a library has all the packages. I mean, a package is like tools for doing mathematical or arithmetic stuff. Let's say I have this sort of data, I want to calculate the mean, I want to do what, it's like the full pipeline for doing a particular kind of analysis. They are all packaged together into let me start from here. I want to remove everything that I've done. This is a function to remove all the objects that you created in your environment. You just run that. If you see here, there were a lot of things, but they have now disappeared. So because I did that, I've also removed all the libraries that I had loaded. So right now I will run everything in this set of codes. Now I have all the libraries. Now I will read my files. So my files, I read using read VCFR. Now, R has a language which is very intuitive. If you were to read a table, can you suggest what the function would be? It is intuitive. It would be something like read table, like read dot table, you know? And to check that, I can quickly type read. It is there. If it pops up like that, it means R knows it already. So you can read your table like that. So when you do read data, read your file in any way, then you have to provide where your data is located. This is like the file path. So you provide the full file path, but best practice is keep your data, like set R, point R to where all your data is, so that you don't have to worry about the file path. And there's a simple way to do that. You can just go to session, set working directory, you go and choose the directory you're working from. Choose the directory that has all your data, so you don't have to worry about setting the full path here. And when you put in your data, you have to be very careful to include the file extension. So if it's a text, it has to be followed with a .txt. This is a gzipped file, so I have to include this gz. So this is how generally you read your data in R. And if I read all my data, remember I'm reading 2 data sets. This one is the genetic data for Stolothrisa, and then this one is for Limnothrisa. Now, we can, kind of play with the data to see the properties. For example, we can read the number of columns in that data, or the number of rows. And if I run this by holding Control and Enter, and then clicking Enter, I see that I have 100 rows. And when I do the same for, well, I have 100 columns. When I do the same for this line, I see that I have 11,107 rows. So there are some questions you'd want to answer. For example, how many variants are in these files and how many individuals? This is how you actually answer them. Yeah. So the number of variants is the number of rows. 11,107. Now, we can perform a PCA on these data sets to see how, structured in space these individuals are. To do that, we are going to convert our data into another kind of data which is called GenAid objects. And it is, as I said, R has an intuitive language. We are converting VCF to GenAid. You see that function tells you exactly what's doing. So when I read this, or rather, let me run all of them, I will create these 2 additional objects. You can see this path changing. Yeah, you see all the 4 objects are now created here. And these functions, rather the objects that I just created, if I run them directly, it gives you all the details. Here we have 100 individuals, which were the number of columns that we read. And then we have 8,400 loci, 16,000 alleles, and then the size is 11.5 mega base pairs. So this is like the basic content. Genetic data has a lot of structure. So we have the table, which is like the first few columns that define parameters that were generated by the sequencing machine. Yeah. And then a lot of other stuff here. We are not going to go through all of them, but basically that. So now we can proceed to do a PCA. But the first thing you will need to do is to calculate allele frequencies for both groups. So for Stolothrisa and for Limnothrisa, you will have to calculate their allele frequencies. And you do that very simply by using this tab function, and then you pass in the genid object that we created. You set allele frequencies to true, and you average all along, so the mean. What else do you do? Okay. Once you have that, you can now use this function, which is in the Adegene, library, to create the principal components of your data. So you pass your data in. This is like the data frame, which is equal to our Stolothrisa genetic allele frequency. In R, if I click tab without doing anything, if I click tab on your computer, it gives you the options that you're supposed to enter in for that function, like this function here. So if I do tab, you see the first thing that pops up is the data. It requires a data frame of data. But if you are used to R for some time, you can as well delete this and it will still work. The first thing you enter is the data. Okay, where was I?